Hello. A very good morning to one and all. It gives me distinct honor and tremendous contentment to welcome you all to the 3 days National Symposium on Plant Health Management organized by the Department of Plant Pathology and Entomology, College of Agriculture, Navsari Agricultural University Campus, Bharuch. In today's inaugural session, on behalf of my institution and on my personal behalf, I would like to take this opportunity to welcome the Honorable Vice Chancellor of Navsari Agricultural University, Dr. Z.P. Patel Sir, and the Director of Research and Dean PG Studies, Dr. S.R. Chaudhary Sir. I welcome you, sir. Your presence will always be inspirational to us. Next, we also have with us Dean Faculty and the Dean Faculty and Principal, College of Agriculture, Navsari Agriculture University, Campus Bharuch, Dr. K.G. Patel Sir, who has always been the backbone of guiding and motivating us for any activity undertaken in this college. I take pleasure in welcoming you, sir. I also welcome all the principals of various colleges of agriculture in Gujarat and all over the country. I also take this opportune moment to welcome and special thanks to all the eminent speakers, the scientists, the researchers, faculty members, participants who have participated from all over the country and joined us in today's inaugural session. Lastly, thank you for being with us, and I hope this timely symposium will help in the exchange of ideas as well as will be a great learning experience. Over to R.R. Vagunde.
Thank you very much, Madam, for a nice and warmful welcome to all the participants and the dignitaries. So, respected Honorable Vice Chancellor of Navsari Agriculture University, Navsari, and patron of today's National Symposium program, Dr. Z.B. Patel Sir, respected Director of Research and Dean PGS, Dr. S.R. Chaudhary Sir, respected Dean of Agriculture, Navsari Agriculture University, Navsari, and Principal of College of Agriculture, Campus Bharuch, and President of today's program, Dr. K.G. Patel Sir, faculty members, students, scientists, colleagues, and all the participants from different parts of India. Good morning to all of you. The sharing of knowledge is a continued process for the development and upliftment of human being. When whole world is facing the problem of COVID-19, under such circumstances, it is a much useful to share the knowledge and ideas by online platform. As we know that India is an agriculture country and we are able to produce an adequate amount of some food grains, fruits, vegetables and legumes. The agricultural scientists, technology and research have sturdy potential to feed the increasing population of India and increase farmers' income. Every year, up to 40% of the global food crops are lost due to pest and diseases. This drives millions of the peoples into the hunger and severely affects agriculture, which the primary source of income for the poor rural communities. The solution of this loss is essential not only for improved farmer income, but also for food security of the world. More than 820 million people in the world are undernourished and daily it is increasing. To achieve the goal of zero hunger and no poverty, it is essential to take steps for a balanced plant health policy for a better future of human being. There is a famous quote by Mr. Bill Gates on food security that innovations that are guided by the smallholders farmers, adapted to the local circumstances, and sustained for the economy and environment will be necessary to ensure food security in the future. And the most sensational quote by the former US Secretary of State John Kerry that give the importance of the food security that food drives life and the struggle for food is the struggle for life. This makes hunger an economic issue, a national security issue, and without a doubt, a moral issue. Through innovation, we can help alleviate hunger and malnutrition today, but more than we can help fulfill our responsibility to tomorrow. And that shows the importance of the food security to the nations and the society. Recently, the outbreak of some invasive pests like fall armyworm, locust, and South American leaf miner Tuta absoluta produce a big challenge in front of entomologists. The nature is too smart for the work distribution. It maintains the law of balance by the observation of the blast disease into wheat crop. And not only engage the entomologists, but also the plant pathologists to reduce the yield due to the such invasive pests and the pathogens. Now the occurrence of the wheat blast increased from south to the some west part of the India. The main objective to organize this national symposium is to gather the expert of the plant protectionist under one platform to achieve the goal of food security. This is the big platform for the students, researchers, faculties, and agriculture entrepreneurs to share their ideas and explore their scientific knowledge for the betterment of farmers and to achieve the goal of food security. In this national symposium, total 168 presentation including oral plus poster will be presented by the students and faculties of the different state agriculture university, central agriculture university, ICR institutes and others private research lab also, and 6 lead lectures by the renowned personality of the respective field. Out of 6, 3 will be delivered from the foreign experts to give the guidance and motivation of these young brilliant students for the plant protection. Now I request Dr. D.M. Patak sir for the introduction of today's function. D.M. Patak sir.
Honorable Vice Chancellor of Navsari Agriculture University and patron of today's function, Dr. J.P. Patel sir, Dean, College of Agriculture and President of today's function, Dr. K.G. Patel sir, and all the participants. A very winter good morning to one and all present here virtually. At the inaugural session of 3 days online national symposium with an international speaker organized by Department of Plant Pathology and Entomology, College of Agriculture, NAU campus, Bharuch. Bharuch is a land of Lord Parsuram, Bhrigu Rishi, and holy river Narmada. Bharuch is remembered strongly in the field of political, spiritual, historical, and now this college get name and fame in academic field in very short time. Let me introduce dignitary of today's function, but before that, I would like to present a शायरी related to national as well as globally COVID-19 situation. गुजर जाएगा, मुश्किल बहुत है, मगर वक्त ही तो है, जिंदा रहने का ये जो जज्बा है, फिर उभर आएगा, गुजर जाएगा. माना रात काली है, भयावह है, गहराई है, कई घबराए है, सदमे भी है, मगर यकीन रख, ये बस लम्हा है, 2 पल में बिखर जाएगा, गुजर जाएगा. I am going to tell you of today's function. He is committed to this college, sent person concentrate on his vision and mission for the development of this college. Due to his 24/7 working pattern, this college is get a name and fame in the field of academic, sports, and cultural. I would like to invite.
Hello.
Yes, sir. You are audible.
I'm audible. Okay.
Yeah, sir. Carry on.
Okay. Good morning, everybody. International Symposium on Plant Health Management to be held at College of Agriculture, Bharuch during 2nd to 4th November 2020. So today with us Dr. J.P. Patel, Vice Chancellor, NAU Navsari, Director of Research, Dr. S.R. Chaudhary, and it is my immense pleasure to be here among learned faculty members, students, and stakeholders remain present through online mode from different states of the country in 3-day national symposium on plant health management of the country in 3 days. So, dear friends, it is my great pleasure to be with you. And today, I am sharing some knowledge regarding plant health management of the country. Dear friends, plants are essential in all the accounts with the human, because 80% of the food are available through the plants on the earth. None of the animals, including human beings, can survive without the plants, because plants is very important for the human life. It provide clean air, water, and food. That is very important parts of the human survive on the earth. So number of the veg plants are very useful to the human being. Among them, the plants are providing sources of food, clothing, furniture, shelters, medicines, because number of the medicines are available from the plant. It may be allopathic or Ayurvedic or many things, and the biofuel, that is biodiesel, like that, electricity from waste management, waste product, natural pesticide that is available from the plants also, rubber, plastics, flowers, we get fragrance from the flowers also, and plants are also provide environment support, and they are provide economic base of the number of the countries also, and so a lot of contribution of the plants to the world is there. So there is a plants is also very important for the nature point of view, because the every food chain plants have a first position. Number of the food cycles, food grades are there. Each and every phase plants give a support for the food chains, and they provide the food chain gaseous balance, prevent soil erosion, ecological balance, because majority of the country has a base of agriculture. Even we talk about India, our India is also agriculture-based country. So it provides the support rainfall, maintain soil erosion, fertility, and shelter for the birds and animals also. Now, nowadays we are facing a problem of the environment. Pollution problems are also there. So plants are very important to clean the environment also, because they reduce the pollution like the carbon dioxide, CO, and sulfur dioxide also. Because number of the diseases to the human being or any microorganisms are related with the air pollutions, because it may be cancer, heart attack, asthma. So many diseases are due to the causing bad effect of the environment. So plants are very important to clean the environment also. Now we are talking about the food web. The, as we say that the plants is very important part that occupy first position in all the food web. It may be herbivorous, omnivorous, or it may be carnivorous, because the plants is getting a energy from the sun. That is, if we put at 100%, then each and every level, only 10% energy used by the first level, second level, and third level. It may be 0.1% at third level, but the producer of the plant is 100%. So you see the importance of the plant, because it will not save the plants on the earth. Then each and every food cycle, that there will be a problem, because the plant account over 80% of the human food, and hence plants are essential for the food security also. So each and every level for the active and healthy life, plants are necessary. But as earlier told, that plants are facing a problem with number of the pest and disease, and that number of the pest and disease pose a threat to food security, because they can damage crops, thus reducing the availability and access to the food, increasing the cost of the foods. Because the nowadays the population is increasing day by day, and there is a problem of reduction in the yield also. So we have to save our crops in many ways. Now, in the current way, in the recent scenario, number of the examples are there. Pest and disease they have threatened to agriculture crops also. In looking to the 1845, the potato leaf blight, it is a very good example that in the Central America, the Irish famine was there due to the disease, that is potato leaf blight, and the number of the peoples, that is 1 million peoples, were killed due to the famine, and more than 1 million people have left their motherland due to the severity of the potato leaf blight. Similarly, after the Second World War, there is a severe outbreak due to the brown spot of the paddy, that is caused by Helminthosporium oryzae. They destroyed 50% to 90% crops in the West Bengal, and millions of people have died due to the famine. It may be 2 million people were died. Now, in the current level, at the 2015, there was a new plant pest and disease threats staple crop around the world, and in that level, the Colorado potato beetle is very important to take the potato crops. And due to that severity and due to that importance, in China, they have started the potato as a staple food strategy, launched 2015. But the severe problem of the potato Colorado beetle, the total crop was failed in the China, and they have a loss of 3.2 million USD annually. So this was a great loss in the China also. Now in African country, you can see that in Tanzania, in Congo, they are having a problem of fissure and wilt for the banana, and they caused the Panama disease. So they at least the crop was failed, and there was a famine in the Africa also, and the total number of the people's economy lie on the banana. So they have a loss regarding the Panama disease. Actually, in Tanzania, the people were purchased 40 to 70 kg banana per month also. Today, due to the increasing global trade and changing climate, plant pest and disease pose a greater threat to the food security also. Because as we know that due to the insect pest, around 40% loss in the crops. Recently, there was a fruit fly attack was there, that is Bactocera dorsalis. Recently, native Asia, but around the 65 countries, there is a threat due to the fruit fly, and there was a problem of the guava, mango, and avocado and banana for the export also. India have also faced a lot of problems with the fruit fly also. Similarly, for the red palm beetle, there was a tremendous loss in date palm. So there's a food security happening in the oasis, the New East and Mediterranean basis. So looking to this, the plants are very important in the human life also. So the nationally and internationally, number of the organizations are working to save the plants. So here, one international plant protection convention was established in 1952, recognized, and later it was recognized by the WTO with the agreement of sanitary and phytosanitary measures. Because without the sanitary and phytosanitary measures, one cannot transport the plant from one country to another. Because knowingly, unknowingly, they can transport the disease, that insect pest, from one place to another area. So there are so many legal things are attached to save the crops. And the more awareness and attention to be needed for the plant health and management is essential. In India also, the NPPTI Hyderabad have initiated some step regarding the plant health management. So nowadays there is a need to alert the people for the indiscriminate use of the pesticide to save the environment. So plant are also very important. Looking into these things, our organizing secretary, Dr. J.J. Patel, and Dr. D.M. Pathak, and his co-organizing secretary, Dr. R.R. Vagunde, and the team of the Bharuch had think forwards, and they have going to start the National Symposium on plant health management. There are a number of the themes they have included in this symposium because they are very important themes. That is biofertilizers, that is product demand, post-plant pathogen interaction, biocontrol, then IPM, IDM, endophytic, microbes, molecular techniques, and population dynamics. These are all very important aspects, all important themes to justify this national symposium. And they have also selected number of the speakers from the lead paper. Among the 6 lead papers, 3 from the out of the country. They are very renowned scientists. So definitely this symposium will be very useful for the fellow participants, that our brilliant students, our faculty members, stakeholders, and those who are directly or indirectly connected with the agriculture or plant health management. So definitely this symposium will be very useful. And after the celebration of the 3 days, we'll come out with the truthful solutions and some suggestions for the policymakers also. So again, I congratulate and good wish to both the organized secretary, Dr. J.J. Patel, D.M. Pathak, and R.R. Vagunde. Again, with this word, I bless and good luck. Thank you.
Thank you very much, Dr. K.G. Patel, sir, Dean of College of Agriculture and President of today's function, for his motivational speech. Sir, your words definitely motivate students, faculty, scientists, and farming community. You are committed for this college and your kindness for the faculty. So I will quote one quotation of Mark Twain. Kindness is a language which the deaf can hear and blind can see. Thank you again, Dr. K.G. Patel, sir. Patron of today's function and Honorable Vice Chancellor of Navsari Agriculture University, Dr. Z.P. Patel, sir, is unable to join with us due to social emergency at early morning. But he sent us a message for a grand success of this 3 days National Symposium. So at the end of this inaugural session, I would like to invite Dr. J.J. Patel, Organizing Secretary of this Symposium, for place a vote of thanks. Because हर दोस्त से बात करना फितरत है हमारी, हर दोस्त खुश रहे हसरत है हमारी, कोई हमें याद करे कि ना करे, पर सबको याद करना आदत है हमारी. Dr. J.J. Patel.
Okay, thank you, Dr. Pathak, sir. Now, at the end of the annual inauguration session of National Symposium on Plant Health Management, myself, Dr. J.J. Patel, is a professor, Department of Entomology, College of Bharuch, and Organizing Secretary of this National Symposium, here for vote of thanks. respected Dr. J.P. Patel, sir, Honorable Vice Chancellor, NAU Navsari, and the patron of this symposium. Respected Dr. K.G. Patel, sir, Dean, Faculty of Agriculture, and the President of this symposium. Our most valued invited speakers, senior professors and scientists, colleagues, and participants across the nation. It is a great pleasure and honor to privilege for me to propose a vote of thanks to all who contributed and participated for making this National Symposium such a resounding success. I avail this opportunity to express my deep sense of revenge, gratitude, in-depthness, and heartfelt respect to the Honorable Vice Chancellor, Navsari Agriculture University, Navsari, and patron of this National Symposium, Dr. J.P. Patel, sir. In his message, he emphasized the use of useful microorganisms and natural resources for the management of pest and disease in agriculture crops. As a devotee personality of plant protection, you are yourself is one of the inspiration for us. Thank you, sir. I am very thankful to the Director of Research and Dean, PG Studies, Navsari Agriculture University, Navsari, Dr. S.R. Chaudhary, sir, for kind permission and approval of the National Symposium, College of Agriculture, Bharuch. I am also great much thankful to Dean and Principal, College of Agriculture, Bharuch, and President of this National Symposium, Dr. K.G. Patel, sir, for his sustained and valuable guidance, constructive suggestions, unfailing patience, understanding, and wholehearted cooperation rendered throughout the preparation of the symposium. I would remain indebted forever. He has given some examples of epiphytotic pest and disease that cause economic losses to the different countries. He has also emphasized on phytosanitary measures, then curative measures, and also the use of strategies of integrated pest and disease management. As a leader of College in Bharuch, you have always inspired and motivated us for getting various difficult achievements. Thank you, sir. My sincere thanks to Dr. Alok Statistics and in charge of IT cell, Bharuch, for his painful support for arranging all IT facilities. I am also thankful to the whole committee of this National Symposium, the senior professors, and my colleagues of this College of Agriculture, NAU, to all the participants who have registered in this National Symposium. I am thankful to all the personalities who have rendered directly or indirectly in this National Symposium. Thank you all. I would like to hand over the virtual platform to Dr. R.R. Vagunde, Assistant Professor, Plant Pathology, for onward technical program. Thank you.
Thank you. Thank you very much, Dr. J.J. Patel, sir, for a vote of thanks. Now, in continuation with our program, now I request Dr. Pranav Datta, sir, and Dr. P. Tamuli for take the charge as a nanotechnology session chairman and co-chairman. Chairman Dr. Pranav Datta, sir, and co-chairman Dr. T. Timuli, sir. We are going to start the nanotechnological session, and I request to chair Dr. Dr. Pranav Datta, sir, co-chairman, Dr. Timuli, sir, and Patidar, sir, and the Singh, sir, for as a reporter, sir.
Hello.
Yes, sir, you are audible. Just a minute. Now, before start this presentation, I request all the participants, those who are giving to the presentation in nanotechnological session, be ready for their presentation. Today's this nanotechnological session theme, Chairman Dr. Pranav Datta. Pranav Datta, sir, is the Associate Professor at Central Agriculture University and completed his PhD in Biological Control and postdoctorate at UK on the nanotechnology. and he established the international standard nanotechnological lab at Assam Agriculture University, Jorat, and trained many faculty, scientists, and premier institutes. And pioneer worker in the nanotechnology in India for the first time in the northeastern region, started work on the application of the science in this agriculture. He published more than 100 papers, authored 12 books, and handled 14 national and international projects. And he commercialized 4 bioformulation products also. The co-chairman, Dr. Temuli, is the associate professor and head in PG Department of Botany, Darang College, Guwahati University, Tezpur. And he completed his PhD in microbial ecology from Northeast Institute of Science and Technology, CSIR, Jorat, Dibrugarh University. And he take the postdoctoral training on metagenomics at University of New South Wales, Sydney, Australia, as a DBT Overseas Associate. He is a recipient of the Insta Teacher Award 2020, and he awarded by the Indian National Science Academy, New Delhi.
Indian National
Now
Science.
coming to the, I request Dr. Ullas Kadam. The next speaker is.
Kadam આવે ને? Don't speak it. ખબર નથી પરથી જે હોય તો આપણે હાજરી પુરાવીને કામ કરો. આપણે હાજરી પુરાવીને કામ કરો.
Now I request Dr. Ullas Kadam sir for his presentation on the RNA nanotechnology for plant viral disease management. Dr. Ullas Kadam sir, he completed his MS in chemistry and biological chemistry from the Purdue University, USA, PhD in biological engineering and technology from the Purdue University, USA in 2014, and currently he is the Humboldt Research Fellow at Max Planck Institute for Molecular Plant Physiology, Potsdam, Germany. And he has been awarded a Young Scientist, SCRB DST Government of India, and he has a very well-known publication in the peer-reviewed journal like the Current Genomics, Nature Plants, Plant Physiology and the Biochemistry, Journal of Biochemistry and Biotechnology, Analytical Biochemistry, Plant Biotechnology, means mostly highly impacted factor journal. He has the publications, and he has a patent also in the use of the circular RNAs with the tRNA like sequence containing 5-MC modified nucleotides to the include efficient RNA silencing in Arabidopsis. And he has received a research grant from the many institute, and the most important thing, he has been awarded by Bisland Doctorate Dissertation Fellowship in 2013 and 14 from Graduate School, Purdue University, USA. Awarded by the SETA Teaching Award for 2012 and 13 by the Graduate School at Purdue University. And he is a winner of the Magoon Award for the Excellence in the Teaching for 2013. It is one of the top award by the College of Engineering, Purdue University of Graduate Student. And he has been nominated by Purdue for International Graduate Student Fellowship also. And he is a reviewer also, journal, Current Science Journal, and the membership of the so many society. Now I request Dr. Ullas Kadam sir to deliver a lecture on RNA nanotechnology for plant viral disease management lecture. Now over to Dr. Ullas Kadam sir. Sir, now the platform is yours.
Just a minute.
No problem, sir. Take your time.
I think somebody else is sharing the screen, so I am not able to share. It shows that only one participant can share. Hello.
Now check the sir. Now check. Now check it.
Okay. Okay, I think now it's done. Good morning, everyone. And today, I would like to thank the organizing committee for giving me this opportunity and inviting for delivering this lecture to this national symposium. And especially, I would like to thank Dr. Rajesh Wagunde for contacting me in this regard. So, I'm audible?
Yes, sir. You are audible.
Okay, okay. So now I'll start with my presentation. So in this presentation, I'm going to talk about mostly RNA nanotechnology and its use for disease management, and that is specifically diagnostics tools. There are other applications also we have developed, but they are uh, under the process of getting patent. So that's why we are not disclosing the later part, that is using RNA nanotechnology for RNA-I or RNA interference in especially viral disease management. So in this lecture, only I'm touching the surface or the first part, that is the detection part, using a technique called dynamic light scattering. So before I go into the details, briefly, I want to give an overview of the gold nanoparticles. So gold nanoparticles, their behavior, the behavior of gold molecule or gold particles is completely different than the material scale behavior of the gold. So at nanoscale, it has very unique optical properties, as well as the surface chemistry and the surface area, everything changes drastically. And because of these properties, also it has a very less biotoxicity. So all these properties makes it a very good candidate for biological applications, especially diagnostics and delivery of nanomaterials into living tissue. And the so if you modify, or if you the surface functionalize these gold nanoparticles, they can be used in a number of ways. So first, the simple way is they can be used using colorimetric detection, just a simple UV vis spectrophotometer. And if we also have another instruments like which can measure the electrochemical potential, then this gold nanoparticle can be functionalized and measured for change in the current or potential based on the concentration or modifications, surface modifications. And most commonly used approach is the using of spectroscopic or fluorometric. In spectroscopic analysis, which is I'm presenting today, is like a dynamic light scattering or other spectroscopic methods, where gold nanoparticles, when they come in contact with the target molecule or the bioanalyte, they give a different spectroscopic spectra, and this spectroscopic spectra can be analyzed, and we can get the relevant information after analysis. Similarly, for fluorometric assays, so gold nanoparticle can act as a quencher, and if a fluorophore is in uh, contact or in a close area with the gold nanoparticle, then this fluorophore do not give any signal. But if analyte replaces that fluorophore, then this fluorophore is free and it is ready to give the signal. And this readout can be used to measure the signal presence of a bacteria or a virus or any microorganism, or some surface modifications or enzymes. Any biomarker can be used in this fashion. And so this gold nanoparticle in diagnostics as well as delivery has boomed in recent past, like recent last 2 decades, or especially in previous decade. And so the technique that I'm emphasizing here for this use is a very simple technique, and it has a very simple principle. It is based on two principles. That is, the first is the particles in solution behave in Brownian motion. or a random motion. So this is the guiding principle for this dynamic light scattering. So when the particles they are moving in a random motion or in Brownian motion, this light scatters these particles, and this is recorded. And this recording, you can expose these particles to a laser beam of say 675 nanometer or any specific length, and this can be used to measure the diameter or the oligomerization of this particle. And the second behavior, the second guiding principle or the second condition for application of this technology is it is assumption that the particles that are under this study, they are in a spheroidal or spherical form. So so if you have a particle which is or if you have a molecule which behave in a spherical fashion, and they are soluble in a liquid solution, then you can use this technique very easily. And there are many advantages of this technique. It is very accurate, reliable, and it can be reproducible in any condition. You can just give these conditions to any person in different lab or different country, and they can just use it without much modification. The second thing is the the compounds or the biological material that you are going to analyze. It doesn't need much preparation, so you can just, it needs very simple preparation. And the mean size, to calculate the mean size of this particular biomolecule or a nanoparticle, it needs just knowledge of the viscosity of the liquid. So what kind of the liquid is it water based, or it is emulsion, or it is oil based? So all just these simple things are essential to calculate this. And then you can calculate the concentration or the size of the particle, or the distribution of the particle size of a very high concentrated, even a turbid samples like a mud, or this kind of samples can be analyzed using this method. And this dynamic light scattering can be also used for the detection of sub nanometers particles. And this technique, this particular method is highly and fully automated, and it is very cheap actually. So we don't, without much preparation, this technique can be readily used for the diagnostics of agriculture or agriculture related biomolecules or pathogens or food waste, food poisoning kind of many things. So next, I would like to talk about the biological issue at the hand. And in this one, especially uh, the problem we are dealing with in this presentation is the Pigeonpea sterility mosaic disease. So the Pigeonpea sterility mosaic disease, it is caused by a virus, and this disease has multi symptoms. Like it shows, initially it shows the mosaic spots on the leaves, and then later infection stages shows reduced leaf area or leaf size, thick leaves, or even short internodes. Uh, this so And after this, after a long period of time, then the plants become sterile. That is, it shows no symptom of flowering. Plants do not mature, and they show only vegetative growth. And this is a very late stage, and at this stage farmers cannot do anything. Just uh, they just can throw the uproot the plants and throw it away. So this leads to huge losses in the field, because the plant actual agriculture produce comes from the flowering and the seeds and everything. And the plant, if they don't flower, the plant is useless to a farmer. So that is why it causes very high economic losses. And the next thing is, these plants the affected plants show stunted growth or dwarfism, and they have a bushy appearance. And this particular disease is caused by a Pigeonpea sterility mosaic virus, and this is an RNA virus. It belongs to Emera virus group. And earlier it was considered as it was carrying 4 or 5 RNA genomes, since it is a multi-partite virus. So a multi-partite virus carries more than one genome, and in this case, this particular virus, it is found that it can survive, or it can replicate and cause disease even with 4 RNA segments, or it can carry up to 6 RNA segments, and that I'm going to show in later parts what are the functions of these RNA segments, and which RNA segments we have targeted for the silencing as well as detection. And once this disease is occurred, then the plant shows up to 90% of the yield losses, or in most cases total yield loss, because there is no flowering. And the only problem in this plant is that early detection measures or early detection technology is not available for the disease management, and that is the best way for the control of this disease. If we can detect the disease at early stage, then we can avoid, or we can uproot, and we can maybe replant some plants there. And this disease, many times physiologically it shows misleading symptoms like nutrient deficiency, and it is labeled as a nutrient deficiency. And people are sometimes farmers are given suggestions to give fertilizer dose or micronutrient doses, and this adds to the cost of cultivation, and then you have no returns on this cost. And this particular disease is transmitted by a biological vector called Escherichia coli, and that here you can see the scanning electron microscope image of this particular mite, which transmit the virus. Now I would like to go more into the genetics of this particular virus. And this, as I said, this virus can have 4 or 6 RNA segments in it. In between 4 to 6, it can also have 5 segments, and these segments have different roles. And the RNA segments 1 to 4 have been identified with a particular role, but the RNA segment number RNA 5 and 6, they have unknown roles, but these RNAs or these genomes are still there. Maybe somebody will in future will identify the role of this RNA segment 5 and 6. So here you can see this RNA 1, which has a excuse me, which has a different roles. It has also polymerase activity. It carries the genomic RNA. It has the endonuclease domain. So this domain is essential for the infection and establishing the infection. Then it also codes for nucleoprotein and this whole nucleocapsid. Inside nucleocapsid, you will have different RNA segments, and that I would like to show it in this figure here. The RNA segment 1, which is known to code for a protein. And here actually I just want to show the in the lower part you see the nucleotide sequence, and in the upper part you see the protein sequence, or the representative schematic diagram of the protein that it codes for. So in the PPSMV1 genome, you have the 4 segments, and this 4 segment codes for different proteins. And the first one codes for RDRP. So RDRP is the RNA directed RNA polymerase. So this polymerase helps in the replication of the RNA genome, as well as making the copies of other genomes in it. So this GP is a glycoprotein. So the glycoprotein helps in anchoring. It forms an anchor with the membrane of the Pigeonpea cells or plant cells. So the cell membranes, it needs some anchor to attack or to attach to this plant cell, and that is the function of this RNA2. It forms around 74 kilodalton glycoprotein. Then another RNA segment, that is it forms a nucleocapsid protein, and this helps in covering the whole genome using this number 3 segment. And the RNA4, which is present in this total RNA, is movement protein. So this movement protein helps once the infection is established. This particular RNA helps in coding for the movement protein, and this movement protein helps in transmitting throughout the cell, throughout the plant, through the plasmodesmata and other vascular tissues. So in the right hand side you see this PPSMV2 genome. So it has a similar genome, but there are uh, two additional RNA segments, RNA5 and RNA6. These two RNA segments, like we still don't know what are the function of these two particular RNAs, but they code for a protein, and maybe in later in future somebody will identify its functions. So for this analysis, we have focused mostly on RNA1 and RNA2, and that success and everything I can provide if somebody wants to do that kind of study, or the primers and everything. Now I would like to go into the details of the economic significance of the Pigeonpea in India. In India, almost 3.6 million hectares of land is under cultivation under the Pigeonpea crop. Around 2.4 million metric tons of grains is produced by the Pigeonpea, and due to this particular virus, almost 1 billion per annum losses are incurred. And especially Marathwada region is the leading producer of the Pigeonpea, and we have a research station specially dedicated for in Marathwada. More than 90% cultivation of the world or global cultivation is located or present in India. The next is uh, what is the nutritious significance or uses of the pigeon pea. So pigeon pea, you all know that pigeon pea is an important edible pulse crop. It has many medicinal values, such as you can give a soup or dal soup to a patient, or some kids who are suffering from viral infections. It also has a very high protein content, and this is important for the vegetarian diet. It produces a lot of it supplies a lot of proteins for vegetarian people. Additionally, it is a rich source of antioxidants and isoflavonoids. So it helps in boosting the immunity, and that is why it has medicinal value. And in agriculture point of view, or a soil scientist point of view, this helps in symbiotic nitrogen fixation, so it can be rotated with other cereal cereal crops to boost the soil fertility or the soil nutritious value. And in most cases, this crop is a drought resistance, so you don't need to water or irrigate these plants. So that is why it is a very important crop for the dryland agriculture or rain-fed agriculture. Now I would like to go into the how we did this, how like how we collected the samples and where we sampled this. So most of the sampling area was belong to the Marathwada region, and that 7 districts of the Marathwada, 7 or 8 districts of the Marathwada. Then after collection of the samples, we extracted these RNAs, then amplified these RNAs using PCR fragments, PCR for fragment 1 initially, and then we also amplified the, initially we had only a sequence of the fragment 1, but later on it was a whole genome sequence. So we also have other fragments sequenced, and all these fragments are cloned, and they are stored in our lab. Then we confirmed these sequences by the sequencing, and then we prepared our primers and probes for those particular sequences. Then after isolation of this RNA, we use the specific primers for the RT-PCR confirmation, so that we have a very specific genome or very specific specific RNA targets which are affecting the Pigeonpea plant, and they are specifically belong to this sterility mosaic virus. So that is why we confirmed these results. These are only coming from the plant Pigeonpea sterility mosaic virus viral. They are part of this viral RNA components and nothing else. And that is that. For that we have a specific primers designed, and we checked it, and we have this presence of this highly specific band, and there is no non-specific bands or no non-specific amplification from these results. The next, for this, applying the nanotechnology, once we have the biological material, that is the RNA particles, virus, and everything with us, then we moved on to the using nanotechnology. And for that we prepared the gold nanoparticles. And preparation of gold nanoparticles, for preparation we use the chemical synthesis method, which is also called sodium citrate reduction method. So in this, what we do is we use a gold chloride solution, or we order a gold chloride powder, and then prepare a solution of 0.1%, and then to this we add a sodium citrate buffer. And once this is added, we boil it. So this is how it looks at 0 second. And once it starts boiling, after 30 seconds it starts to change color to a bluish color. And after 60 seconds of boiling, this color becomes red. And after 120, or after 1 minute, it becomes bright red. And this can be confirmed by um, by by obtaining UV spectra of gold nanoparticles. And here you can do a whole UV scan of a whole range from 200 nanometer to 1000 nanometer or 800 nanometer, depending on the instrument you have. And in this one, you will see the blue line in which we have the gold nanoparticle update. So that is 120 after 120 seconds. And it shows a very very particular peak, or a very unique peak that belongs to 20 nanometer particles. And in light green you see the first one, the 0 seconds, and there is almost no non-significant peak observed. So this shows that gold nanoparticles are of uniform size. And if they are not uniform size, then here you will see a peak which is very broad, not so narrow. And that means you have particle size distribution of very high, like maybe from 10 nanometer to 100 nanometer. But this peak shows that particles are of uniform size, and they are around 20 nanometer. And one additional thing I want to say is, here you can make a particle of any size, maybe 10 nanometer or 100 nanometer or 50 nanometer. For that, you have to just play with the adjusting the concentration of gold chloride along with the sodium citrate. So if you change this concentration of sodium citrate or gold chloride, like if you increase the concentration of sodium citrate, then you can change the size of the nanoparticle. And this is very useful if you want to use or try what is the effect of different size nanoparticles in detection or a delivery of a biomolecule inside plant cell. The next thing is, we once we have these gold nanoparticles, we functionalize these gold nanoparticles. The functionalization means that, okay, here you see this is a gold nanoparticle, and the functionalization means you add another biomolecule onto the surface of this gold nanoparticle. And here you see gold nanoparticle, it is citrate capped, means since it is prepared in the citrate solution, on the surface there will be citrate capping, that citrate molecules will be present, and it will be negatively charged. And this gold nanoparticle, since it is made up of a gold molecule, this gold has a tendency of forming a covalent bond with sulfur, and that is called a gold thiol interaction. And excuse me, this gold thiol covalent bond is very useful in making diagnostic tools or delivering this DNA or RNA or a protein through this gold thiol interaction. And you can also cover this surface of the gold nanoparticle using PEG or thiolated PEG molecules or thiolated OEG molecules. And these thiolated OEG molecules, they will cover this surface and will allow only a small number of DNA or RNA to surface functionalize, and that you can check using two simple methods. One is the obtaining UV with spectra. So on the left side you will see the bare gold nanoparticle or the naked gold nanoparticle, which are just sided capped and which doesn't have any molecule on it. They have a small spectra and little bit broad spectra. And once they are functionalized with the DNA. So here we used the thiol modified single stranded DNA. Uh, so this DNA, you see the increase in the peak as well as the shift. This is called red shift, means its size has increased because of the covering of the gold nanoparticle around the surface. And the next method you can also use is, here I am showing the TEM images, that is a transmission electron microscope. And in transmission electron microscope, you see the on the left hand side this gold nanoparticle, they are bare, they don't have any covering around it. Whereas on the right hand side, they are covered with the single stranded DNA which are thiol modified, and you see a small gap. That is because of this single stranded DNA. And so this confirms that our probes which carry different molecules, two different single stranded DNAs, they are successfully modified or successfully functionalized. And once these probes are successfully modified or functionalized, they need to be purified or characterized using agarose gel electrophoresis. So here I will show you different 4 or 5 different types of gold nanoparticles that can be identified using agarose gel electrophoresis. The first one is the only citrate cap gold molecule. So since these molecules have only gold nanoparticle on it, they will run the fast. And you will see here that there is a band of which belongs to gold nanoparticle which are citrate capped, and then citrate capped gold nanoparticle modified with thiol OGO. So they will be present here. Okay, so thiol OEG, that is it is kind of PEG. Okay, so the next one is with a single stranded so this band here is they are carrying with a single stranded DNA. And the fourth band here, you see it belongs to the particles which carry two different single stranded DNA. And the last band, which will be the of highest molecular weight, that is of a gold nanoparticle carrying three different single stranded DNAs. And here we have gold nanoparticle which can be functionalized with 40 nucleotide or 80 nucleotides. And after functionalizing this nanoparticle, the nanoparticles weight as well as the movement is increased and the movement becomes slow. And that is why you can separate these molecules using the agarose gel electrophoresis. So once you identify in this one the molecules which are which carry this single stranded DNA and which don't carry any DNA or a higher order DNA, you can separate this, and then you can use only this only nanoparticles which are carrying single stranded DNA and use them to hybridize. So here this is the our characteristics of forming nanodimers or gold nanoparticle dimers. So in this formation of gold nanoparticle dimers, so you have one gold nanoparticle attached or functionalized with a PS, that is a probing strand, other with a capture strand CS, and there is a linker in between these two nanoparticles, and that linker is the target strand, and this target strand is the similar sequence which is similar to the Pigeonpea sterility mosaic virus components like RNA1, RNA2, and in this one we have used only two RNA sequences, RNA1 and RNA2, because of their significance in the disease establishment. So here also you can separate these nanodimers from the unhybridized gold nanoparticles. So the high, this dark band here you see, which is running faster, is of unhybridized gold nanoparticles, and the second band, this is of your interest, and you can purify this band by gel extraction method, and that can be used for the dynamic light scattering. The next thing that we want to see is now now our dimers are ready. So these dimers are of this format. So we want to test the whether these dimers produce a specific results, and that specific results, whether we can quantify it and detect it, so that we should be able to detect the Pigeonpea virus from the lysets or from the isolation. So here you have a single nanoprobe and single nanoprobes. Okay, so when these single nanoprobes, which are unhybridized, when you subject them to this dynamic light scattering, they will show, they will give you a size or particle size distribution, and this particle size distribution will be very uniform, and this is around 20 nanometer, and that is also confirmed by the TEM, transmission electron monograph. And when you add a TS, that is a target strand, then it forms a dimer, and this dimer will show the shift, red shift in terms of the scattering or spectroscopic term. It will be red shifted, means the size, it will absorb the light towards higher nanometer, but in terms of the size, its size will also increase. Now earlier it was 20 nanometer particles, but upon forming dimers, its size has increased beyond 50 nanometer or sometimes 60 nanometer, and it's clear indication that using this dynamic light scattering, you can identify whether it is a monomer or a dimer, whether it is just a single nanoparticle, or whether it has hybridized with the target molecule. Once it is hybridized with the target molecule, then it is well and good, because that way you can quantify how much the infection is there, and this can be easily used for the diagnostic. Another thing is, you can also if you keep this reaction going on for longer duration, beyond 5 minutes, then the reaction will lead to forming multimer, okay, trimer, or higher oligomeric higher oligomeric particles. And here you can see the after 10 minutes or 15 minutes, these particles can form a trimer, and the trimers, in case of trimers, you will see a shift towards higher higher size, as well as or red shift, as well as you will see a small peak here because of the additional nanoparticle present there, and this can be attributed to trimer or higher order oligomers. So this particular detection shows that this method, dynamic light scattering, can be used specifically to detect target molecules or analyte of your desire. In our case, this virus, PPSMV, and we also tested the non-specificity of this method. So for that, what we did is we used the same probe strand and strand, which are functionalized with the gold nanoparticles, and instead of using the target sequences, we used non-target sequences. Okay, so if there is a non-target sequence, then what will happen is there is no dimers, no dimer is formed. Or even if you keep for higher or longer duration, like more than 10 minutes, 15 minutes, or half an hour, then also it doesn't form any trimers. So what happens is, if there is a non-specific infection, if there is no virus present, then you will see a no change or no shift in no change in the size, or no shift in the absorption of the light. So based on these two experiments, you can clearly say that the dynamic light scattering is very simple and very specific towards uh, the target, specific target, that is in our case the virus. Additionally, when you are developing any new technique or new technology or approach, anyone who works in this area, they they have to establish that the limit of detection. So what is the limit? Like anybody, like if you detect if you develop a new tool, then anybody will ask, what is the limit of your assay, or what is the limit of your technology, like how low it can detect. And the lower it can detect, the better it is for the diagnosis. And in our case, this dynamic light scattering, we show that it can detect up to 10 picomolar, or zero point 0.1 nanomolar. And it can also detect a higher concentration, but just for the sake of this presentation, we are showing only four different concentrations. And this detection is a very linear, like we can quantify based on this detection. And since this can detect with a so low concentration, that means this technology can be used for the detection of viruses at a very low concentration, at using very simple method. Next, I would like to summarize this presentation, that this present method, the dynamic light scattering based RNA detection, is highly specific, and it has a limit of detection which is a very low, around 10 picomolar. The procedure is simple, it doesn't need much preparation, and it is very rapid. Like in few minutes, 4 or 5 minutes, you can detect the you can detect in 4 or 5 minutes, you can detect this presence of virus. And this same method can be used for another biomolecules or pathogens. It can be used for fungal infections, or identifying bacterial infection, or any modifications in protein, or change in the physiological or metabolite conditions. In addition to this, as I told you previously, that we are also working on development of rapid RNA-based RNA-based and nano-based approaches for detection of viruses as well as other pathogens. And the same technology we have applied for controlling the PSMD disease. That is, we are controlling this viral disease using RNAi and gene silencing. And we don't want to tell the details yet, but we have very promising results of using gold nanoparticles and using them for the delivery of RNAi constructs, double stranded constructs into plant cells, and control the disease. And hopefully in future, we will get we are going to apply for the patent as well, and we will get the patent. With this, I would like to thank my collaborators and the supporters. The financial support I have received is from the SERB from India, and Alexander von Humboldt, especially my collaborators, Dr. Chauhan sir, Pragler, then Dr. Shelke, Suprasanna sir from BRC, and my old friend Nunli or Zongu from UT Austin, Texas. With this, I would like to open this presentation for discussions or any questions you might have. Hello.
Hello.
Yes.
Yes, sir. We have seen message in the chat box. Very nice lecture and innovative approaches. And as you already told that you are going to for this patent, so it's give the very good idea for the person who are the working in this area line of area. Datta sir, are you in the session, sir? Pranav Datta sir.
Yes, yes,
Sir,
Raju. Dr. Raju.
any question regarding this presentation?
Actually, I would like to thank Dr. Kadam for his appreciable presentation on RNA technology for plant viral disease management. And Dr. Kadam, I'm sure your talk will be very helpful for the scientists who work on application of the novel science that is nanotechnology, more particularly in the agriculture. And in your summary, you have mentioned that rapid nano-based kit, if it comes in the market and if it has been commercialized by your institute under leadership, it will definitely help in certification as well as quarantine program.
Yeah.
I once again congratulate you, Dr. Kadam,
Thank you, sir.
and all the best for your future
Sir,
days.
there is one question.
Oh, yes, please.
By the Dr. Chin Kapadia, faculty from Navsari Agriculture University. He is basically from the biotechnology department. Yes, Chintan Kapadia, sir.
Sir, this RNA are very small molecules. Okay,
Yes.
and there is a uracil. So there may be some non-specific binding.
Yeah.
So how will eliminate those non-specific binding from your studies when you take any kind of sample? And the RNA is very small molecule, like which you are going to target.
Yeah.
And when you isolate something from the cell, there are so many different RNAs present in the particular sample. So how will you eliminate particular things?
Yes, for this technology, what we rely on is the presence of the target sequence. So once you know the target sequence, okay, so you can obtain that particular target which is complementary to the target molecule. Okay, so if you know the genome sequence of the RNA, then you can ask company synthesize the complementary to that RNA sequence, and this complementary reaction is very specific. This complementary hybridization that we have tried, I have shown in the non-specific binding that if there is a if the target sequence is absent, or if you modify the target sequence, there is a very less or no specific binding. So this way you can avoid the non.
But sir, there is a chances of mutation with the RNA as they are very small molecules, and there is a uracil in the so if you develop a kind of nano-based kit, it's a very nice attempt. No issue, there's a very innovative attempt. But what will be the chances of again, like target may escape?
Yeah. So actually, if you ask about this particular technique, this may not identify the small mutations, like what we call is point mutations or just one nucleotide mutation. But there are other methods, the fluorophore based methods, or what do we call there is called like high melting temperature technique. Okay, so in space binding, and if there is just a change in one nucleotide, that can be detected using HMT, and for that you just need a fluorophore attached to the this. You can get a fluorophore modified nucleotides or fluorophore modified bases, and based on this just one change, one nucleotide change, this fluorophore signal will be different at a different temperature, and that can be measured, and that is how people also detect this SNPs, okay, single nucleotide. Other approaches for this purpose, but not this one, because this one is based on the size of the molecules. Okay.
Yeah. Thank you, sir.
Welcome. Yeah. Is any other question?
Hello.
Yes. Are there any other questions?
I think there is no any question from the participants.
Okay.
Thank you very much, Ullas Kadam sir, for a nice lecture. You
Welcome.
have there is a one question regarding by the Dr. Pooja Pandey. I think she is from the Anand Agriculture University faculty,
Yes.
and basically from she is a virologist, and she has asked one question regarding sir, can we have any idea about biosensor?
Yes. So this is also actually kind of biosensor. Okay. You can say this is a biosensor, and biosensors are just like for the biosensor or any sensors you want to detect, you have to have a knowledge of the particular target molecule, and that we called analyte. Okay. In terms of analytical chemistry. So if you know the characteristics of analyte or target molecule, then we can use uh, we can develop many biosensors. In my first slides also I have shown that we can use biosensors in a colorimetric detection. That is very simple, and that is what people use in now COVID-19 rapid antigen test. It is a biosensor. So just presence or absence of reaction, you can see the presence or absence of a COVID. And this is also used in pregnancy test and many things. So similar approaches we can use on food poisoning or this viral infections. And yeah, yes, there are a number of biosensors available, and we can develop our own biosensors based on your interest. So like what organism you study, and what particular crop or what particular food type you study. Based on that, we can design or develop these biosensors. Yes.
Thank you, sir. Actually, I want to know, it will be target specific, right?
Excuse me, can you repeat it? It's not really audible.
Target specific biosensor. We cannot generalize it. Can we,
Yes,
sir?
yes. Yes, we cannot generalize the, but the guide, like underlying principle is the same. And for target principle, we just need to know the more information about the target. Okay. So if it is a bacteria, you need to find out the biomarker present in that bacteria. What is the main gene that is present in that bacteria, and it is not present in any other organism or any other uh, for in the case of cancers. Okay. So there are a number of biomarkers for different cancer. There are also number of genes for viruses. In our case, in Pigeonpea sterility mosaic virus, we have this RNA1 and RNA2. These are very specific targets present in PPSMV only, and not in other viruses. So this can be this can be used as a biosensor for this particular, and it will be very specific biosensor.
Okay. Thank you, sir. Thank you.
And like if you want to discuss, we can also discuss later one-on-one.
Thank you, Ullas sir, and I will share your email ID, and if you permit, your contact number also.
Sure.
So those who are interested uh, for these any issues and the query regarding this presentation, so I will share your contact
Sure.
number and email ID to the concerned participants. Thank you very much, sir, for a nice lecture. Now
Thank you.
we are moving towards our next session regarding nanotechnological presentation by the different scientists, and this is the presentation for letter of appreciation from the College of Agriculture, Bharuch, to the sir for you. Now the participant who have submitted the presentation nanotechnological session. The list already we have shared in the WhatsApp group, and as per the list, Dr. Pranav Datta sir
Yes.
is the chairman of the session, as we know, and Dr. Dr. P. Tamoli sir. Sir, Tamoli sir. Sir, I think he.
Yes.
Yes, sir. Hello.
I'm present.
Okay. And Patida sir and Patak sir from the Central Agriculture University of Meghalaya. So they both will be acts as the reporter for this session. Okay. Now the first person. Now I will read out the sequence for the presentation for the this presentation. First three person. First is the Gitashri Das, second is the KR Padma, and the third one is the Jyoti Kumari. I request Gitashri Das to unmute. Hello, you are audible, Gitashri?
Yes, sir.
Are you going to share, or I will share from this side? Hello.
Hello, sir.
You will share or I will share the screen, your presentation?
Sir, I will share.
Okay, I will say you go for the presentation. Is it okay for you?
Yes, sir.
Okay, just a minute. Okay. Very good. Go ahead. Just a minute. Datta sir.
Yes.
And Tamuli sir. Hello. Sir, 7 minutes for the presentation. And now Gitari Das.
Yes.
You start your presentation.
Okay, sir.
Now Datta sir, the session is yours.
Okay, thank you, Rajesh.
Okay.
Good morning, everyone. Respected chairman, co-chairman, and all the participants present here. I am Gitashri Das. Today I am here to give a presentation on the topic if nanopriming with silver and zinc oxide nanoparticles showed positive effect on physiological parameters of chickpea seed with significant suppression of fusarium wilt incident of chickpea. We all know that seed is a basic and key input for agriculture. Of all the crops, pulses has an specific importance for the vegetarian population of our country. Sir? Yes, sir?
Please, you put your slide on presentation mode, and because your slide is moving, in the first slide, yeah, and put it in a slideshow mode.
Yes, sir.
Yes. Okay, now you can proceed.
We all know that seed is a basic and key input for agriculture. Of all the crops, pulses has specific importance for the vegetarian population of our country. Among the pulses, chickpea is an important pulse crop which is considered as the king of pulses. India produces about 150 million tons of food grain per year, but losses has remained static at 10%. The loss of food grain is increasing with increase of food production. The main reason for this is improper storage condition, which may reduce the physiological activity of the seed. So it is urgent need to increase the quality of the seed, more particularly in storage. Out of different methods, the priming is considered to be an important approach. For this study, the chickpea seeds were collected from from RARS Silongani, Assam, of variety JG16. For this, the two experiment was done. One to study the physiological parameters in storage condition, and and other one is to suppress the fusarium wheel by using nanopriming agent. For storage condition, seeds were treated with two nanoparticles, silver and zinc oxide, nanopriming agent containing solution alone and in combination with each other for 1 hour, 2 hour, and 6 hours. Then the seeds were seed dried till moisture reached to 9%. Then the prime seeds were packed in an HDPE semi-permeable bag and 9 months. Physiological parameters like seed germination percentage, speed of germination, shoot and root length, seedling fresh and dry weight, seedling vigor index were observed at 2 months of interval. After collecting the seeds, the germination percentage, infection percentage, and moisture content were determined as per ISTA protocol. These are the two nanoparticles that were taken from the Nanotechnology Laboratory from Department of Plant Pathology, Assam Agricultural University. These are the treatment combination for the storage condition. This germination percentage decreases with increase of storage condition. The seeds primed with zinc oxide nanoparticle for 1 hour. This was followed by seed primed with both silver nanoparticle and zinc oxide. Similar trend was also found in 2 hours and 6 hours of nanopriming seed. Among the 3 priming times, 6 hours was found best for 9 months of storage. Similar trend was recorded for the speed of germination. Then speed of germination, then germination index, root length, shoot length, seedling fresh weight, seedling dry weight, seedling vigor index. This is the summary table of 6-hour nanoprime seed for 9 months of storage. For pot experiments, seeds were treated with nanoprime solution for 6 hours and sowed in a fusarium inoculated pot. These are the treatment combination for pot experiment. The physiological parameters like seed germination percentage, shoot length, root length, number of branches, fresh and dry weight, seedling vigor index, and number of pods per plant, seeds per pod, seed yield per plant, and 1,000 seeds weights were recorded. This table shows the treatment for that is the seed primed with zinc oxide nanoparticles showed the highest physiological parameters of chickpea in suppression of wilt disease. This is a general view of the pot experiment. Here shows the effect of nanopriming on plant growth against fusarium wilt of chickpea. Here shows the root growth against fusarium wilt. This table shows that the seed production of the nanoprime seed. Here found the seed which were primed with zinc oxide nanoparticle showed the highest seed production. This was followed by the seed primed with both nanoprime zinc oxide and silver nanoparticle. Conclusion: seed germination percentage, germination index, speed of germination, seedling vigor index, fresh and dry weight of roots and shoots were observed decreasing with increasing storage time in all the treatment. Zinc oxide nanoparticle at the rate of 100 ppm showed the highest seedling fresh and dry weight. Nanopriming with both the zinc oxide and silver nanoparticle found next to the priming with zinc oxide nanoparticle for all the priming time and at all the storage period. Lowest seed germination in all the storage period for all the priming time in control. Out of the different priming time, 6 hours were found to be the best, which results in highest physiological parameters of chickpea at 9 months of storage. In pot experiment, highest percentage of seed germination was recorded, where seeds were primed with zinc oxide nanoparticle with reduced seedling infection, higher shoot length, root length, branches, fresh weight of root, dry weight of root, fresh weight of shoot, and dry weight of shoot was found in treatment 4, that is that seed primed with zinc oxide nanoparticle, followed by the seed primed with both the zinc oxide and silver nanoparticle. Seed primed with zinc oxide nanoparticle gave the highest seed production as compared to the seed treated with chemical fungicide and silver nanoparticle. Thank you, sir.
Thank you, Gitashri, for your nice presentation. So is there any question, very specific question, one or two? If not, we can proceed for the next presenter, that is Dr. K.R. Padma. K.R. Padma, are you?
I think she is not present, sir.
Okay, proceed for next presenter,
Yes,
Jyoti Kumari.
yes.
Jyoti Kumari, are you present?
Jyoti Kumari.
Jyoti?
Yes, sir. Yes, sir. I'm present, sir.
Just a minute.
Okay.
Sir, there is one question from Sunset Jani. Can you use any salt solution for priming control like AgNO3 and zinc chloride too? Just check in your chat box, sir. Jyoti Kumari.
Yes, sir.
Okay, shall we start?
Yes, sir. We can start, sir. Sir, how can I put it in play mode? Sir,
Slide.
its option is not coming.
Just say next. Hello.
Yes, sir.
Just say next when you want to go on to the next slide. Okay.
Okay. Thank you, sir. Thank you, sir.
Datta sir, ready?
Yes, but we should, Jyoti Kumari, you please
Yes, sir.
put your slide in slideshow mode.
Yes, sir.
Slideshow mode, okay?
Sir, actually, sir, I haven't shared the screen. The screen share is with management.
So, are you able to share the screen?
Sir, actually, my some problem issue is coming in my.
No problem. Just go ahead. Go ahead, Jyoti.
Okay, sir. Sir, can I start, sir?
Yes, you can start.
Good morning to respected chairman, co-chairman, dignities, and all participants over here. Myself, Jyoti Kumari, did my graduation in 2020, that is Prasad Central Agriculture University, Pusa. Sir, I did my internship program at Assam Agriculture University, Jorhat, under the guidance of respected Dr. Pranam sir. So here I am presenting on nanoparticle. When we start an experiment, 3 points must be clear, that is what to do, how to do, and why to do. Now let's start and find whatever I did. So my objective is synthesis of silver nanoparticle. Please bring objective. Yes, sir. Sir, my objective is synthesis of silver nanoparticle, its characterization at the end to check its efficacy against the fusarium auxilium. That is the answer of what I did. Overall objective is described. Sir, please put next slide. Sir, next after objective, sir. Okay, sir. Sir, in this, I followed a protocol as instructed by my guide, Dr. Pranam Datta sir, and guided by my senior, Dr. Pranjal Kaman sir. Sir, I used Trichoderma asperellum, which is a fungus, and I used the 7-day broth culture of this Trichoderma asperellum, and after centrifugation, I obtained the supernatant, which is further adjusted to a required pH, that is 10, as per protocol. So after addition of the aqueous solution of 1 mm AgNO3 solution, we put in a rotatory shaker for, sir, leave it. Yes, sir. Then we put it in a rotatory shaker for 5 days and maintain the dark environment of the shaker by wrapping the flask with a brown paper. Sir, next slide, sir. So next slide. Yes, sir. Sir, after 5 days, we analyzed the physiochemical characteristics of synthesized silver nanoparticle with the help of some instrument, which are UV spectrophotometer and Jetta sizer. Sir, here the two here it is the showing that synthesized silver nanoparticle that is ultrasonicated, and the other in the comparison, one is the reddish brown in color and other is the greenish in color. Sir, the greenish one is supernatant of trichoderma spirillum and the reddish brown is of synthesized silver nanoparticle is showing. Sir, next slide. Sir, next slide. Yes, sir. Sir, after the analysis, using the UV spectrophotometer and the Jetta sizer, one thing to mention here that the uniformity of particle size I used to maintain by using an ultrasonicator. While using this UV spectrophotometer, I obtained the peak at approximate 408 nanometer, and we are showing the graph is uniform, which means the particle size I obtained is uniform in nature. Using the Jetta sizer, I found the result of the peak at negative minus 30.9 millivolt. That is the data potential of the silver nanoparticle. Sir, next slide. Sir, next slide. So the overall method I explained and also showed the how I know that a silver nanoparticle has been synthesized. Now I check the efficacy. That is why I did the overall experiment. For checking the efficacy, I use the Fusarium oxysporum as per my experiment. Sir, we know that vascular fusarium wilt how adversely affected the crops, that is of wide range crops. For testing the efficacy of silver nanoparticle, I use the Agarwal method. For this, I use two PDF plates condition. One is control and other is treated. So next slide, please bring the next slide. Yes, sir. So for two conditions, one is control and other is treated. In treated, we pour the 100% AgNP concentration in a well and inoculum of Fusarium oxysporum at the center, and other is a normal sterilized condition I adopted of the PDF plates with Fusarium oxysporum inoculum at the center. So after the 8 days, we found this comparison. So bring the previous one. So bring the previous one, the table one, sir. Yes, sir. So after 8 days, we found this comparison. We checked the radial growth in both condition, that is in control or treated and control condition. We found the result that, the mycelial growth inhibition percent has been decreased in the treated one, but in control, it is comparatively it is higher. So we came to conclusion that the AGN can be considered the most influencing controlling metal against the one of the most damaging soil-borne pathogen, that is Fusarium wilt caused by the Fusarium oxysporum. This is difficult to even manage. That is, if by any other method. So, sir, bring the last one, sir. So conclusion. Sir, again next one. So next one at the end. Next. Yes, sir. So, at the present study indicates the reduced use of chemical fungicides ultimately may lead to the reduced pesticides on the environment. Our result supports the hypothesis that the silver nanoparticles suitable for the formulating new type of fungicide material. So, at the end, I am thanking everyone for listening patiently. So, that's it.
Thank you, Jyoti Kumari, for your presentation. But in your presentation, I have seen the character. You have to give more and more concentration in the characterization, especially to identify the functional group. Then, with the help of electron micrography, you have to determine the size, shape. Okay. Then crystalline pattern with the help of say PLS. Okay. So that is an important area to determine, so to characterize your nanoparticle. And further, you can do more work on mode of action, how these nanoparticles are acting on your targeted organism. And, and lastly, one more request I do have.
Yes, sir.
That dose, we have to standardize the dose of particular nanoparticle
Yes.
to see their efficacy against, to find out the best effective dose against
Yes,
the organisms. So
yes.
is there any specific one or two question they can raise, any participant they can raise? If not, we can proceed to next presenter, that is Dr. Sharodi Borua.
Sir, there is one question.
Okay.
Can you use any commercial fungicide and GNO3 effect on fungus in the chat box?
Yes. Sir, AZNO3 used as an inoculator. It is not a synthesized silver nanoparticle. Silver nanoparticle we synthesize using the AZNO3. So the question, sir, again repeat the question. I think must be there is confusion.
Is in the chat box. Can you use any commercial fungicides?
Can we use any commercial fungicide and AZNO3 effect on fungus? So I'm not clear about this.
Commercial fungicide we are using.
Yeah, we are using, so already we are using the commercial fungicide and AZNO3 combination. I'm not clear about that. Sir, we did an experiment on silver nanoparticle effect. We have AZNO3 is used in this synthesis.
As a sub, yes.
Yes, sir. So, any more question?
Okay, can we proceed for the next presenter?
Yes, sir.
Dr. Sharodi Borua. Sharodi Borua, are you present?
I think, sir, Parismita Bora 4 number, biogenic synthesis.
No, she is in poster session.
Poster presentation.
Okay.
Sharodi Borua.
She's
here.
Yes, she is present, sir. But Sarodhira, please unmute.
Hello.
Yes.
Am I listening?
You are audible.
Am I audible?
Yeah.
Okay. Thank you. A very good morning to respected chairman and one and all present in this virtual seminar. Myself, Dr. Sarodi Borua from Krishi Vigyan Kendra Tinsukia, Assam Agriculture University. Today, I am here to present our research paper on the topic of biologically mediated nanocytogen-based bioformulation of trichoderma aspergillum as an effective seed-treating agent against thorn plant pathogens.
Any technical issue, Sarodi?
Sharodi, any issues?
Sir, am I audible?
Yeah, you are audible. Share your screen.
Is it okay, sir?
Okay, you just share your screen.
Is it okay, sir?
Yes, now you gave it in slideshow mode.
Okay. Okay, sir. As we all know, trichoderma, a promising biocontrol agent which is free living, highly interactive in roots, soil, as well as in foliar environment. And the main mechanism behind the efficacy of trichoderma against different plant pathogenic fungi is mainly the mycoparasitism. It also colonize and penetrate root tissues, and which ultimately leads to induced systemic resistance. Also inhibit pectinases and some other associated enzymes for plants. But all these facts are already but our aim is to enhance the biocontrol potentiality of trichoderma. As because literature says that trichoderma can be stimulated for the production of chitinase by addition of chitosan in its bioformulation. Chitosan, it is nothing but a cationic polysaccharide which is having fungicidal and fungistatic as well as antibacterial and antiviral properties. With these points, we have taken three objectives. First one is the identification of novel nanomaterials for formulation of trichoderma. Then second one is the standardization of nanobioformulation protocol for doping microorganism and the chitosan nanoparticle. And third one is the bioefficacy of the formulated product in vitro and as well as in vivo condition. Our ultimate goal is to develop the nanochitosan-based formulation of trichoderma for enhancing plant nutrient uptake, growth, and mainly for disease management. For that, we have selected three sources of chitosan. First one is the commercial source of chitosan. Then second one is the one pathogenic fungi, that is Fusarium oxysporum, and three biocontrol agent Beauveria, Metarhizium menisopli, and Trichoderma viridi. Then we have isolated chitosan from these fungal cells by following the standard protocol of Agdam and his co-workers 2010. And the isolated chitosan was converted to chitosan nanoparticle by following the standard methods of Calvo and his co-workers 1997 with slight modifications. Then after synthesis nanoparticle, we need to go for characterization of all those nanoparticles. For characterization process, we have taken help of some of the equipments like UVB spectroscopy, FTIR, jetasizer, dynamic light scattering equipment, and transmission electron microscopy. For that, we have taken help from some of the art institutes like NAIST, Jorat, Assam, Nehu, SEF unit of Shillong, Meghalaya. Now coming to the results of the characterization study, we have found that in case of UVB spectroscopy study, all these nanoparticles are showing highest peak range of 310 to 350 nanometer, which is the permissible range in case of chitosan nanoparticle. And these results clearly show that yes, nanoparticles has already been synthesized or already been formed. But for further confirmation, we have also done the FTIR study, and from our FTIR study, we could able to identify all the major functional groups of chitosan, and which clearly shows that the synthesized nanoparticles were nothing but chitosan nanoparticle. Then coming to the zeta potential analysis of chitosan. As we all know, see surface properties of particles, we need to go for zeta potential analysis. And our zeta potential analysis record says that all the values of zeta potential are in the range of minus 30 millivolt, which states that the synthesized nanoparticles are stable in nature. Then coming to the transmission electron microscopy, the photographs of transmission electron microscopy shows that the synthesized nanoparticles are nearly spherical in shape. And the electron diffraction pattern of chitosan nanoparticles shows that synthesized nanoparticles are amorphous in nature. Then coming to the determination of the appropriate size of the nanoparticles, we have taken the help of the equipment dynamic light scattering equipment. And with the help of this equipment, we have determined the appropriate size of nanoparticles synthesized from Fusarium oxysporum. It will be 273.2 nanometer. And in case of Metarhizium menisopli, it was found to be 172.5 nanometer, and in case of Beauveria bassina, it was found to be 78.36 nanometer. And in case of Trichoderma viridi and commercial product of chitosan, it was found size of the nanoparticles were 89.03 nanometer and 300.1 nanometer respectively. Up to this point, chitosan nanoparticles were synthesized and characterized, but our ultimate aim is to go for preparation of the nanobioformulation of Trichoderma aspergillum by adding nanochitosan. That is why we need to go for compatibility study, and we have done the compatibility study of nanochitosan with Trichoderma aspergillum with 16 treatment combinations. And results showed that all the synthesized nanoparticles in all the tested concentrations were found to be highly comparable with Trichoderma aspergillum. These are the photographic representation of compatibility study of chitosan nanoparticle 1, 2, 3, 4, and 5. We have even taken the compound microscopic photographs of Trichoderma, conidia and conidiophores of Trichoderma just after the compatibility study to see whether is there any abnormalities. But we have not found any abnormalities in case of conidia and conidiophores of Trichoderma aspergillum. Then, based on the compatibility of Trichoderma aspergillum as well as the smallest size of the nanoparticles, we have selected 3 best nanoparticles for our in vitro efficacy test. And we have tested in vitro efficacy of Trichoderma aspergillum and chitosan nanoparticle alone and in combination against 3 targeted pathogens: Fusarium oxysporum, Sclerotium rollsii, and Rhizoctonia solani, which are the major soil-borne pathogens. And results showed that the combination of Trichoderma aspergillum and chitosan nanoparticle at a rate of 0.03% was found to be significantly effective in terms of radial growth inhibition of all 3 tested pathogens. These are the graphical representation and these are graphic representation. And similar results were also found in case of chitosan nanoparticle 2 and chitosan nanoparticle 3. Then, based on the in vitro efficacy, we have selected the best nanoparticle, which was found to be the chitosan nanoparticle 3 synthesized from Trichoderma viride for preparation of nanobioformulation. And we have liquid bioformulation of nanochitosan amended Trichoderma aspergillum by adding polymeric surfactant cum emulsifier cum dispergent (SDS) along with some of the oil and adjuvants. And we have also done the EDX study of the formulated product to see whether the chitosan nanoparticles are finally present in that formulation. And yes, chitosan nanoparticles are present in that particular formulation. And after that, we have done the in vitro bioefficacy of particular formulated product at 4 different concentrations at 0.05%, 0.1%, 0.2%, and 0.3%. Results showed that formulated product at the rate of 0.3% was found to be the, significantly effective in terms of radial growth inhibition of all the 3 targeted pathogens. And these are the pictorial representation of in vitro efficacy of formulated product against Fusarium oxysporum, Sclerotium rollsii, and Rhizoctonia solani. Then coming to the in vivo bioefficacy of the formulated product, we have selected 3 crops for these 3 targeted pathogens, pea, tomato, and cabbage. With these 4 treatment combinations, we have tested the in vivo efficacy. Observations were recorded mainly on % disease reduction, growth parameters, as well as in physiological parameters. And we have also tested the germination percentage, and results showed that highest germination of 90, 100, and 85.33% seed germination was recorded in case of seeds treated with the formulated product at the rate of 0.3% in case of pea, tomato, and cabbage. These are the overview of my pot experiment, and this table clearly shows that seeds with 3% was found to be significantly effective in case of protection over control in case of all 3 targets. And we have observed that seed treated with formulated product showing better results in terms of in case of physiological parameters also. We have the plant growth parameters like root, shoot length, fresh weight, dry weight of 3 crops, and the table showed that seeds with the product are showing better results treated with carpet or non-inoculated control. And similarly, in case of physiological parameters also, like content, rate of photosynthesis, leaf area also is treated with the formulated product are showing better result. And from these experiments, I can surely say that the combination of nanoparticle and biocontrol agent is found to be the best for management of soil-borne plant pathogens, especially like Fusarium oxysporum, Sclerotium ralsii, and Rhizoctonia solani. And it also opens up the possibilities of reduced use of synthetic pesticide, which we all know very much harmful to us and harmful to the environment. And this use of nanobiopesticide or nanobioformulation could play a major role in case of integrated pest and disease management system. And I am very much happy to share one information is that for this particular nanobioformulation, we named it as a trichogen N, and we have in this year itself we have for patent for this particular formulated product. And with this, I would like to conclude my presentation. Thank you.
Okay, thank you, Dr. Sharadi Barua, for your very informative presentation. So I have one request. Actually, you have to see in the coming days, you have to see what is the effect of this nanobioformulation on the physiological, biochemical, biochemical parameter of the host system. Then you can get a lot of information which will be helpful in your coming days. Okay, so now I request the participant to raise one or two question, very specific question to the presenter. One question is there from Natasha Kaishap, and she has mentioned that can this be used for degradation of preformed sclerotia of Sclerotium and Sclerotium ralsii and Rhizoctonia species, or say Rhizoctonia solani?
Yes, sir. Thank you for the question. Sir, I want to say that we have not tested on preformed sclerosia, but there is a possibility of using this nanobioformulation in case of preformed sclerosia, as because we have tested in Sclerotium ralsii as well as in case of Rhizoctonia solani. I am sure about that. It may work in case of preformed sclerosia also, especially in case of Sclerotium group of fungi.
Okay, thank you. Thank you, Sharodi Barua, for your presentation. Then can we proceed to next? Farhin Ahmed. Is she present?
Sir, he is a present. Yeah.
Farhin Ahmed.
Farhin Ahmed. Please unmute yourself. Farhin.
Yes, sir, I have unmuted it.
Are you going to share your screen?
Yes, sir. Yes, sir.
Okay, okay. Proceed.
Is it okay, sir? Hello?
Okay. Yes.
Yes.
Yes, sir. Can I proceed? Hello?
Yeah, process start.
Good morning. A very good morning to respected Chairman, sir, respected Dr. Pranab Datta, sir, Co-Chairman, respected Dr. Fatik Tamuli, sir, and one and all present here. Myself, Farhin Ahmed, I am presently pursuing my Master's in Botany from Guwahati University. So my presentation is on efficacy of zinc oxide nanoparticles against Sclerotinia sclerotium causing white mold disease in French bean. I have done the internship under in the guidance of respected Pranab Datta sir in Agricultural University, Assam Agricultural University. So French bean, that is Phaseolus vulgaris, is also known as common bean or kidney bean, is cultivated worldwide for its edible tender green beans and dry fruits. Its leaves are also been eaten occasionally, and the straw is also used as fodder. So we can say that this vegetable is in great demand throughout the world, with an annual global production of 12 million metric ton per year, with India being the highest producer with 4 million metric ton production per year. So, Phaseolus vulgaris is susceptible to a large number of diseases causing pathogen. For example, white mold disease caused by Sclerotinia sclerotium, bean rust disease caused by Euromyces appendiculatus, and many more. However, among all the diseases caused by Sclerotinia sclerotium, caused in Phaseolus vulgaris, the white mold disease caused by Sclerotinia sclerotium is very common and has primary significance, causing serious and unpredictable yield loss every year. So, talking about the management strategies, it may include eliminating of source of inocula, selection of resistant plants. It can include utilization of effective fungicides. However, it is very difficult to control fungal pathogen because a number of fungus have developed resistance to many conventional fungicides, such as, for example, dicarboxymides. Moreover, synthetic fungicides have been found to have adverse effect on human health and environment. And the traditional fungicides which are being used for the treatment are gradually becoming ineffective due to the development of new physiological races of the pathogen. So hence there is an urgent need for a safer and noble solution for this purpose, which doesn't harm the you can say doesn't harm the environment as well as human health. So nanotechnology is providing great promises in this field. Zinc oxide nanoparticle has emerged as a promising versatile inorganic material. It's biocompatible and has a great economic value. So the objective of my study is to study the effect of zinc oxide nanoparticle on mycelial growth inhibition of Sclerotinia sclerotium. Again, to study the effect of zinc oxide nanoparticle on sclerotial production and germination. So materials and methods used for this experiment. I have done this experiment in the nanotechnology laboratory, Agriculture University, Jorhat. So the glasswares and sterilization techniques all were done thoroughly for this experiment. So preparation of potato dextrose sugar. For this, I have taken 3.9 gram of PDA media, is PDA powder, and I have suspended in 1000 ml distilled water. And I have heated it so that the media dissolves completely. The pH was measured and was maintained at 7 by adding hydrochloric acid. For this purpose, sodium hydroxide can also be used. Now the medium was poured in cultured tubes and flasks and plugged with non-absorbent cotton and was autoclaved at 121 degrees Celsius for 20 minutes for sterilization. So culture of the Sclerotinia sclerotium. This was collected. Pure culture was collected from culture bank maintained at Nanotechnology Laboratory of Department of Plant Pathology, Agriculture University, Jorhat. For the maintenance of the culture, pure culture of Sclerotinia sclerotium was maintained on PDA medium by periodic transfer in fresh medium. A loop of inoculum from culture tube was transferred to PDA plates and slants and incubated in BOD incubator for 7 days at 20 to 21 degrees Celsius and was maintained as pure culture. For source of nanoparticle, the zinc oxide nanoparticle was collected from Nanotechnology Laboratory of Department of Plant Pathology, Jorhat. So for in vitro efficacy of zinc oxide nanoparticles, the technique which I have followed was, hello?
Sorry, you have only one, you go to the main point, that is result. You have only 1 minute left.
Okay, sir. So I have used food poison technique here. And the formula which I have used for growth inhibition percentage was of Skidmore and Dickenson. So this is the result. So chemical check was done using carbendism at 0.2%. For the result, the carbendism showed the highest activity in inhibiting the radial growth of the tested pathogen. But while comparison between the tested concentration of zinc oxide nanoparticle, the highest mycelial growth inhibition percentage was recorded at 225 ppm, that is 93.48%. And this was followed by 100 ppm, 50 ppm, 25 ppm, 10 ppm, and 5 ppm, and 1 ppm. Similarly, for number of sclerotia was highest in control plate, which was 27, followed by 5 ppm, which was 24, and 10 ppm, which was 20. And in rest of the concentration, there was no sclerotia found. This is the table here. You can see the picture here at 72 hours of inhibition. As you can see in the plate for 225 ppm, the mycelial growth is completely inhibited. And for sclerotial germination, in all the plates except the control, the sclerotial germination was nil. This is the picture of after 15 days of inoculation. You can see in control plate there were 27 sclerotia. In 5 ppm there were 24, and in 27 ppm, in 10 ppm there were 20.
It's okay. You please come up.
Hello. Yes, sir.
Okay.
Can I continue? Hello.
Last slide.
Yes, sir. So from this, this can be concluded that my result was positive, and this zinc oxide nanoparticle can be used as effective antifungal agent, and this can be used as alternative against those harmful fungicides. So thank you.
Thank you, Farhin, for your work and for your good presentation. So I request participants to raise one or two very specific questions to Farhin Ahmed. Probably there is no question, so we can proceed to the next presenter. That is
Thank you, sir.
RT Kumari. Okay. RT Kumari, are you present?
He is present, sir.
Okay.
Please unmute RT Kumari. RT Kumari. So might be, sir, she's technically present but physically not present.
Probably. So we can proceed for next presenter.
Yeah, next.
K.A. Barbute. K.A. Barbute, are you present? So K. Barbute is absent. Next presenter is N.A. Musmade. Musmade. N.A. Musmade.
If you are present, just send the message. Sir, this Kanifnath Borgute is present, sir.
Okay.
Kanifnath, please unmute yourself.
Yes, sir. Am I audible, sir?
Yeah, you are audible. Just a minute. Burgote sir, are you sharing the screen? We should
Yes,
share
sir.
from.
I will share, sir.
Okay. Continue, sir.
Yes, sir. Good morning, all of you. My name is Kanifatan Burgote. I am from Maharashtra, Solapur. Now I am going to present my topic, that is in vitro by efficacy of silver nanoparticles against collatericum gliosporides causing prototin pomegranate. Studies was carried out in at Vasantrao Naik, Marathwada Agriculture University, which have antimicrobial effects on different test pathogens, fungal, bacterial, viral. That's why this present investigation was done in vitro by efficacy of silver nanoparticles by using trichoderma species. We have characterized the take this species of trichoderma metum and aspergillum for the conduction of the studies, and these species are treated with silver nanoparticles by different harvesting at different days of intervals, and spectroscopy and spectroscopy and transmission electron microscopy. Then the in vitro efficacy of silver nanoparticles against collatericum gliosporides causing fruit rotting pomegranate were tested by poison fruit technique at different concentration at the rate of 150 ppm and 100 ppm, and we get max inhibition growth of test pathogens. In the studies, we have the culture filtered trichoderma harjanum and hematum for this study. Then we have synthesized the silver nanoparticles by 1050 aqueous solution, and this trichoderma, aspergillum, harjanum, and hematum, 3 species of trichoderma species are used. Then we have characterization with the help of spectroscopy, and we used the base as a base media. Then synthesis of by silver nanoparticles. 50 ml of aqueous solution of silver nitrate was treated with 50 ml of trichoderma culture, species culture, and synthesis of silver nanoparticles 250 ml. Then we have take this culture filtrate and harvested at different times of intervals, and we have got the characterization of UV's spectroscopy. We got the peak of different nanometer size, that is 380, 400, and 420 respectively. Okay. In transmission microscope, we have get the magnification of silver synthesized silver nanoparticles. So this is different filtrate species, that is trichoderma aspergillum, trichoderma harjanum, and hematum are 6 days, 12 days, and that is PDB broth.
Kapinath sir, please continue.
His voice is not audible.
Maybe, sir, he log out and again log in, and I request him to unmute.
Yes, sir. Now I am audible.
Okay, yeah, you are audible. Continue.
So 1 milligram of AgNO3 solutions treated with 6 days incubated culture filtered trichoderma species. A color change get dark brown color with characteristic surface plasma resonance. That is at trichoderma spirillum at 420 nm, trichoderma harzianum at 380 nm, and trichoderma hamatum at 24 hours incubation was recorded. Then we get form the stability of the aqueous solution treated with, as shown in the figure. So these peaks get from UV spectroscopy. That is trichoderma spirillum. The peak got at 380. Then trichoderma harzianum got at 420, and trichoderma hamatum at 380. So this is the result. This is the result of from transmission electron microscopy of silver nitrate culture filtrate and treated with silver nanoparticle solutions. So we got this size of silver nanoparticles of trichoderma harzianum species. That is maximum range of silver nanoparticles found, that is 22.93 to 36.8 mm. And the this is of trichoderma harzianum. So we got 8.45 to 15.3 nanometer synthesized by using trichoderma culture harzianum culture filtrate. So this this is the photographs of trichoderma hamatum. That is, we got 36 nanometer to particle size is 36.68. This image shows us a single particle of silver nanoparticles ranging from the 22.93 to 36.68. Then this on the basis of this after this result, we have carried out the test of, that is, in vitro evaluation of antimicrobial activity of silver nanoparticles by using trichoderma spirillum, trichoderma harzianum, and trichoderma hamatum against the test pathogen Colatrichum gliosporidis causing fruit rotting pomegranate. So this we have prepared this different concentration of the three species: trichoderma harzianum, hamatum, and aspergillum at 150 ppm, and then trichoderma harzianum. We have also treated the only culture filtrate with against test pathogen, that is trichoderma harzianum culture filtrate, trichoderma hamatum culture filtrate, and trichoderma aspergillum culture filtrate against Colatrichum gliosporidis in poison food technique. So that is trichoderma harzianum. So this is our treatments. That is, we get that is trichoderma spirillum silver nanoparticles at the rate 150 ppm. We got 48.7% inhibition, and in treatment 5, that is trichoderma harzianum silver nanoparticles, we got at 150 ppm 48.25% inhibition. So these are the results of different treatments. That is near about 50% inhibition. We got in this result. Then, so this is the in vitro evaluation of silver nanoparticles against Colatrichum gliosporidis causing fruit rotting pomegranate. So the treatments in these plates, we get we seen the, this is pure culture of Colatrichum gliosporidis causing fruit rotting pomegranate, and other is poison food technique. Treatments is done by poison food technique, and in this we have added the at 150 and 100 ppm concentration of silver nanoparticles, which are different trichoderma species, that is aspergillum, harzianum, and hamatum, and we got this in the trichoderma harzianum at 150 ppm. That is treatment number 5. So this treatment number 5 get maximum inhibition of test pathogens, Then after that, the treatment number 6, that is trichromatum at the rate of 150. That is treatment number 6. We found the number 3, the trichromatum at 100 ppm. We get near about 40.8 micropercent inhibition. This is in vitro. Then this is graphical. Graphical photographs of in vitro evaluation test against Polatricum phosphorus. So this is the then the so in this study we have treated the silver particles with culture filtrates of Trichoderma species against Polatricum gliospores causing granate, and they 6 days old culture we have get max darkness of the culture filtrates. So on the basis of darkness culture filtrate, we have tested and used for the next studies, that is Trichoderma aspirant, Trichoderma herzianum, and characteristics of surface plasma resonance band was observed at 380 nanometer for Trichoderma aspirant, 420 for Trichoderma hamatum, and 380 nanometer for Trichoderma hamatum after 24 hours of incubations. The maximum synthesized silver nanoparticles were observed in 6 days incubated culture filtrate. That's why we have used the 6 days culture filtrate for the future studies. And AGNO3 solution is followed by. So we have also used the culture filtrate, culture filtrate from different pieces of Trichoderma for the studies by positive techniques. And the report of Trichoderma transmission microscopy, that is maximum synthesis of silver nanoparticle on the size of particle, that is Trichoderma spiral at size of Trichoderma spiral silver nanoparticle 0.7 to 0.9 and to 12.80 nanometer, and then Trichoderma herzianum silver nanoparticle size and hamatum silver nanoparticles at this particle single particle size is 22.90 nm. So we got Trichoderma herzianum and Trichoderma hamatum at the rate of 150 ppm and 100 ppm respectively, got minimum 50% of the test pathogen. That's why the best with silver nanoparticles are Trichoderma herzianum and Trichoderma hamatum, which have found maximum effects. Found maximum effectiveness. Trichoderma aspirillum at the rate of 100 ppm. It has most also inhibition near about 7.
Datta sir.
Yes. Yes, Rajesh.
Yes.
Rajesh?
Hello.
Okay. That Dr. Bhargava is not audible now.
He completed his session, sir.
Okay. Actually, thanks to Bargute for your good presentation, but I have one very simple query. Whether he tested any chemical as a control to compare the efficacy of nanoparticles of different sources, say Trichoderma herzianum, hamatum, as well as Aspergillum. Dr. Bargute?
Bargute sir, Kapinath sir, if you are audible, give the answer. I think yeah, there is issue. Next is Arti Kumari. Actually, she joined from and she will go for the presentation from Geeta Shri Das.
Okay.
Sir, am I audible?
Yeah.
Yes.
Yes, sir. So is the presentation visible?
Not yet. Just a minute.
now?
Yeah, it
Yeah.
is sharing.
Yeah. Shall I proceed, sir?
Please note, Arti Kumari, the time, duration.
Okay. Yes, sir.
Okay, proceed.
A very good morning to everyone present here, respected chairman, co-chairman, reporter, and all the participants. My name is R. Kumari, and I'm going to present my research paper on the topic on toxicity of zinc oxide nanoparticles on microbial biological control agents and mammalian cells. As you know that nanotechnology is one of the most advanced science of the 21st century. The unique physicochemical properties of the nanoparticles set it apart from its bulk counterparts. Among all the nanoparticles studied so far, zinc oxide nanoparticles are one of the most celebrated ones. They are extensively used in various applications such as in biomedical and it is also used in textile industries, in paints, cosmetics, etc. They possess antimicrobial, antibacterial, antiviral properties as well as UV blocking properties. Zinc oxide nanoparticles have been found effective against various phytopathogens such as Xanthomonas oryzae and against virus like tobacco mosaic virus. Then it has also been known to boost the yield and growth of crops. However, the inevitable exposure of these nanoparticles to human and animal health, it raises concerns regarding its potential hazard. So it is very essential to understand the interaction between zinc oxide nanoparticles and beneficial microorganisms and mammalian cells. So with this background, the following objectives were formulated. First, to study on toxicity of zinc oxide nanoparticles on microbial biological control agents. And second, to study the cytotoxic effect of ZNOs on mammalian cells. So for this study, we were in need of zinc oxide nanoparticles, which were synthesized by chemical reduction method, as shown here. Thereafter, these were characterized using various techniques. UV spectroscopic analysis was done, and the absorbance peak was obtained at 376 nanometer, which means that the nanoparticles, zinc oxide nanoparticles, have been synthesized. Thereafter, zeta potential value of minus 23 millivolts was found, which means that the synthesized nanoparticles were stable in nature. Then the average size of the nanoparticles were determined by dynamic light scattering technique, and it was found to be around 48 nanometer. Thereafter, to visualize various nanoparticles and to see the morphology, shape, size, electron microscopic studies were done as shown here. Thereafter, to ascertain various functional groups associated with ZNOs, FTIR analysis was done, and here is a list of various functional groups that were found associated with it. This is the atomic force microscopic image of ZNO. Now, thereafter, the synthesized nanoparticles were further used to determine the toxicity of ZNOs on microbial biological control agents. For this, microorganisms used were Trichoderma hertzianum, Beauveria bassiana, Bacillus thuringiensis, and were tested against 3 different concentrations, 50, 100, and 200 ppm. Now, to determine the effect of ZNOs on biomass production of fungal biological control agents, mycelial weight inhibition technique was used, and the result obtained as follows. In both the cases, for Trichoderma hertzianum and Beauveria bassiana, dry weight was found to reduce with the increasing concentration of ZNOs, as evident here. The inhibition percent was found to be highest at 200 ppm. These are some of the pictorial evidences. Thereafter, we determined the effect of ZNOs on biochemical parameters. For this, total soluble sugar and total soluble protein were estimated following standard protocol. And we found similar results here also, wherein the total soluble sugar and total soluble protein, it declined with the increasing concentration of ZNO. Here are some of the pictorial evidences. So now, third, we determine the effect of ZNOs on bacterial biological control agent, that is on Bacillus thuringiensis, following this diffusion method. So we found that there was no inhibition zone at all the tested concentration, which means that ZNOs were Bacillus thuringiensis were resistant to ZNOs at all the tested concentration. Now moving on to the next part of my study, that is to determine the effect of ZNOs on mammalian cells. So for this study, mammalian cell line used was Vero cells. Now, Vero cells are derived from the cells, kidney cells of an African green monkey. Before going deep into that, we need to ascertain the uptake of ZNOs by Vero cell following standard protocol, and the result we obtained as shown here. Even after 1 hour of exposure, the ZNO uptake was very high. As we can see here, it was highest for 200 ppm. That means it increased with the increasing concentration. And again, with the increase in the time of exposure, that is after 4 hours and 8 hours, we found significant increase in the ZNO uptake, which is very evident from this table. Now moving on to determine the cytotoxic effect of ZNOs on Vero cells. First of all, we need to determine the effect of ZNOs on cellular morphology of Vero cells by scanning electron microscopy. So as evident from these pictures, at 50 ppm concentration, there was no cellular disintegration. However, at 100 and 200 ppm, cellular integrity were somehow disintegrated. Then, moving on to the biochemical test to determine the percent viability of Vero cells after exposure to ZNOs by MTTSA. So this MTTSA, it measures the amount of formazon crystals produced by Vero cells. And we found that the highest viability of Vero cell was there at 50 ppm concentration, that is about 77.01%, and it gradually declined with the increase in concentration, and it was minimum at 200 ppm, that is 69.57%. So again, when we analyzed our MTT results with the standard scale, we found that it falls in the range of 80 to 60%, which means it shows weak cytotoxicity. Now, moving on to determine the percent cytotoxicity of ZNOs by lactate dehydrogenase test. Now, this lactate dehydrogenase test, it can measure even a slight aberration on the cellular membrane, as lactate dehydrogenase enzymes can leak from these cellular disintegrations. So percent cytotoxicity was found to enhance with the increasing concentration. As we can see here at 50 ppm, it was 12.31%, which increased to 23.28% at 200 ppm. It is very evident from this graph also the viability percent is declining and the cytotoxicity percent is increasing. Thus, from our results on Vero cells, we can say that zinc oxide nanoparticles poses mild toxic effect against Vero cells. Now, to summarize the entire study, the zinc oxide nanoparticles at 3 different concentrations, 50, 100, and 200 ppm. It exhibits weak toxicity on fungal biological control agents. However, on bacterial biocontrol agent, that is Bacillus thuringiensis, it was found to be completely resistant. Then, when study was done on Vero cells, the internalization percent was found to be very high, and it increased in a concentration and time-dependent manner. The viability of cells measured by MTTSA was found to decline gradually with the increasing concentration, and the LDH assay results were found to be in line with the MTTSA result, wherein the maximum cytotoxicity value was found to be 23.28% at 200 ppm concentration. Our SAM analysis results, it's revealed that at 50 ppm concentration, there was no disintegration of cells. However, 100 and 200 ppm, the cellular disintegrations were evident. Now, to conclude, zinc oxide nanoparticles, it poses mild toxic effect on fungal microorganisms such as Trichoderma herzianum and Beauveria bassiana, and against mammalian cell, that is Vero cell, and it was found to be completely resistant for Bacillus thuringiensis. Thus, we can say, however, the results need to be further assessed by various genotoxicities and in vivo assays. Thank you.
Thank you, Arati, for your presentation. You are on time because you have completed your presentation within the stipulated time. And I have one query. You see, literature says zinc oxide nanoparticle are biocompatible. So, based on your word, what is your comment? Okay, based on your word.
sir, based on my work, we can say that zinc oxide nanoparticles, it has a low toxic effect. Means these are, we cannot say it is completely compatible. However, we need to assess the result further by various assays.
Okay, so thank you. Is there any specific question from the participant?
Sir, there is one question by Raghunandan BL. How do you justify the resistance of BT against nanoparticle? Probably reasons.
Yes.
Yeah, so maybe the reason might be they have got certain detoxifying enzymes within the bacterial cell. As we all know that all microorganisms are different and they have their own cellular defense mechanism. So maybe they have got certain zinc oxide resistant genes or some enzymes which can which prevents the release of zinc oxide ions from the nanoparticles. Until and unless zinc oxide ions are released from the nanoparticles, then only it will cause harmfulness. Otherwise, only in the nanoparticle state it is pretty much compatible.
Yes, thank you, RT.
Raghunandan sir.
Yes, sir. Yes,
Is
sir.
it okay?
Actually, I would like to know what exactly the reason, because she said 100% resistance against nanoparticles. So what would be the reason? Because at the same time, fungal biocontrol agents are having a inhibitory effect, is it not? So in case of BT, it is 100% resistance. What would be the reason? The probable reasons? Okay, it might have some detoxifying mechanisms. Okay, we'll agree with that.
Okay.
And okay, sir.
Okay. The next, yeah, you continue.
You continue.
Sir, actually we found that there was no inhibition zone at all. So that means it is resistant. But sir, sir, sorry,
Continue.
sir, I forgot.
It's okay, no problem. And Dr. Raghunandan, you can directly contact with Dr. RT Kumari. Okay. Hello.
Next speaker is Narayan Musmode.
Musmode, right.
Sir, share your screen or I should share. Narayan sir.
You share, sir.
Okay. Just a minute.
I'm audible, sir.
Yeah, you are audible. And sir, please take care about the time duration.
Okay, sir. Okay, sir. Okay, sir. Good morning, everybody. Respected chairman.
Yes,
and
sir, you
co-chairman.
continue.
Yes, okay, sir. Okay, sir. Good morning, everybody. Myself, Mr. Narayan Musmade, Department of Plant Pathology and College of Agriculture, Navsari. Respected chairman, respected co-chairman. Today, I am here to oral presentation on synthesis of chitosan-mediated silver nanoparticles for potential antifungal application. Next, please. Introduction. Nanotechnology is an innovative and emerging discipline in the field of the science and technology. In concern to the nanotechnology, all these speakers will give the information about the nanotechnology. That's why I am going for the details of my study. First, the chitosan has been widely used for the cosmetic and the material-based applications. However, in recent times, in the plant systems, chitosan has been reported induced in-facilitated disease resistance as well as antifungal, antibacterial activity. That's why chitosan has been selected for the synthesis of the silver nanoparticles. Then the source of the chitosan was selected. The mud capsules contain 30% chitin 16% proteins and 55% calcium carbonates. That's why we are selected the mud crab cells. Then the various metal nanoparticles, the silver nanoparticles are the most extensively intrinsic metal nanoparticles which having the electrical conductivity, optical properties, oxidative catalyst, and antimicrobial activity. That's why we are selected silver nanoparticles for the antifungal activities. Next is the objectives of my studies is the synthesis of the chitosan from the mud crab cells, and second objective is the in vitro efficacy of chitosan mediated silver nanoparticles against the Sclerotium rholpsi. The Sclerotium rholpsi are the soil-borne fungus. That's why we are selected, and it causes the more than 500 species. So first one is the extraction of the chitin and chitosan from crab cells. We are selected or collected the mud crab cells from the nursery local markets. Then first it washed and dry. After the drying, the grinding and sieving of the mud crab cells. After the drying, we use the treatments of deproteinization. The deproteinization treatment is 4% NOH for 90 degrees Celsius at 2 hours. And the ratio of the NOH and shell is the solid solvent ratio is 1:20 as the weight by volumes. Next please. The next step is the washing. We use the washing of the nanoparticle chitosans up to the pH solution is come to the 7. Then use the next treatment is demineralization. Demineralizations we are use 2.5% HCl for 6-hour treatment at 30 degrees Celsius, and the solvent solution ratio is 1:20. The next step is washing and drying. Again, we are washed with the distilled waters, and the pH of that solutions come to the 60 degrees, come to the 7th, and it's by 60 degrees Celsius for 24 hours in the oven. Then decolorizations and dewatering. After the decolorization, we have acetone treatments for 10 minutes, and then next step is again the washing and next please. Final product is the chitin was produced. The chitin was produced, and we do the acetylations of these chitins of 50% NOH weight by volumes for 2 hours at 150 degrees Celsius solids, and solid solvent ratio is the 1:15. And again, we are washing the materials and washing and drying after 60 degrees Celsius for 24 hours, and final product is the chitosan was produced. And this chitosans we are selected and continue for the synthesis of the chitosan mediated silver nanoparticles. Next is the methodology. The methodology first is the biosynthesis of silver nanoparticles using the chitosans. The chitosan solution of various concentration 0.2 and 0.5% were made in the acetic acid in separate inorganic flask. Then it was stirred in the magnetic stirrer until the homogeneous solution was achieved. After that, we have prepared the 4 different concentrations of the silver nitrates is the 0.5%, 0.5 millimolar, 1 millimolar, 1.5 millimolar, 2 millimolar at different pH 7, 9, 11, and 13. And we are added to the separate chitosan solution and against the magnetic stirrer. After the magnetic stirrers, we will mix the solutions in the silver nitrate and chitosan concentration is 1:1 proportion in the boiling tube, and the mixtures was kept in autoclave at 15 psi pressure at 120 degrees Celsius for 40 minutes. Then after the completion of autoclave cycles, we see the color of the solution was changed from the colorless to the light yellow and finally to yellowish brown in color indicates the formations of silver nanoparticles. Next please. The separation of nanoparticles. Separation of synthesized nanoparticles, we use the centrifuge machines, and for the centrifugation 15,000 RPMs for 20 minutes, and the supernatant was discarded, and the plate was collected and washed against with the water and centrifuged 3 times to ensure the better separations of the free entities from the silver nanoparticles. Finally, purified suspensions of the silver nanoparticles was dried at hot air oven at 250 degrees Celsius for 2 hours, and final product was the dark color nanoparticles powder was obtained and used for the further studies. Next is the characterizations of nanoparticles using the UV visible spectra analysis. The silver nanoparticles was monitored periodically in UV visible spectrophotometer. The spectral absorption was done in resolution 1 nanometer between the 300 to 800 nanometers, and the equivalent amounts of the reaction mixture is 1 ml was diluted in milky water in 9 ml. Subsequently analyzed at the room temperature at different incubation period 30 minutes, 1 hour, 2 hours, 4 hours, 6 hours. Next please. The in vitro assay of antifungal activities of nanoparticles. The antifungal activities of nanoparticles were evaluated against the Sclerotium rholpsi by using the poison food technique in vitro conditions using the potato dextrose agar medium and selected the different concentrations 10 ppm, 50 ppm, 100 ppm, and 150 ppm. Next please. The results. Visual observations. You see in the slide the before and after the heating of the nanoparticles, the color was changed. and the color of the solution was changed from the colorless to the light yellow, and finally it's converted into the yellowish brown. We will be indicate the synthesis of the silver nanoparticles. After that, we are characterized by using the UV visible spectra analysis. All the spectra exhibited in the absorption band in the range of 400 nanometer to 435 nanometers. In this experiment, we are taken the 4 different concentrations of the AgNO3, 2 concentrations of the titosans, and the 4 different pH. Total treatment is the 32 treatments. Out of that, we are the selected one treatment which gives highest lambda max as well as the absorption. So therefore, we will see here the absorption patterns of the solutions was kinetically monitored in the range of the 300 and 800 nanometers, and the characteristics absorption peak was observed at 421 nanometers, and absorption is 0.38 in UV spectra of silver nanoparticles synthesized from the cracked shell waste. Next, please. In these tables, you see the effects of the different pH and AgNO3 concentration on the silver nanoparticles synthesized through UV visible spectra at the 0.2% concentration. You see here the 4 different concentrations of the AgNO3. The concentration of AgNO3 is increases. That ultimately the absorption of UV spectra is also increases. And the pH 7, 9, 11 we are selected. You see the last one is the pH 13. It gives the highest absorption is 0.38, and lambda max it is a 421.5. And you see this simultaneously in the figure was given in the right hand side. The higher peak given in the green color is the highest absorbance at the 2 millimolar concentration, and lambda max is 421.5. So, and the peak area, it is the area under the absorption wavelength, it is a 16.45. Next, please. The effects of pH on the synthesis of the titosan-mediated silver nanoparticles at 2 millimolar AgNO3. You see here the pH increases. The ultimately the absorption is also increases. So therefore, in this graph, we are concluded that all the experiments using the 4 different pH. So that's why I was concluded that the alkaline condition is increases for the reductions of the metal ions, as well as the UV regions with increases with pH, which reflect the size of the silver nanoparticles. The size of the particle decreases with the increasing pH, or we are the concluded here. Next, please. In table second, the effects of the different titosan and AgNO3 concentrations at the 13 pH at AgNO3 and silver nanoparticle synthesization through UV spectra. So we see here in these tables the 0.2% concentration and 0.5 concentrations of the titosans. We are given here for the characterization and the synthesis of the silver nanoparticles. You see here the 0.2% titosan concentration, which gives the highest absorbance as well as the highest maximum lambda max. So we are concluded that the 0.2% titosan was the best for the synthesis of the silver nanoparticles. Next, please. The last one is the table 3, in vitro efficacy of titosan-mediated silver nanoparticles against the plant pathogens Sclerotium rholsi. So you see here the 4 concentrations of the titosan-mediated silver nanoparticles: 10 ppm, 15 ppm, 100 ppm, and 150 ppm. Simultaneously, we take the AgNO3. It is also 4 concentrations, and the growth of inhibitions was calculated after the 7 days. So therefore, we are concluded that the 150 ppm, which was gives the highest inhibitions over the control, is the 88.2% inhibitions over the control. Next, please. You see the figures here. The in Sclerotium rholsi, in titosan-mediated silver nanoparticles in left hand side, and right hand side it is the only AgNO3 concentration. The 150 ppm is the best. Next, please. The conclusions. The green synthesis of silver nanoparticle is a good facility which is easily produced and extensively used to control of the phytopathogen. Titosan from the bio-waste can be used as a reducing agent and the capping agent in the synthesis process, and titosan is readily available in these local markets. Next is the good characterized titosan nanoparticles. We are produced at 2 millimolar AgNO3 and 0.2% titosan concentration at 13 pH. And AgNO3 silver nanoparticle produced from the titosans as 150 ppm, which inhibits the growth of the Sclerotium rholsi by affecting the cellular functions, which cause the deformations in the fungal hyper. Thank you.
Thank you, Dr. Narayan, for your nice presentation. It is good that you have standardized a protocol for synthesis of silver nanoparticle.
Okay, thank you, sir.
So the mediation of the chitosan. But I have one doubt that what is the role of chitosan in synthesis of silver nanoparticle? Can you please tell me
Yes,
how
sir.
actually?
Yes, sir.
Yes, please.
Yes, sir. Sir, chitosaurs, we are seeing the structures of the chitosaurs. There is the NH2 groups. NH2 groups is replaced by the AG. Because sir, chitosan, it is used as the reducing as well as chelating as well as the capping agents, and it is the excellent source for the synthesis of silver nanoparticles. At the process of the reductions, sir, the NH2 groups, it is the replaced by the Ag. NH2 with remove the H ions, and then the Ag plus ions was attached to the chitosan, and they form the chitosan mediated silver nanoparticles.
Okay, and you are telling that your process is a biosynthesis process, right?
Yes, sir. Yes, sir. Biosynthesis means, sir, we are collected the chitosan from the capsules. That's why we are seeing the.
Whether you have studied the shelf life of your synthesized nanoparticle, what is the shelf life? Up to how many days their size remains same?
Yes, sir. Yes, sir. It is the 6 months, sir.
6 months. Okay, good.
Yes, sir.
And please do your statistical analysis for table 3. Then you can
Yes, sir.
draw
Yes, sir.
the conclusion, right?
Yes, sir.
And
Yes, sir. Yes, sir.
congratulations from my side.
Yes,
Very
sir.
nice work you did. And any specific question from the participant they may raise? One question from Chauhan. Dr. Chauhan, is there any use of nanoparticle in pesticide residue analysis? This is the question.
Yes, sir. Yes, sir. Yes, sir.
Please respond to this question. Is there any use of nanoparticle in pesticide residue analysis?
Yes, sir.
Okay. Okay, if there is no, and one more question from Raghunandan.
Raghunandan sir, please.
Sir, yes, sir. Sir, I would like to know. So, he's presented the in vitro test, silver nitrate, that is AgNO3. So, no effect on pathogen. So, what will be the reason? Generally, it is bactericidal, fungicidal effect is there. Silver nitrate, AgNO3.
It has very good property, right?
Throughout the presentation, compared to control, no effect of AgNO3 on pathogen.
Sir, due to the low concentration, sir.
No, 150 ppm till 200 ppm are tested, no? There might be some, there might be at least, there may be some inhibition of this AgNO3 against pathogen must be there. Please check it out.
Yes, sir. Yes, sir. We are, sir, taking the observations after the 7 days, sir. That.
Okay. Okay.
But still, there should be some effect on the pathogen because nitrate is highly effective as an antimicrobial agent. Okay, so still, we can discuss it later on. So we can proceed to next presenter, Dr. Pranjal Kaman. Pranjal Kaman,
share your screen. Please take care about the time. The next session chairman and co-chairman is already present, Pranjal and the sub.
Yes, start. Pranjal, your time starts now.
Pranjal, please proceed. Yeah.
Probably some network issues. Dr. Pranjal? Rajesh, one request.
Yeah, got it, sir.
Okay.
Pranjal, please proceed.
And out of these, biotic factors are considered to be one of the most devastating one. And out of these biotic factors, rice blast caused by Bendafortia gracia is found to be the most devastating one, as it may yield loss up to 100%. So for this purpose, various management practices are used like biocontrol agents, botanicals, and many other things. But it has found not to be effective against the pathogen. So here we are trying to synthesize gold nanoparticles from metarism and isophily for its effective management with the following objectives. So first objective is the synthesis and characterization of novel nanoparticles like gold from metarism and isophily, and the second objective is on evaluation on efficacy of novel nanoparticles against magna-protect ratio and its effect on host biochemical parameters. So for this purpose, biomass production of metarism and isophily was done in potato dextrose broth by addition of CMC in laminar airflow. And after 7 days, the growth of metarism is come out in the potato dextrose broth as shown in the figure. After that, what we do? We synthesize the gold nanoparticles protocol given by the Emerson et al. 2017. Here, what we do, after harvesting of the metarism and isophily, it was filtered through Whatman filter paper. After that, we have add 1 millimolar chlororic acid. And after that, what we do, then the pH was adjusted to alkaline condition. And after adjusting the pH, we kept in a rotary shaker for 7 to 10 days. By wrapping with a brown paper to make it a dark condition. After that, we observed the change in the color of the extract. Here we observed that the color changes from black to purple in color. This ascertains the formation of the gold nanoparticles. After that, for further confirmation, we have gone for characterization of the synthesized gold nanoparticles by different equipments like UV spectrophotometer, dynamic light scattering, zeta sizer, and transmission electron microscopy. UV spectroscopy results shows that a characteristic surface plasmon resonance band at 540 nanometer. And the size of the biosynthesis gold nanoparticle was found to be 32.5 nanometer with polydispersity index of 0.50. This indicates that the biosynthesis gold nanoparticle is highly dispersive in nature. And we have also done data potential, and we found that the source of the gold nanoparticle is found to be negative with minus 20.7 millivolt. And this indicates that the biosynthesized gold nanoparticle is highly polydispersed, highly stable in nature. FTR analysis shows that various chemicals group, out of which one is the amide group, which can be seen from the graph that 3495 centimeter is. This ascertains the formation of the gold nanoparticles. After that, transmission electron microscopy study revealed that the shape of the gold nanoparticle was found to be triangular to quasihedral in shape. After that, regarding the second objective is evaluation on efficacy of novel nanoparticles against magna-protect ratio and its effect on host biochemical parameters. For this purpose, magna-protect ratio was isolated from typical symptoms showing in the blast of the rice blast. And this is the pure culture of the rice blast in oatmeal agar, and this is the slant, and this is the microscopic observation. After that, after isolation, the biosynthesized gold nanoparticle was tested its efficacy against the rice blast, that is magna-protect ratio, at 3 different concentrations, that is 150, 100, and 50 ppm. And observation we have taken was the mycelial growth inhibition. And for comparison, we have taken tricyclosol at the rate 600 ppm. Results shows that the biosynthesis gold nanoparticles at the rate 100 ppm, at the rate of 150 ppm is found to be effective in reducing the mycelial growth of magna-protect ratio at 90.66%. This is the table. And for pod experiment, we have done the following treatment combinations, and which I will not display. And the observation recorded was percent disease incidence, and for primary and secondary metabolites were also recorded. Primary metabolites like total chlorophyll, total soluble sugars, total soluble proteins, lipid peroxidation, and secondary metabolites like alkaloid, flavonoid, and phenol. And this is the general view of the pod experiment. And we found that the highest total soluble sugars, total soluble protein, total chlorophyll, and lipid peroxidation, phenol, flavonoids, and alkaloids in treatment 10. That is when biosynthesis gold nanoparticles applied as seedling dip treatment plus soil application plus foliar application as compared to the control. This is the photographs of the showing the inoculated control, and this is the best treatment. And these are some of the photographs showing the biochemical parameters. That is the total soluble sugars, soluble protein, chlorophyll, and peroxidation. And this is the phenol and flavonoids. And conclusion, from this experiment, we can conclude that gold nanoparticles at 100 ppm concentration is found to be effective in suppressing the rice blast pathogen caused by magnaprotect ratio with enhanced plant growth parameters. And this will help in development of a suitable new fungicides for management of a rice blast disease. Thank you all.
Thank you, Pranjal, for your presentation. And you have, it is good that you have completed before time. And only one specific question from the participant, please, because we are behind the time. Okay, if not, you are going, we can move to the last oral presenter, R. Misra, Ritima Misra. Ritima, are you here?
She is here, sir.
Yes, sir.
Please go ahead.
Is it visible, sir?
Not yet.
Still not.
Is it visible, sir?
Not yet.
Sir, what I can do?
You just share your screen.
Sir, I have shared.
First, you open your file, open your folder, open your file, that is presentation, and then share your screen. Now it will be visible. Okay.
Yeah, it is sharing now. It will take some time.
Is it, sir?
Yeah.
Shall I continue, sir?
You are presenting. Yes, now it is okay. You can proceed.
Yes, sir. So a very good good afternoon to the respected chairman, sir, co-chairman, sir, and all the participant present here. And the corresponding author of my presentation is Dr. Pranab Datta, sir, and this presentation is presenting by me, Ritima Mishra. Today I'm here to give a presentation on the topic of efficacy of biosynthesized silver nanoparticle against Sclerotium ralsei causing collar rot of Pipali, that is Piper longum. I'm pursuing MSc in botany in Guwahati University. I have done an internship under the guidance of Dr. Pranab Datta, sir. So let's see what I did during my 1 internship. So here it's introduction. Piper longum is a climber belonging to the family Piperaceae and is considered important for the medicinal and culinary uses. Various diseases are found associated with Pipali like Phytophthora wilt, collar rot, alternaria leaf spot, deficiency diseases, etc. Out of which, collar rot caused by Sclerotium ralsei seems to be the most devastated one, causing a heavy loss to the crops. So first of all, we can follow the strategy like biocontrol, botanicals, etc., but have very low or we can say no effect. So the use of the chemical is most effective and promising one. But since the pipali is a medicinal plant, hence we use of chemical is so hence the use of chemical is discarded. So alternatively, to manage this disease, a new technology called nanotechnology can be used efficiently. So here our motive is to treat the Piper longum with a nanoparticle extracted from black tulsi, that is Ocimum sanctum. So reason for choosing the pipali plant. I have chosen this pipali plant because it is locally available and has a medicinal value. Here, its medicinal value, anti-allergic activity. In combination with the 7 medicinal plants used for the allergic rhinitis as antihistamic allergy. Anti-tumor, it can work against tumor also. Intestinal disorder, the fruit of pipali can inhibit the growth of entamoeba histolytica, exhibiting anti-amoebic activity, which is reported also in mice. Then respiratory disorder. The concoction of fruit and the root used against cough, cold, chronic bronchitis, etc. Anti-inflammable activity. It can the decoction of pipali fruit show the anti-inflammable activity. Other uses, traditional uses are it can work against worms, bronchial disease, used to boost up the memory, etc. So brief intro with a nanoparticle. It is usually refers to the inorganic material, sizes between 1 to 100 nanometer, same as ultra fine particles. Nanoparticles can reveal the size-related properties notably different from those of either fine particles or bulk material. There are different types of metals, out of which a silver nanoparticle have been considered most prominent one due to its effect and rapidly growing application in different environmental areas. Silver nanoparticle have been used against different fungal pathogens. So here we use this silver nanoparticle extracted from black tulsi. It has been reported against diseases caused by Alternaria alternata, Rhizoctonia solani, Fusarium oxysporum, etc. So method of the experiment. In the lab, we did our experiment in a very systematic order. First step is a cleansing and sterilizing glasswares and PDA plates. For this, first we have to wash the glasswares with a hot water, then keep them for drying, plug the glasswares by a cotton piece gently, then sterilizing. For the sterilization, keep all those in a hotter oven for at least 160 degrees Celsius for 2 hours. Then second step of PDA media in the conicals. So why we need to prepare the PDA media? It is semi-solid or solid designed for growth of microorganisms. For the preparation of PDA media, we have to take a commercially prepared potato dextrose agar powder and mix with water to prepare.
Go to the
We can PDA and a conical. Yes, sir.
Please go to the
Then
result.
take out those flags from observation here. Here for sufficient, then do the periods of incubation. One is
So after that, poster presentation will be start. Who are the poster? Parishmita Bora, Deepavinta Biswas, Munni Das, TD Parmar, Sumit Jani, and the Punawati. So be ready, and please take care of one thing. You have to complete your poster within a time. Only within 3 minutes.
Okay, Dipanita Bishwash, are you present? Hello.
Hello, sir, it's Parishmita Bora. Am I audible?
Yes, yes, you can continue, please.
So I'm sharing the screen. Sir, is it visible?
Not yet.
Sir, is it visible now?
No.
Parishmita.
Yes, sir.
Wait, I will share from my side.
Yes, sir. Please share.
So it is no wastage of the time. Deepavita Biswas, you are present.
Yes, she is present now.
Oh.
Let us finish from Parishmita, okay? Yes, Parishmita, please proceed.
Okay, sir. Good afternoon to respected chairman, co-chairman, honorable dignitaries, and talented participants. My name is Parishmita Bora, and today I'm going to present a poster on biogenic synthesis of silver nanoparticles, its characterization, and its effect on the protein of Fusarium oxysporum with two objectives in mind. We all know that nanoparticles are particles of matter whose smallest function in at least one dimension is on the nanometer scale, and silver nanoparticles are one of the most celebrated ones. We also focused on proteins, since if we can destroy the proteins, there is a high chance of harming or killing the organism at an early stage. Now, in this research work, for the first objective, we synthesized nanoparticles from an indigenous strain of potential biocontrol agent, Trichoderma aspergillum, and silver nitrate by following a standardized protocol. In figure 1, you can see that we have mass cultured the Trichoderma aspergillum in PDB, which was later used for synthesis. In figure 2, we have shown the synthesized silver nanoparticles, which are reddish in color, and alongside it, supernatant of Trichoderma aspergillum, which was not treated with AgNO3, which is greenish in color. You can observe there is a change in extract color before and after synthesis. The next step was characterization. For that, we used UV spectrophotometers, zeta sizer, and DLS. In the resulting chromatogram shown in figure 3, we observed a peak at 400 nanometers, around 400 nanometers, indicating partial homogeneity. Also, in the following zeta sizer analysis report, on figure 4, we found that the zeta potential was minus 30.9 MV, which means that the nanoparticles were quite stable. Also, in the DLS, which is also given in figure 4, we found that the polar disparity index was 0.427 for an average size of 46.94 nanometers. Now, coming to efficacy, for that we cultured Fusarium oxysporum in PDB in two treatments. The first treatment, which I'm going to refer to as control from now on, had Fusarium oxysporum in PDB, and the second treatment, that is T2, had 100% concentration of AGNP and F-oxysporum in PDB. These treatments were observed for 10 days in various replications, after which no further growth was observed, or actually negligible growth was observed. So if you see in figure 6, which is the treated F-oxysporum, and compare it with figure 5, there has been less growth in T2. Also, 3 replications of each of these treatments were taken. Their mycelial mats were taken out, and their fresh weight, air-dried weight, and oven-dried weight was noted. Now, and they were tabulated as shown in table after figure 7. We also calculated the percentage decrease in weight of T2 compared to control and noticed a good decrease, especially if you observe for oven dried, we saw a decrease by 9.756%, which is good. Now for objective 2, we estimated a total soluble protein using a method given by Lowry et al. In figure 8, from right hand side to left, the right hand side corresponds to the control, the middle one is for T2, and the last being a blank. Now the absorbance for these were measured, and it was found to be 0.098 for the control and 0.073 for treatment. And then we plotted them in the absorbance and protein concentration graph. If you now see or focus on the graph, the absorbance of control given in red, that is 0.098 for the control, and corresponded with the x-axis, that is the protein concentration. The protein concentration is more for control compared to T2, which is the AGNP treated Fusarium oxysporum, whose absorbance is given in purple, that is 0.073. Thus, you can see that it has a considerable effect on the protein. So, not wasting a lot of time, we come to 2 broad conclusions. This study supports the hypothesis that silver nanoparticles can be used to make new fungicidal materials, and that they have an inhibitory effect on protein synthesis of Fusarium oxysporum. Of course, much more research needs to be done for further analysis. And so with this, I will end my presentation.
Okay, thank you, Parishmita. And participant can
within time?
reach their query through our email ID, okay, because we have limitation of time. Next, Dipanita Vishwas.
Thank you, sir.
She is a present, sir.
If not, then we can proceed to Munni Das.
We will share, sir. Dipavinta, are you present?
And Dr. Rajesh, please share Munni Das slide.
Okay, sir. Munidas.
Because we have problem of time.
Yeah. Munidas, please unmute.
Yes, sir.
Just a minute. We are sharing your poster. Yes, proceed.
Proceed, please.
Good afternoon to all, respected chairman, co-chairman, rapporteurs, and organizing committee members. I'm Munni Das, going to present before you a poster on nanotechnological approaches for preservation of food. I'm from the Department of Food Science and Technology from Assam Agriculture University. Regarding introduction, I would like to say that nanotechnology is the study and manipulation of matter at incredibly small sizes, generally between 1 and 100 nanometers. The rapid development of nanotechnology has transformed many domains of food science, especially those that involve the processing, packaging, storage, transportation, functionality, and other safety aspects of food. In food science, nanotechnological applications include a specific type of nanomaterial that is incorporated into a specific food product in order to develop certain desired properties. Sir, the full poster is not visible.
Just a minute.
Okay, sir.
Yes, please continue.
Novel and nano antimicrobials have shown promising effects on safeguarding food deterioration. As shown here, though different types of nanoparticles are there, most commonly silver, gold, zinc oxide, silica, and copper are used in food industries in a different way, and this can be used mainly as nanoemulsions because nanoemulsions can be used as a delivery system where actually bioactive compounds are loaded. Nanoemulsion based delivery systems improve the bioavailability of the nano encapsulated bioactive compounds and increase food stability. As shown here, this can be utilized for food matrix design where composition and structures are studied followed by physicochemical properties and then sensory attributes. Ultimately, fate of the bioavailability of nanoparticles are studied. Similarly, we can see here the other use of nanoemulsion is as excipient foods, where nutraceutical-rich foods are consumed with non-loaded excipient nanoemulsions. Now, if we look at this diagram under the heading Use in Food Industry, then it will be more clear that nano-encapsule means food ingredients will be converted to smaller particles, and over that smaller particles, a capsule will be generated.
Then we may use some material which has the ability to alter the pH of GI fluids so that nano encapsule gets dissolved and food materials release, and thus it becomes available for the growth and development of human body. In food industry, nanoparticles can be used in different manners such as nanocoatings, nanocomposites, and then nanoencapsulations. In nanoencapsulations, a particular green nanoparticle is encapsulated within certain polymers. Then comes nanosensors. Nanosensors can detect pathogenic bacteria, food contaminating toxins, taste, smell, etc. In packaging material, say in case of food container, if we use silver or copper coated nanoparticle, then in that container, when we put a particular food product, it will prevent the microbial growth and ultimately we can get a longer shelf life. Nanoparticles like copper, silver, gold, zinc oxide has antimicrobial agent. So if we mix these nanoparticles with food materials, then there will be no question of contamination with other microbial agents. Now, there are huge numbers of future prospects of nanotechnology in food technology, of which I want to mention few of them. Number one, smart packaging to detect damage. Number two, antigen-specific biomarker to detect the spoilage organism. Number three, RFID, that is radio frequency identification-based nanotransistor to detect changes in abiotic factors such as temperature, humidity, etc. From these, we can conclude that nanotechnology has a great role in prolonging the shelf life of food products, and the use of an appropriate nanotechnique can help in the production of high quality and wholesome food products. With this, I want to end my presentation. Thank you.
Okay, thank you, Manidas, for your presentation.
Yes, sir.
And next, Dipanita Vishwas,
Yes, sir, I am present.
please. Please share your screen.
Yes, is my slides are visible?
No, no.
Hello, sir.
Not.
No,
You
sir.
please. Yes.
It's visible?
Yeah, it will take only few seconds. Just a minute.
Yes.
Your desktop is visible.
Yeah, it is visible?
Yes. Now you can proceed.
Yes. Okay. So a very good morning to all our respected chairman, co-chairman, rapporteur, dignitary, and participant. I am the Banvita Bishwas from Maulana Abul Kalam Azad University of Technology, going to present my MSc dissertation research work, that is, silver nanoparticle and zinc oxide nanoparticle are compatible with fungal biocontrol agents. Chemical pesticides are associated with environmental and human health hazard. Its toxic nature and development of the pesticide-resistant to pest encourage the use of biological pest control agents. Several studies have been reported that nanoparticles inhibit the growth of plant pathogens. So the combination of nanoparticle and biocontrol agent might be helpful in the plant health management. Objectives of the studies were to understand the effect of nanoparticle on beneficial microorganism and the compatibility of the selected nanoparticle with beneficial microorganisms. For the compatibility study, Trichoderma hertzianum and Beauveria bassiana were used. Commercial silver nanoparticle, microsilver nanoparticle that is biologically synthesized, and zinc oxide nanoparticle that is chemically synthesized has been used. For the method, we use poison food technique where the fungal biocontrol agents were allowed to grow on a media suspended with nanoparticles. From the study, it can be concluded that the commercial silver nanoparticles are compatible with both biocontrol agent up to 5000 ppm. Microsilver nanoparticle have compatibility up to 400 ppm, and zinc oxide nanoparticle have a compatibility up to 300 ppm with both biocontrol agent. So this is the study. Thank you.
Thank you, Dipanita, for completion your presentation within time. So can we proceed to next presenter,
Thank you, sir.
Punavati?
Yes, sir. Yes.
Punavati hai naam? Probably she is absent. Then TD Parmar, Dr. TD Parmar.
Yes, she is, I think, present, sir.
Okay, please.
Any screen presentation share करूं, doctor?
Hello, sir. Hello.
Yeah, please proceed. Hi, madam.
Okay. Good afternoon to respected chairman and co-chairman and all participants present here. My name is Tanvi Parmar and I am going to present. Hello. Hello.
Your
You are audible, Tanvi. Continue कर जाए, okay?
Sir, I cannot see the screen.
Just a minute.
Okay, sir. Hello.
Yes, you can proceed, madam.
Okay,
Then we
sir.
start.
My name is Tanvi Parmar and I am going to present my poster on green synthesis of silver nanoparticle using Azadirachta indica leaf extract and their antifungal activity against Alternaria solani causing early blight of tomato. My main objective of this study to determine the inhibitory property of neem-based silver nanoparticles on colony formation of Alternaria solani. In the introduction, in Middle Gujarat area, early blight cause very severe disease. Because of that, occur very heavy yield loss. As a part of my research work, I took this problem and I isolate the fungus and for species identification, I done my molecular using ITS1 and ITS4 universal primer, and I found the name Alternaria solani, and I also did in vitro evaluation of plant extract against Alternaria solani, and I found neem was very good efficacy. And after that, we decided to use neem with silver nanoparticles for check the inhibitory effect of silver nanoparticles against Alternaria solani. And the methodology we can see in the flow chart. First of all, take 5 gram of fresh neem leaves and put in the beaker with 100 ml of water and cover the beaker with aluminum foil to avoid direct contact of light. Then put the beaker in water bath with 95 degrees temperature for 40 minutes. After 40 minutes, neem extract collected and used for further activity. After that, add 8 ml of neem extract with 192 ml silver nanoparticles, uh, and mix AgNPS and neem extract. After that, put the flask in 48-hour room temperature in dark place. After 48 hours, the solution color was changed in light yellow to dark brown color due to the bioreduction of Ag plus ion into Ag0. The silver nanoparticle solution was centrifuged at 30,000 rpm for 20 minutes, and after centrifugation, collect the silver nanoparticle, and these collected silver nanoparticles kept at 100 degrees Celsius for 24 hours to remove excess water, and the powder silver nanoparticle were used without further modification.
Madam, your voice is not audible.
Tanvi, you are not audible.
Hello.
Yes, please proceed, madam.
Yes, sir. After the results show that the person growth inhibition at different concentration were recorded and presented in table 1. Among the 8 treatment, biosynthesized silver nanoparticle with concentration of 200 ppm proved to be the most effective with 59.26% growth inhibition of Alternaria solani. I conclude it is evident from the data that as concentration of silver nanoparticle increased, there was a corresponding increase in growth inhibition of pathogen, and this contributed to minimize the risk and hazard of toxic chemical fungicide, especially on vegetables which produce for the fresh consumption. Thank you.
Thank you, madam.
Yes, sir.
So our next presenter is Sumit Jani. Sumit Jani.
He is a
Sumit
present.
Jani, are you present?
Yes, he is a present.
Okay.
Sumit Jani. शेयर कर दे पे वांदो नहीं. Sumit, this is the first call for you for the presentation.
She has submitted the presentation to you.
Sir, he submitted. We are
Okay.
sharing it. Sumit, please continue. એને unmute કર્યો છે ને.
Hello, am I audible, sir?
Yes, you are audible. Continue.
Okay. Good afternoon, respected teacher. Myself Sumit Narayan Bhai Jani. I am from Sardar Kushinagar Dantewada University. I am a student of MSc Scholar, Department of Biotechnology. So today I am going to present my poster presentation on green synthesis of silver-based nanoparticles and then potential for the antimicrobial activity. So starting with the era of the nanotechnology. Nano means the dwarf, means extremely small. In 1959, the Richard Feynman the considered as the father of nanotechnology. Then after the nanotechnology is the interdisciplinary science which deal with the smaller dimension. So the first term regarding this research of nanoparticles. So nanoparticles are the ultra fine particles which are the size is less than 100 nanometers.
Sumit, be specific. Go in our objective and methodology.
Okay, sir. Okay. So starting with the green nanoparticles. Green nanoparticles are from the plant-related nanoparticles which are synthesized from the silver nitrate. Our objective is to synthesize the optimization and physical characterization of silver nanoparticles and to study the antimicrobial activity of silver nanoparticles. First of all, I collect the plant sample of the plants selected were Catharanta roseus because I studied that which have high reducing potential activity are present. And then after the synthesis of silver nanoparticle, after the physical characterization, then after performing the antimicrobial activity. So first of all, I collect the plant sample of Catharanda roseus. The common is periwinkle. The common name is Parmacea. Then after I prepare the plant extract. Then after the reduction of the you clearly show in the figure that the AgNO3 convert into the dark brown color. Their preliminary indicate that this is a confirmation of nanoparticles. Then the nanoparticle are characterized by UV visible spectrophotometer. You clearly see then the peak was 100 for 20 nanometer around its results. Then after the DLS based particle size and zeta potential analysis, the particle size was present around 50 nanometer, and the zeta potential indicates that the stability of nanoparticles. Then after performing the FTIR based functional group analysis, which are how the mechanism of nanoparticles and our plant extract, the mechanism is to provide to how the nanoparticles are coating to the secondary metabolites interpretation. So secondary metabolites are coating to the Ag group and reduction throughout the whole reduction and Ag plus 0 ion, then are converting to the silver based nanoparticle. Then after the we are performing the functional group which are indicated which are bending shifting on the basis of nanoparticle through the FTR activity. Then after the TGH, thermogravity analyzer. So how our nanoparticles are affected on the basis of this temperature effect. So we are performing the TG analysis, thermogravity analyzer. So 40% weight loss, and we measure, and then 1000 degrees Celsius temperature, we are nanoparticles are stable. Then after last figure you so that the antimicrobial activity. So first of all, these two plates are from the left side are present of E. coli and as well as the commonly human plant pathogenic bacteria. So our target is basically plant pathogen. The last of right side plate are the antifungal activity of silver nanoparticles. So target bacteria, target plant pathogen are the commonly plant pathogen from the SDAU University are Alternaria bansai and Fusarium oxysporum. Because we know that the Alternaria bansai are the commonly plant pathogenic fungus for highly for the cumin blight disease. So we have the significant result from our nanoparticles to comparison the commonly then after the commercial effect of hexaconazole, we are comparison of a silver nanoparticle. We are significant gave result. So outcome of this outcome of my research, the preparation of plant mediated and non toxic nanoparticle which are highly consumed from plant world for antimicrobial activity for plant pathogen, and there are the in future we are converting into the nanopesticide formulation to against a different type of plant pathogen. Okay, sir. Thank you. Any regarding query, please ask the question.
Okay, thank you.
Thank you, Sumit, for the presentation. Now sir, with the permission of the chair, now move toward the chairman and co-chairman. Pranav Datta, sir.
Okay, so I sincerely congratulate all the presenters for their very nice and informative presentation. And thank you to my co-chairman, Dr. P. Tamuli, then rapporteur, Dr. Patidar, and Dr. Mahesh Pathak, and more particularly the organizing committee for organizing such a beautiful virtual national seminar and selection of, say, me and my team as chairman, co-chairman. National seminar on plant health management. Thank you, Dr. Rajesh, as well as the organizing committee.
Thank you very much, Pranav Datta, sir, for chairing the session. Dr. T. Muli, sir, for co-chairing the session. Dr. Patidar, sir, and Dr. Mahesh Pathak, sir, as a reporter. Sir, as you have heard the session, now moving towards next session. So the next session is on the pesticide residue, and this session will be chaired by Dr. L.V. Getia, sir, co-chair by Dr. K.D. Parmar, sir,
Okay, that's why I called you. Thank you. Okay, I am highly thankful to Dr. Rajesh Wagunde, who has handed over the session relates on the pesticide residue. I am with my colleague Dr. Kaushik Parmar and reporter Dr. Singh. Friends, first of all, I welcome all the participants in this session. We have 12 oral presentation and 8 poster. I request all the scientists, please rapidly you should go directly with the results. Don't focus on introduction due to time limits. Please rapidly go to the results part and finally conclusion. Okay. So first,
there is a one.
Hello.
Yes, sir. The second speaker, Ishrat Jan, is going for the poster instead of the oral. Please take a note, sir.
Okay.
Okay. So I request to Dr. This Vishwanath Purvella for the oral presentation. Vishwanath Raghuteja Purvella. Okay, sir. Gettiya, sir.
Hari.
Sir, continue with your first oral and then poster, sir.
Yeah, exactly. Okay.
Now the session is yours, sir. You have to take the charge of the platform. Okay, sir.
Okay. Vishwanath. Vishwanathan. Is Vishwanathan is there? So kindly share your PPT.
Sir, from
is Vishwanathan is present or absent?
Sir, he is login, but I think he is physically absent. Technically present.
Okay, okay, so, sir. Then we will go with the third participants. Don't waste our time. Okay.
Yes, sir. You continue.
So I request Dr. K.D. Parmar for the presentation. Sir, he has 4 presentation. So I request Dr. K.D. Parmar to present all the 4 papers simultaneously without interruption. And at the completion of one paper, we will allow for the interaction and then we will proceed for the next. Okay, I request Dr. K.D. Parmar for the presentation, please. Dr. K.D. Parmar, please share your screen. So it is visible? My screen is visible?
Visible, sir.
Can anyone reply? Okay.
Yes, you are visible, sir.
Yeah, good afternoon all. I am Dr. K.D. Parmar, working as an assistant residue analyst at ANPN Pesticide Residues, Anand Agriculture University, Anand. Today I am going to present my one research, that is residues and persistence of thiophantasm 25WG in mango and its risk assessment. So without touching this introductory portion, I just skip the introduction portion because of lack of time. The field information, the crop was a mango with a Kesar variety. The experiment was held at horticulture farm, BA College of Agriculture, AU Anand. The soil type is sandy loam, plot size 25 meter square, that is 4 trees per one replication we have taken. And the design
in experiment was completely randomized design with 4 application, and the thiamethoxam was applied at 25 and 50 gram active ingredient per hectare. Means this 25 is the recommended dose, while the 50 gram is a double of the recommended dose. And the water volume was 1000 liter per hectare. And this is regarding how we are taking the samples in the 4, of course, in the 4 applications. That raw mango we have analyzed, that approximately 1 kg raw mango was collected and with peel and stone, and a representative sample was analyzed. While in the case of ripe mango, 1 kg was collected and with peel without stone. In the ripe mango, we have removed the stones. And in soil, a 1 kg soil was collected, and it so for some further under shade and ground to pass through 2 mm sieve, and subsample was drawn for the residue analysis. And after application, 0, that is 2 hours after spray, 5, 10, 15, 21, 25, 28, 35, and 55 days after last application, the sample was taken. And the whole analysis was carried out on LC-MS, that is liquid chromatography mass spectrometry, and that make up Simaju, Nexera X2, and MS is 8050. And our LOQ is 0.01 microgram per gram, that is ppm. That is for parent compound, that is thiamethoxam, as well as its metabolite CGA 302704. And before start our actual analysis, we just confirm that method which we have adopted is suitable for to extract our target compound or not. So for that we carried out a recovery study that we call a method validation data. So we have carried out recovery study at 3 level, that is 0.01, 0.05, and 0.1. That in both the compound, that is thiamethoxam and its metabolite CGA 302704. And actually the CGA is a clothianidin. It's a metabolite of thiamethoxam. And we have found this our recovery was in the satisfactory range. So we can adopt our method to extract our target compounds. Something. Just a moment. So this is a whole extraction procedure, because it's a very well established procedure. So I'm not going to touch it, but simply we have used the capture method, that is quick, easy, cheap, effective, rugged, and safe for the sample preparation as well as sample cleanup. So before injecting our samples in our instrument, we need to clean our samples, because there are so many co-extractives which are extracted with our samples. So we need to clean up all the samples. So for that we need to require a clean up our samples. And in the case of soil also, we require to extract our soil samples to all the present pesticides in our soil that we need to extract in our organic solvents, and we are going to inject that organic solvent in our instruments. So this is a procedure for extracting the soil samples. And this is a LC-MS parameters. The column was Fortis C18, 1.7 micrometer, 2.1 mm inner ID, and 50 mm length. And these are the scan modes. This positive and positive scan mode we are analyzing. And a precursor ion and product ion. On the basis of precursor and product ion, whole the analysis was confirmed. And these are the some LC time program. while in the double of the recommended dose, the same trend was found, and also in 35 days it was found below the determination level. But in ripe mango, which was collected on 55 days after last spraying, the residues of thiamethoxam, that is 0.01, and in higher dose that is 0.015. So it shows the thiamethoxam is quite persistent pesticide. Even in ripe mango, it was found at the slightly above the determination level. And in soil, it was found in below the determination level, which also was collected at 55 days after last spraying. While in the case of this metabolite clothianidin, it was of course the trace amount of clothianidin was there, but at the last application, it was found below the determination level of 0.01 milligram per kg. So based on this, we have taken this risk analysis while working out the maximum permissible intake MPI, that is 4.8, and it is calculated with the help of this body weight and acceptable daily intake. This acceptable daily intake of thiamethoxam in mango is 0.08 milligram per kg body weight per day. So from that we can calculate the TMRC, that is theoretical maximum residue contribution, with the rural and urban in both the peoples. In rural it was found is 0.15 and in urban it is 0.22. While this TMDA, that is theoretical maximum dietary intake, that also in rural 1.10 and 1.64. Supposing your TMRC and TMID is below this MPI value, means the recommended dose of pesticide can be allowed for the spraying in the mango. And another most important term, this hazard quotient. If your hazard quotient 0.015 and 0.021 is below your acceptable daily intake, below the 0.1 ppm, then it can be allowed and it can be recommended to spray in mango. So conclusion, after this all analysis, this 3 foliar sprays of thiamethoxam 25 Wg in mango at 10-day interval at recommended dose, that is 25 and 50 gram active ingredient per hectare, starting from fruiting stage, resulted in residue below the MRL 0.2 milligram per kg. That MRL is given by FSSI in mango fruits if harvested 7 days after the third spray, because of course our concentration is not 0.2 at 7 day, but in fruit crop the minimum 7-day waiting period must be there. So we are suggesting that 7 days after the spray you can harvest your mango. And considering the MRL FSSI 0.2 milligram per mango, the insecticide can be recommended on mango with 7 day of pre-harvest interval. Thank you.
Okay, thank you, Dr. Kaushik. Now, anybody wants to interact? Only 2 questions. If anybody wants to interact rapidly. Okay, thank you. Next research paper for the presentation. Again, I request Dr. Kaushik for the next presentation.
This residue and dissipation pattern of tetranilipol 200 SC and its metabolite in tomato fruit and soil. Actually, this experiment was carried out at Agriculture University main vegetable research station with the variety of GT5, and these all are agronomical practices and field trial informations. But the dose of that tetranilipol was 60 and 120 gram active ingredient per hectare, and the water volume required was 500 liter per hectare. Same the sampling data, 3 application, and in the case of sampling data collection, the fruits approximate 1 kg tomato fruits were collected, homogenized, and representative sample was drawn for the residue analysis. While in the case of soil, 2 kg representative samples were collected, dried under shade, and ground to pass through 2 mm sieves. And the interval between application and sampling was at 0, that is 2 hours after spray, last spray, 1, 3, 5, 7, 10, 15, 20, and 30 days. And again, it was analyzed on LCMS-MS, Symposium 8050, and the LOQ was a 0.05 milligram per kg body weight. Means my method can extract up to low level up to 0.05 milligram per kg of this tetranylprone as well as its metabolite DCS-CQ63359. Recovery data was also generated and it was in acceptable limit. As per the Sante guideline, your recovery must be at 70 to 110%. So all the recoveries were within the acceptable range. And this is the extraction procedure, same method. Quencher method was adopted to extract the pesticides from tomato fruits and soil. And these are some LCMS parameters which we have adopted while analysis of this tetranylprone and its metabolite. And looking to the results after in the recommended dose, after 2 hours, the initial deposit of this tetranylprone was 0.2 ppm at recommended dose, that is 60 gram active ingredient per hectare, which was persist up to 26 ppm. Means on first day it was 0.3 ppm, 3 day 0.29, but this pattern was highly erratic because of this kind of little bit systemic nature of this tetranylprone. The dissipation pattern was highly erratic. See, same in the case of high dose as well, 0.39, 0.34, 0.52. But in the case of metabolite, initially it was a below the determination levels, 0.05. But it was at 0.1 on 7 day. Means the metabolism take place after the application of the pesticide after 5 days. So if we are looking to the dissipation pattern, it's highly erratic. Okay, so based on that, the conclusion is the 3 foliar spray of tetranylprone 200 SC in tomato at 10-day interval at the rate 60 and 120 gram active ingredient per hectare, starting from fruiting stage, resulted in residue below the MRL because MRL is not established. So we have taken the 0.05 microgram per gram as a default MRL. So based on that, if the fruits are harvested from 15 day after the third spray, the PHI of 15 day could be suggested if the tetranylprone is recommended in tomato.
Okay, thank you, Dr. Kaushik. Again, the topic is open for discussion. Only 2 questions from the audience. Okay, again I request Dr. Kaushik for his next presentation. Dr. Kaushik, please.
Third, my topic is residue dynamics of chlorpyrifos 20EC in or on groundnut. That experiment was also conducted at Anand Agriculture University with the variety of TG37, and these are the all agronomical practices. Plot size and spacing and date of sowing was in 2017, and time of application was a pod formation stage, and the application was a 200 and 400 gram active ingredient per hectare, and water volume was required 500 liter per hectare. Here the sampling data, sample weight and preparation. Groundnut leaves also we have analyzed, and 500 gram samples was collected, homogenized and represented sample was drawn for the residue analysis. We have also analyzed the groundnut pods. 1 kg pods were collected, separated for the seeds and hilum, and we are analyzing seeds and hilum separately to get the actual residue persistence of this chlorpyrifos in groundnut. So hilum also analyzed separately, and seeds were coarsely ground. Appropriate waste was taken for this. Rest of the sample was subject to the oil extraction. And for the oil extraction, we have adopted the sock slate method. So this oil also we have collected from the sock slate method. This oil also recovered after sock slate extraction was subjected to residue analysis, and whatever the leftover portion of that seeds, that calls a cake, and that we have also analyzed after extracting the oil from the with the help of sock slate extraction. The coarsely ground seeds left after the oil extraction were used for the residue analysis. And it's for the soils, 500 gram of soil samples were collected, dried under shade and ground tea pass through 2 mm sieves. And interval between the last application and sampling was 0, that is 1 hour after application, 1 day, 3 day, 5 days, 7 day, 10 day, 50 days, and 25 days. And the spore and soil were collected at 67 days after the last application. And the whole experiment analysis portion was carried out on GC, that is gas chromatography, that is 1310 make of thermo with the equipped with flame photometric detector. And the LOQ was 0.05 microgram per gram for leaves, hulum, oil, cake, and soil. Here also recovery experiment was also carried out, and the recovery was found within the acceptable range. And this is the extraction procedure of grain leaves, same QHR method. And this is the extraction procedure for seed, oil, and cake. And this hulum was also separately extracted and analyzed. And this is the extraction method for hulum and soil. And these are the some GC parameters. The column was capillary column, 30 meter length. And this is a home programming, initially 160 degrees Celsius temperature, and the ramp was 60 degrees Celsius per minute, and which was completed at 300 degrees Celsius per minute. And these are the some injected temperature, detected temperature, and by this calculation residue in ppm, we have calculated the actual concentration of this chlorpyrifos in groundnut matrices. So this is an instrument, gas chromatography, equipped with a FPD. While looking to this dissipation pattern in grain leaves, the initial dose of concentration of chlorpyrifos was 5.30 ppm at recommended dose, while in double of the recommended dose it was found to be 15.82, means it's very high, but it degraded rapidly, and on 10th day it was found to be 0.07, while on higher dose it was 0.08 on 20 days. Means in lower dose, in recommended dose, it was found below the determination level on 15 days, while in the double of the recommended dose, it was found below the determination level on 13 days. While in the case of seed, hulum, oil, cake, and soil, all the samples are free from chlorpyrifos residue, means the trace amounts was already there, but it was below the determination level of 0.05 ppm. So after this whole analysis, the conclusion, the 2 foliar spray of chlorpyrifos in groundnut at 10-day interval at the rate 200 and 400 gram active ingredient per hectare, starting from pod formation stage, resulted in its residue below the MRL of 0.05 microgram per gram. Of course, the MRL is not, but we have taken the 0.05 microgram per gram MRL as a default MRL of at a level of LOQ. And in the groundnut, if plants are harvested from 10th day after the second spray, therefore PHI of 10th day could be suggested if chlorpyrifos is recommended in, here groundnut.
Okay, thank you, Dr. Kaushik. Now topic is open for discussion. Again, I will allow only 2 questions, but audience are, I think, inactive. Again, I request Dr. Kaushik for another presentation. Dr. Kaushik, please.
I also carried out the one experiment on the VD side, because nowadays VD side much more amount the farmers are using this VD side like pendimethalin in chickpea. So this the experiment was carried out at agronomy farm, BA College of Agriculture, with the variety of Dahot yellow, and it was designed in the randomized block design, and the method of application, this is mostly pre-plant incorporation by incorporating in 2 to 5 centimeters of soil with knapsack sprayer fitted with flood jet nozzle. Means the application was carried out before the planting of this chickpea, and at the rate of 677.25 and 1354.45 gram active ingredient per hectare, and water volume was required 500 liters per hectare. And these are the sampling data. A chickpea plant was approximately 500 gram well collected, homogenized and representative sample was drawn for the residue analysis. Grain pod, when collected is at the grain pod formation stage, also collect the 500 gram and homogenized and representative sample was drawn for the residue. This green pod are analyzed with its outer layer, not only grains. This whole green pods was analyzed. While dry prod, approximate 500 gram were collected, homogenized, and a representative samples was drawn for the residue analysis. While in the case of soil, also we have analyzed, and the interval between last application and sampling was 30, 35, 40, 45, 50, 101, and 136 days after application. And it was analyzed on GC, gas liquid chromatography, equipped with electron capture detector, and the LOQ was 0.05 microgram per gram in all the chickpea matrices. Also, we have carried out the recovery study, and the recovery was found within the acceptable range. And this is a procedure, extraction procedure of chickpea plant. This is the procedure of chickpea grain and dry pod and soil. And these are the instrumental parameters. This is a picture of this that instrument is gas liquid chromatography equipped with electron capture detector. And looking at the data, before showing application of pendimethalin at recommended and double of the recommended dose, the samples were collected 30 days after application. It was found below the determination level in all the samples collected at different periods. Okay, even in green pod, dry pod, and soil also it was collected, and all the samples were below the determination levels at 0.05 milligram per kg body weight. So based on that, the conclusion is pre-plant incorporation of pendimethalin chickpea at rate 677.25 and 1354.45 gram active ingredient per hectare resulted in residues of pendimethalin in chickpea were below the determination level at either dose, in lower as well as in double of the recommended dose, collected at 30, 35, 40, 45, and 50 days after application. So this residues of pendimethalin in green pod also found below the determination level in standard and double of dose when collected at 101 days after application. While in dry pod, the soil collected harvest did not reveal any residues of pendimethalin at either dose.
Okay, Dr. Kaushik, I request you go through your chat box because we are not allowed the many as a unmuted person. So the question is asked in the chat box. So please refer the chat box and you can solve the query from the different scientists asked in the chat box. Please, Dr. Kaushik.
One question I've asked: what is BCS-CQ63356? It's a code of metabolite of that particular compound.
Is it a metabolite of thiamethoxam, sir?
Yes, sir. Yes.
Oh.
Another question is there, Dr. Pano. The matrices are different. How the similar log was established? Why the samples are not collected on early stage? I think that question was asked from Dr. Susil. Why the samples are not collected on early stage? Kindly let me tell you. Kindly let me know in which crop you are talking about.
We have to unmute first, Dr. Susil. We have to find it out.
Okay.
Yeah. Please allow Dr. Susil. Unmute it for the discussion with the Dr. Susil.
I think he might be asking about the chickpea.
Yes. Sir, you are unmuted now.
Yes. Of course,
Dr. Parmar, do I audible to you?
Hello.
Yeah, exactly, sir.
Regarding the chickpea, you have taken the observation from the 30th day. But Dr. Kaushik, you know, in North India, the chickpea leaves are also consumed as for the saag preparation. So why don't you take the initial stage of the plant stage? Because leaves are very
Yes,
much
sir, we have
consumable.
applied this pendimethalin before the sowing, but of course we have taken the samples when the germination was start. But at that time also, it was found below the determination level because the germination was start after 8 or 10 days. So we might have able to collect the that much sample. And at that time also, but this thing was not included in our protocol, so I didn't include it in my this research paper. But at that time we have collected and it was found to below the determination level of 0.05 ppm.
So, Dr. Parmar, you have to first mention in your presentation what is the objective of the study, because unless the audience don't know what is the actual objective of the study, it is very difficult to correlate with the result what you have presented in your presentation. One more thing.
Yes, sir. I already included it in my introduction portion, but because of the lack of time, I just keep it. Because in each and every introduction, I included my objectives.
But you have to mention in one another time what is the objective of the study. Another thing, in many of your study, particularly in chickpea or somewhere else, where you have taken so many matrices, plant, leaves, soil, and your LOQ is the similar. As far as the study are different, the methodology are the different. However, the instrumentation is the same, but the limit of quantitation value will be the different. How you can come forward with these similar values? Is there any coincidence, or this is nothing but the observation you have presented in the seminar?
No, sir. We have carried out our method validation portion with different concentration, but at 0.05 ppm, we are getting a satisfactory result.
But Dr.
So that's why we kept a 0.05 as a default LOQ.
But Dr. Parmar, in the case of, as you mentioned, you have followed the Center guidelines. In Center guidelines, you have to perform the recovery study at the level of the LOQ. So, but you have across the board, you have taken 0.05 ppm. I think this is the biggest lacuna of your study.
Yeah, 0.05 is the LOQ value.
It is a LOQ value.
Below that, we can achieve the acceptable range, but at the safe side, we can keep as the 0.05 LOQ default value.
Okay.
Okay, thank you, Dr. Sushil, for nice interaction, nice discussion. And I am highly thankful to you to narrate it that now onwards, I request all the speaker, please first you go through the objective. Because of my instruction, Dr. Kaushik had not narrated the objective because it was a part of the introduction. But leave the other introductory portion, only focused on objective, and then you proceed for the presentation. Now again, I think the first scientist, Vishwanatha Raghuteja Puwala, please. Is Dr. Vishwanatha Raghuteja Puwala, please share your screen. Dr. Rajesh?
Yes, sir. I'm just unmuted him.
Vishwanatha Raghuteja Puwala. It is on third number in the general list, sir.
But I think he is not in list, participant list right now.
This is not online.
He is not online, sir, right now.
Okay, then
okay, sir.
So, is that John?
No, he is there.
Okay.
Vishwanath.
Then I, sir, then I request Nitesh Litoria.
Sir, good morning.
Vishwanath is there. Vishwanath Raghuteja is there.
Okay, then please allow him first.
Vishwanath,
Vishwanath Raghuteja Povalla.
Vishwanath, unmute yourself.
Okay, sir. Yes, sir. I have unmuted, sir.
Okay.
Can you hear me, sir?
Please set your screen.
Sir.
Vishwanath.
Dr. Vishwanath, please share your screen.
Sir, can you share my screen, sir? Sir, I don't know that we have to share, sir.
Just wait 1 minute, huh?
Okay, sir. Okay, sir. I will wait, sir.
Vishwanath.
Sir, tell me, sir.
Do you have your presentation right now?
Sir, I have it in my laptop, sir, but I don't know that. I thought that you will share the screen, sir. That's why it is not there in my phone, sir.
So, you have submitted to us?
Yes, sir. Yes, sir. I have submitted, sir.
Oh. Next. Okay, we will search it, and after some time, you can join. Getia, sir.
Yeah, okay, Dr. Rajesh, I request both Vishwanatha and Dr. Rajesh, please communicate, and again you just ask Dr. Vishwanatha to send or resend the mail, okay?
Sir, can I resend it now,
be
sir?
ready after this. So I now I request Nitesh Litoria. Nitesh Litoria, is he on screen? Nitesh Litoria, please unmute.
Yes.
Okay, Kaushik, you
now you are unmuted.
Call him here too, so that he can also. Yeah, okay, he is here. Nitesh Litoria, please.
Can you hear me,
Share your
sir?
screen.
Yes, Nitesh, you can share your.
Yes, very good.
Okay, good afternoon to all of you. Here I am presenting my work on dissipation behavior, residue status, and risk assessment of a combi product, that is trifloxystrobin plus propinel in tomato. So as we all know that tomato is one of the most common consumption human diet, and due to its antioxidant property, it acts as a functional food which benefit to human health. On the other hand, tomato is very susceptible to this different type of pest and disease, which result in the extensive use of different type of pesticide. Nowadays, control and cure of this disease mostly depend on the fungicide. Nonetheless, after their application, pesticide residue may remain in the crop and create a health risk because of their toxicity. So by this brief introduction, objective is after applying of any pesticide, its behavior on plant, desiccation pattern, residue status on fruit, and its risk assessment is of particular concern to the consumer safety. So here is the experiment detail, variety, season, design, plot size, method of sowing, treatment details. So these are the extraction method for the tomato fruits as well as soils. For trifloxystrobin and its metabolite CZA321113, that is acid of trifloxystrobin, we follow the Quacher method, and for particular propinel, which done as a carbon disulfide, that is dithiocarbonate, this method was followed. So these are the parameter for the trifloxystrobin and its metabolite, and these are the LCMS parameter, and these are the GCMS parameter for propinel as carbon disulfide. So before starting any experiment, we have to standardize the method, whether that method is a particular suitable for compound and as well as crop. So we standardize the method for trifloxystrobin in tomato and soil. So these are the recovery data for the fruit and soil. These are the recovery data for its metabolite in fruit and soil, and these are the data for propinel in tomato, fruit and soil. This all our data is relevant with the scientific guideline, that is 70 to 110% in any matrix. So these are the desiccation pattern of trifloxystrobin, in which it shows that by default we consider, yes, earlier sir asked how do we decide LOQ. Generally we don't follow the LOQ for particular compound as well as particular commodity. We generally do multi-residue method, in which entire all compound as well as all commodity at a certain level gives the best result. So we follow that particular reading as a LOQ. So here 0.05 is LOQ. So at 15 days, the decibels at standard dose, the decibelation becomes the level of below the level of quantification, and in higher dose it becomes after 20 days. So throughout this study, we did not detect the metabolite. Here is the desiccation of propinel, in which same pattern was followed. 15 days after 15 days, standard dose become the below the level of quantification, and in double dose it becomes after 20 days. So here is the desiccation pattern of the compound in different tomato fruits. This is the desiccation kinetics, half-life and regression coefficient of trifloxystrobin as well as propinel in standard as well as double dose. We also carried out this assessment for the human safety. So by considering the acceptable daily intake, that is 0.04 for trifloxystrobin and propinel, it is 0.007. We calculated anticipated residue contribution of each fungicide, which is calculated by multiplication of tomato consumption and average body weight. From that, we calculated targeted hazard percent, which is observed. I will show in next table, which observed less than 1. It shows there the maximum possibility of obvious toxic effect. And from this targeted hazard percent, we calculated the hazard index for both the fungicide. And if this hazard index is less than 1, that indicates that it is safe for human consumption. So here is the table in which we can see that hazard index is 0.067 for rural area, because I have calculated rural and urban, because the consumption pattern of rural and urban is the different. So here hazard index for rural and urban are less than 1. So we can see that at the recommended as well as double dose, if we consume the tomato fruit, it is safe for human health. We also calculated the TMRC, that is theoretical maximum concentration, residue concentration of particular pesticide at different days. And generally it was said that if it is less than the MPI, that is maximum permissible intake. So here we have calculated the TMRC value for each days for each dose, that is standard dose and double dose for trifloxystrobin and propinel standard dose and double dose. So this TMRC value here it is in microgram, and maximum intake is 0.24 and 0.4. So in each dose, all the values are less than the MPI value. So we can say that consumption of that particular at any days, it is safe from human risk. So conclusion is 3 failure applications were given, and initial residue was found 0.15 to 35 for trifloxystrobin and 0.47 to 0.90 for propinel. And thereafter it declines and becomes below LOQ level at 15 days for standard dose and 20 days for double dose. So here is the apply, and by considering the MRL of trifloxystrobin, that is 0.7 and carbon disulfide 2.0 in tomato group, the PHI of 1 day can be recommended. And in this study, the THQ targeted hazard percent and hazard index was less than 1 in both the pesticide and standard and double dose. As well as TMRC is also the below the MPI. So we can say that application of transfloxystrobin and propinep on tomato does not show any human health risk at standard dose, as well as also we can say sometimes farmer applied more compared to standard dose. So we can say that it is safe for human health. Thank you.
Okay, thank you, Dr. Nitesh. Now I request all the scientists for interaction through chat box. Those who is interested to discuss or anybody have any query. Okay, Dr. Sushil wants to ask. Dr. Rajesh, please unmute Dr. Sushil so that he can interact with the oral interaction. Dr. Rajesh,
Hello?
please. Yeah. Sushil,
Hello.
please.
Sushil here. I congratulate Dr. Victoria for a nice presentation. I have 2-3 queries regarding his research, his work. First of all, in his recovery slide, can I have your recovery slide?
Just a minute. Yes.
In your recovery study, at I think at this fortification level in case of fruits.
Yes.
At the level 0.5, I think is there another one? Tomato fruits. Here you can see at this level in case of propinep, even in case of trifloxystrobin, the recovery of these two compounds is reduced when the fortification level is increases.
Yes.
What might be the reason? And another thing regarding this similar question from the earlier speaker, you also have taken the similar fortification level, and you have tell me that the procedure is for multi-residue method. But
Yes,
as far as my concern and as far as my knowledge, these two compounds, the trifloxystrobin and propinep, are two different methods and two different instruments are required for the analysis. In
Yes.
case of propinep, it has been analyzed on a colorimetric based method, and so the logic of taking this similar level is not, and it is not up to digestible me, because at least for when you analyzing the trifloxystrobin at the level of 0.05, that means 50 ppb, it is too high even for LCMS, because you have used LCMS. What is your take on this point?
Generally what we do, sir, we never standardize method for particular single crop as well as single pesticide. So some some compound shows the very low response in particular, whether it is LCMS highly sophisticated, but continuous response we never get. So initially we get the particular compound as well as particular in a regular response. So based on that, using multi-residue as well as multi-commodity, we fix that particular 0.05 LOQ. And as far as the guideline, you should have a recovery at X, 5X as well as 10X. So we, based on LOQ, we fix the 0.05, 0.25 and 0.5 as a recovery level.
So
and reason behind the increasing the concentration and decreasing the recovery may be the saturation of particular compound in the
that's the point I want to raise here, because for particular in case of trifloxystrobin, in your method validation
yes, it is
study,
decreased. Yes,
you have not incorporated the linearity study.
Yes,
so that might be a reason. And another, in case of your risk assessment study,
yes,
average dietary intake you have calculated on which day's sample? Which days the concentration which you have considered for the analysis for calculating the ADA? First day.
That is 0 day.
0 day.
Yes.
Okay, so you have to mention in your gist or in your presentation
yes.
what the calculation of ADI the 0-day sample were considered for the calculation of?
Actually, ADI, that is acceptable daily intake, is given by the codex as well as EU. So here we consider a reference of codex, that is 0.04 for trifloxystrobin as well as 0.07 for propionate. So ADI we never calculate. It is given by the codex at a time of registration or fixing of MRI.
And one more thing, when you're working out the ADI, you are considering the toxicity of the compound.
Yes.
Right?
Yes.
So my point is that when you are taking compound which are having two different molecules in the formulation.
Yes.
So is there any guideline or is there any references available which shows that you have to consider the compound separately or accumulatively?
No,
When you
actually,
accumulate,
there is no any guideline. Generally, technically, individual compounds should be calculated because in combi product the concentration will be changed, and your active ingredient dose is also changed. So if you are calculating the hazard index or any TMRC value, you have to calculate individually. Then you take the sum of the each pesticide.
okay,
Sir,
thank you.
so Dr. Sushil, I just want to say
Yes.
something because nowadays in a recent, the ADI is calculated on the sole compound. Supposing we are having a compound which is a mixer, that is for example, trifloxystrobin and tabuconazole. Okay. So ADI value is calculated with the help of only sole compound with the recommended dose.
Yes.
They have calculated the acceptable daily intake data with the only this trifloxystrobin, and again they have carried out this another experiment on tabuconazole. So like that, they have calculated the ADI value. So it's a totally different thing.
Dr. Parmar, you have not got my point. What I am asking that suppose in case one compound has toxicity level which is beyond acceptable limit, that is more than 1, and your one compound is having less than 1. In that scenario, what will be our mode of action or what is our modus operandi? In that case, suppose your propionate has high toxic. In that case, what?
Yes.
That's why I have asked this question, because both are the toxic and both are you are applying at the same time.
Sir, here in this slide you can see that I have calculated the hazard quotient for trifloxystrobin, and this one is propionate. This hazard index shows the application of both the compound trifloxystrobin as well as propionate. So this is the value of both the compounds. So in this value, both the toxicity level we can calculate.
Okay, thank you.
Okay, I request both Dr. Nitesh and Dr. Sushil, due to time constraint, please rapidly.
Okay, thank you. I'm convinced.
Okay,
Thank you,
thank
sir.
you.
Thank you.
Now I give the third chance again to Dr. Vishwanatha. He is with us. Okay, Dr. Vishwanatha, please share your screen and present your research paper.
Just 1 minute, sir. We have a presentation.
Okay, allowed. Dr. Rajesh, can you help him from your side?
Just 1 second.
Okay, no problem.
Okay, sir. Now,
Okay, you can proceed, please.
Sir, good afternoon, sir. Sir, can you hear me, sir?
Yeah, audible.
Sir, can you hear me, sir? Sir, is it audible to you, sir?
Yeah, visible
Hello?
and you are
Sir.
audible. You can proceed rapidly, please. Starting with
Okay,
your
sir.
objective and
Okay, sir.
come to result.
Okay, sir. Good afternoon, sir. Myself, P. Viswanath Raghuteja, and my presentation is about residue analysis in okra fruits from IPM and non-IPM plots. First of all, coming to introduction, the scientific name is Abelmoscus isclantus, belongs to family Malvaceae, chromosome number 130. And as we know, the farmers are depending only upon pesticide sprays and its repeated applications with the improper waiting periods. No, it leads to the toxicity of the horticultural commodities. So hence various IPM modules have been worked out with reference to this consumer safety. The main objective is determination of pesticide residue levels in okra fruits of IPM and non-IPM plots. Materials and methods. The IPM plot, the crop grown is okra variety Akanamika with a size of 25 by 20 meters at Rabi, during Rabi 2018 at College of Horticulture, Venkatraman. Next slide, sir. Next slide, please, sir. The IPM plot consists of different practices such as summer gloving, sowing maize as a border crop, mulching, and Akanamika variety was sown with marigold seedling transplantation, yellow sticky traps, light traps, pheromone traps, and direction of bird perch. These are all the mechanical practices which were done. Next slide, sir. Next slide, please, sir. Next slide, sir. Hello, sir. Is it audible, sir? These are the components of IPM plot of okra, sir. Reflective plastic mulching, light trap, marigold seedlings transplantation. In this IPM plot, only botanicals and bioagents were sprayed, sir. Only in chemical is only imidacloprid, sir. At 15 days interval, the NSKE, neem oil, sweet flag extract, imidacloprid, Beauveria bassiana, Bacillus thuringiensis were sprayed at 15 days interval, sir, in IPM plot. Next slide, sir. This is the IPM plot of okra, sir. Coming to non-IPM plot, the non-IPM plot also okra crop was grown, variety named Akanamika and same. Only the difference is the chemicals were sprayed at non-IPM plot, sir. Imidacloprid, lambda cyhalothrin, thiamethoxam, flubendiamide, buprofezin, chlorantranilprol. These all the chemicals were sprayed at 50 days interval, sir, in the 90 days period. This is the non-IPM plot of okra, sir. Coming to methodology involved in residue analysis, 1 kg okra fruits were collected from IPM and non-IPM plots, sir, and they were harvested within 24 days. The steps involved like chopping, they were harvested within 24 days of spraying, and at -4 degrees centigrade they were stored, sir. And analysis was undertaken at PRF-QAL, Pesticide Residue and Food Quality Analysis Laboratory at US Raichur, sir. The residue analysis was determined for imidacloprid from IPM plot and all the chemicals from non-IPM plots. Sir, these are the steps involved: chopping of fruits, blending in grinder, and taking 10 grams of blended mixture. Next slide, sir. Addition of 10 ml ethyl acetate and weighing 10 grams anhydrous sodium sulfate, and it should be added, sir, and homogenization done at 10,000 to 13,000 rpm for 3 minutes, sir. High volume centrifugation for 5,000 revolution per minute for 5 minutes, transferring 7 ml of extract to 15 ml centrifuge tube, having 25 mg of potassium sodium acetate and 150 milligrams magnesium
Okay,
sulfate.
it's standard method. Please come on. result. It's standard
Okay, sir.
method. That's at
Filtration.
least
These
come
are
on
the working
result.
conditions, sir. This is the
Yeah.
LCMS method, sir. Liquid chromatography mass spectrometry. Coming to the residues, the results. The residues were detected from non-IPM plots, and the residues were not detected in IPM plot. In non-IPM plot of okra, these residues detected were above the MRL level, sir. Imidacloprid, it was 6.7, greater than its maximum residue limit. Same for thiamethoxam, flubendiamide, and chlorantraniliprole, sir, which was more than that of its maximum residue limit, sir. Hence, it will be toxic for people if they consume these kind of commodities because there will be toxic chemical and it will lead to the human digestive system and other systems, sir. The LOD value was 2, sir. LOQ was 10, sir. These are the graph representing the residue levels of various insecticides in non-IPM plot of okra, sir. Imidacloprid 6.7, thiamethoxam 3.8, flubendiamide 7.9, chlorantraniliprole 6.52, sir. Next slide, please, sir. These are the chromatogram pictures, sir. Imidacloprid of non-IPM plot. Next slide, sir. Thiamethoxam chromatogram, sir. Flubendiamide, sir. This is chlorantraniliprole, sir. Chlorantraniliprole. Coming to summary and conclusion, the studies were carried out to determine residue levels of imidacloprid, thiamethoxam, flubendiamide, and chlorantraniliprole from IPM and non-IPM plots, sir. And LOD was 2, sir, and LOQ was that is limit of quantification was 10, sir, 10 parts per billion. The residues of imidacloprid was 6.7, thiamethoxam 3.8, flubendiamide 7.9, chlorantraniliprole 6.5 ppm, which were above their MRL values. Retention times were 1.964, 3.389, 12.36, and 11.59 minutes, sir, for all the chemicals, sir. Next slide, please, sir. Next slide, sir. Finally, this pesticide residue analysis was carried out to determine amount of residues in IPM and non-IPM plots. The results revealed that no residues were found in okra fruits grown and harvested in IPM plot. The pesticides from non-IPM plot were above maximum residue levels through high-end LCMS machines. So, hence okra grown in IPM plant having less pest damage with high marketable yield without any residues. Hence, we can suggest IPM as essential safe consumption of okra without leaving any residues and for maintenance of this ecological balance. Thank you, sir.
Okay, thank you. Now topic is open for discussion. Please, Dr. Sushil, again, he has some observation. Please unmute Dr. Sushil Singh. Okay, proceed, Dr. Singh.
Mr. Raghu Teja, can I have your table? Can I have your table number 1?
Sir, actually the slide is with.
Okay, I request Chairman sir to please.
We are sharing here slide.
Dr. Rajesh, please share. Table 1.
Okay, table 1.
Table 1, yeah.
From result, table 1 result, table 1.
Yeah, yes,
result.
it is here.
Here, please. Okay. Here you can see that the spike level is 10 gram per kg.
Gram per kg, sir. Yes, sir.
How you have taken that much amount for spiking? Is this spiking or spray?
Spiking, sir. Spiking after the blending of this okra fruit, sir. 10 gram.
You know what is the level of spiking? 10 gram per kg.
Sir, sorry, sir.
This is 10,000 10,000 ppm level, you know.
Yes, sir.
It is too much.
This is too much. This will totally
Very high.
Very high. I think there is some problem in your study. And another thing is, here you can say that the recovery is okay, the retention time is okay. LOD is 10 ppb. Generally, the LOQ is 3 times higher than the LOD. So it
Yes,
must
sir.
be something around 6 ppb. But how you have got this similar result for all these compounds?
LOD and LOQ were same, sir. Based on the results we got
That's
from analysis, sir.
Mr. Raghu, you see here, the simple thumb rule is that LOQ is 3 times of the LOD.
Yes, sir.
So that means it should be around, LOQ should be around 6 ppb.
Okay, sir.
This is the very basic fundamental rule.
Okay, sir.
And another thing is, you have reported that 6.7 ppm reported in case of a non-IPM,
Yes, sir.
thiamethoxam. In case of
Yes.
IPM, I think somewhere here colantripral. So was your method is multi residue or a single residue method?
Single residue, sir, for all for 5 samples, sir.
So why you have chosen this single residue method? Why don't go for this multi residue method?
Sir, actually, for single residues, I have gone for LCMS method, sir. Liquid chromatography, liquid chromatography, mass spectrophotometry. For multi residues means we have to go for GCMS, it seems, sir. Gas chromatography, mass spectrophotometry, which can be analyzed, which can be used for analysis of up to 74 chemicals.
Mr. Ravi, you know, sorry, Raghu, do you know all these compounds are not analyzed on GCMS?
Yes, sir.
It is only, these are not amenable to GC. These are the amenable to LCMS only. They can analyze either HPLC or LCMS. Another thing is your residues detected on on what day? Just after the spray, because you have not mentioned in your study.
No, sir. Within 24 hours, I have harvested, sir. After spraying, within 24 hours, these fruits were harvested and stored at -4 degrees centigrade, sir.
So that's why you are telling that your values are much higher than MRL value. But you
Yes,
know,
sir.
you have to wait for the waiting period if they are the standard pesticides and recommended in this crop. Then their study will be more fruitful and more realistic. Because
yes,
you have
sir.
to
But,
follow the waiting period, then go for the fruits of the okra is higher than what is the residue of these compounds is higher than the MRL value.
Yes, sir.
Right?
Yes, sir.
Okay, thank you.
But only thing is,
Dr. Kaushik wants to ask. Mr. Vishwanathan, Dr. Kaushik wants to ask something.
Okay,
Dr.
sir.
Kaushik,
Okay,
please
sir.
ask.
Doctor, what was the dosage of all pesticides? All at recommended dose or double of the recommended dose?
No, sir. Recommended dose only, sir. It is recommended dose only, sir.
And after 24 hours, you got that much of pesticides residue?
Not 24, before 24 hours, sir. Yes, sir.
Before?
24 hours, sir.
Means after application, you have collected the samples after 24 hours?
Within 24 hours, sir, after application.
Within 24 hours, even though the concentration which you have shown, it's very high. Say,
Yes,
for
sir.
example, in the case of chlorantraniliprole, 7.9 ppm, it's too much high. And supposing you are analyzing on LCMS, it got saturated.
LCMS, sir.
So that might be some mistake, I think. What is the opinion of Dr. Susil?
Dr. Kaushik, you asked the scientist that is it his work or he is presenting on someone else's presence?
No, sir, this is my work only.
So I think you have analyzed or you just given to sample to the laboratory and they provide you the result.
Sorry for the interruption, sir. But
I have given the sample to the laboratory, sir.
Okay.
Hello, considering the time,
Okay,
please.
no problem.
But also had nice observation. So, being a PhD scholar, Vishwanatha, please consider the points which are suggested by both learned scientists, Dr. Sushil and Dr. Kaushik. They are the learned person in the field of pesticide residue science. Okay.
Okay, sir.
So when you want to ask something, you can also contact them personally too. Considering the time constraint, time limit, I am not allowing much more discussion. Now I request NN Chaudhary. NN Chaudhary, please. Dr. Rajesh.
Yes, NN Chaudhary.
And if NN Chaudhary is not there, then I request Nirmal Chauhan.
Yes, sir. NM Chaudhary Nidhi Ben. Yeah. Nidhi Ben from sir. I share unit 9 number pesticide residue. I think she is sir present in the session but not responding. So we will go for the next presentator, sir.
Next presentation then Solanki. Because two presentation by NN Chaudhary. PP Solanki, please. Dr. Rajesh. Okay. Okay, proceed. Hello.
Yes, Preeti, you can start.
You are not audible. Rajesh, please unmute.
Sir, I have sent a request to her to unmute, but she is not unmuting her.
Oh, she is in another room. So, okay, Dr. Kaushik went there. Wait for few seconds only.
Sir, please take care of the time.
Yeah, definitely. Milan Joshi. She is not.
No, she is on Milan Joshi.
Yeah, he's shared the
on
screen.
Milan Joshi's. Okay, yeah, she is on Milan Joshi. Milan Kumar and Joshi.
Okay.
Kindly unmute the Milan Kumar and Joshi.
Yeah. Okay.
She is on desktop of the Milan Joshi. Okay.
Please start, madam.
Good afternoon, everybody.
Okay, directly come to the objective, then results.
Sir, presenting the residue study, residue and persistence of chlorpyrifos 20% EC in Bengal gram under open field. This is the field experiment and treatment details. The crop was Bengal gram and variety was a GG2, and it's taken in It was taken in February 2017 in Department of Nematology, BSCA College, Anand Agriculture, Anand. And this treatment details are it's a standard dose at chlorpyrifos given 500 gram active ingredient per hectare and a double dose of 1000 gram active ingredient per hectare. And this is the plot. It was the plot size. There was 8 into 4 meter squares, and spacing is a 30 into 10 centimeter. It's a field experimental site, and we are using the for sample analysis standard method. It's a Quacher method. It is the recovery study. Before the starting the experiment, we are doing the recovery study. Different matrices, green pots, seeds, and soils, and that recovery levels are X, 5X, and 10X, like 0.05, 0.25, and 0.5 ppm. This is the residues and dissipation studies. Resulted in initial deposit of results. The result in the initial deposit of a 6.6 ppm and a double dose at 16.97 ppm in green pots at a standard and double dose respectively. The residue of chlorpyrifos immediately declared at first day at 1.89 ppm and 6.19 ppm respectively, standard dose and double dose. The residue of chlorpyrifos immediately declined and gradually after 10th day and were BDL on 15th day at either dose. The dissipation pattern of chlorpyrifos and gram pots followed biphasic mode for both doses. Like first phase is a 0 to 3 days and the second phase is 3 to 10 days. In the week, the half-life values for standard dose were 1.2 days for phase 1 and 1.3 days for phase 2. While in the double dose, the half-life value were 1.1 days for phase 1 and 1.45 days for phase 2. The seed and soil sample collected at harvest, but there is no relevant residue at either dose. The conclusion is an analytical method for using Quacher along with ICMSMS was an accurate and precise as per validation criteria Center guideline. As on no MRL is available for chlorpyrifos, LOQ of 0.05 milligram per kg is taken for default MRL for chlorpyrifos. On the basis of this value, post-harvest interval of 10 days can be suggested for chlorpyrifos. Thank you.
Okay, topic is open for discussion. Rapidly, only 2 questions. No more discussion considering the time limit. Okay, thank you. Now, next is Bhavika Rabari. Rajesh bhai.
Yes, sir.
Milan has already said, okay. Bhavika Rabari, next.
Yes, sir.
I request Bhavika, please directly come to the objective and result and discussion part.
Okay, sir. My research topic is on persistence of mancozeb and hexaconazole in green chili under open field. This is the field experiment and treatment details. The field experiment carried out at Main Vegetable Research Station, Anand Agriculture University, Anand. and the variety used is GC101. 3 treatments were given at the lower dose and higher dose and untreated samples. The field experiment, this is the picture view of field experiment site. This is the flow diagram in which the extraction method was carried out. We are going for the quick, easy, effective, rugged and safe method, Quacher method. And then after completion of the extraction, the samples were going in the LCMS for the injection, and then results were calculated in the Excel sheet. This is the recovery data in the green chili samples at different fortification level 0.05, 0.25, 0.5 in green chili, red chili as well as in soil, and it comes in the acceptable range. The same for the mango gel. This one is the total residue estimated days after emergence. In case 2 further applications where of both the combined products, mancozeb and hexaconazole at lower dose and higher dose was given, in which initial stage the mancozeb residue was 1.6 ppm and 3.10 ppm in higher dose. Then after days after increasing, the residue value of mancozeb is decreased in biophysics mode, and after 50 days, the residue values of mancozeb is 0.17 and 0.21. In the case of hexaconazole, initially the residue value is 0.44 and 0.82. Then days after, it is increase or decrease due to the systemic nature of the pesticide. In the red chili, residue estimated at harvest sample is 0.9 and 3.2 in mancozeb and hexaconazole. Due to the residue estimated in red chili is increased due to the loss of moisture content in the red chili sample, and during we are find out soil sample, there is no residue found in a mancozeb or hexaconazole. It is detection limit, and then after we are calculating the DT50 value, which is half-life value, and half-life value of mancozeb as well as hexaconazole. We are calculated in mancozeb were calculated in biphasic, and hexaconazole is calculated in monophasic mode. Conclusion, after completion of the research, we concluded that the hexaconazole as well as mancozeb, both the pesticides are persistent, and due to the systemic nature of the fungicide, and while further the residue in the chili sample were increased to the increasing moisture content, and residue are in soil samples were collected either dose at harvest, and we didn't reveal any residue of mancozeb or hexaconazole above the limit of determination. Thank you, sir.
Okay, now topic is open. Only one query or one questions. Please be ready. Another Mr. Ravi Kalsaria, if he is
from participant side. The next presentation, please.
Ravi Kalsaria, please.
Yes, sir. Yes, sir.
Okay, rapidly, please. Just share your screen. Dr. Ravi.
आपला विजय जानो है विजय जानो પેલે આપી દે ને કોનો રહી ગયો છે પછી પૂછજે એને મને લાગે રહેના જયનું રહી ગયો છે એકલો નથી આવ્યો.
He is on the same, I think he is on Milan's. Okay, Dr. Ravi is on another computer, please.
Yes, sir.
Dr. Ravi.
Yes, sir. Hello.
Oh, okay. Yes, share your screen, please.
Yes, yes. Okay, sir. Good afternoon, all. Okay. Myself Ravi Kalsaria. Oh, my voice is audible.
Yeah, exactly. Hello? Please proceed.
My voice is audible.
Yeah, audible and visible too. Your presentation is visible and you are audible. Please proceed.
Okay, okay, sir. Okay, thank you, sir. Okay, good afternoon all. My research experiment title is Residue and Persistence of Imidacloprid in Green Pea and Soil After Application of Imidacloprid 48 FS in Green Pea as Seed Treatment. First of all, introduction. As you know, the green pea is commonly grown leguminous vegetable in world over. Seed treatment of imidacloprid is commonly.
Dr. Ravi, come to objective and results directly, considering the time constraint.
Okay, sir. Okay. My research objective is the decimation pattern of insecticide in one green pea, followed by gap. To establish the maximum residual limit on green pea, the safe waiting period and pre-RFC interval to calculate the half-life of this pesticide, to generate the data of establishing the MRL for imidacloprid in green pea by Food Safety Authority, and residue and persistence of imidacloprid in green pea and soil after application of imidacloprid in green pea as a seed treatment. This is a field experiment detail. The experiment was conducted Department of Agronomy, BSc, College of Agriculture, Anand Agriculture, Anand formulation imidacloprid 48 FS. These are the treatment detail. T1 is control, T2 is a lower dose, and T3 is double of the recommended dose. This is a experiment detail, plot size. Experiment is an RBD3 application, date of sowing, date of application, date of drenching, first picking of green pea 29 January 2019, sampling interval. From each treatment load, green pea, 2 kg green pea were collected randomly at 0, 1, 3, 5, 7 and 10 days after the final application of the insecticide of imidacloprid, and sampling of harvest, mature pods, seed fodder and soil to 97 days at harvest. This is the experiment site. The conducted Department of Agronomy, BA College of Agriculture, Anand. The certified reference material were supplied by Biocrop Science India Limited, Mumbai, and all chemical used in residue analysis were LCMS grade. The analysis was done by the Symposium LCMS 8050. This is the instrument and apparatus, high volume homogenizer and refrigerator centrifuge. These are the apparatus: balance, bath, vortex mixer, auto sampler, glassware, auto pipette. We are using the quencher method, quick, easy, cheap, effective, rugged and safe. Quencher extraction and cleanup technique has become a more popular and routinely used for the pesticide residue analysis or food matrices. This technique is more efficient. It requires the less solvent, low sample volume, less time, pesticide better analytical program. These are the various steps of extraction and cleanup. This is the instrumental parameter. We are using the 14C18 column, and determination of imidacloprid was carried out by multiple reaction. Non-iodic MRM and ESI probe was operated in the positive mode. This is the accuracy precision data were obtained through the recovery of spike sample. The recovery was conducted in 3 applications by fortifying the sample with the insecticide at 0.01, 0.05 and 0.1 milligram per kg. The recovery of imidacloprid from 80 to 110% based on the matrix math calibration standard were within the acceptable range of 17 to 120%. This is a decimation table. In order to claim the level expansion of the green pea, a field experiment was conducted. The Department of Agronomy, a BA College of Agriculture, Anand, during the Rabi 2018-19. The initial residue of imidacloprid was found below the limit of quantification 0.01 milligram per kg in green pea, immature pod with succulent seed and succulent seed at either dose, and green pea mature pod with seed, mature seed, dry fodder means without pod and root, and soil sample collected from either dose at harvest 97 days did not reveal the residue of imidacloprid above the limit of determination. Conclusion: the residue of imidacloprid was found below the limit of quantification 0.1 in green pea, immature pod with succulent seed and succulent seed at either dose, and the harvest, the mature pod with seed, mature seed, dry fodder without root and pod, and soil sample collected from either dose at harvest 97 did not reveal the residue of imidacloprid. So the residue of imidacloprid detected in green pea was below the Codex and EU limit of 5 milligram per kg. So we have suggested the insecticide can be recommended by the green pea with 0-day PHI. Thank you.
Okay, thank you, Ravi. Topic is open for discussion. Instead of Nidhi, anybody wants to present? Otherwise, this Nirmal Chauhan. Is he joined with his name? Nirmal Chauhan. Rajesh bhai.
Yes,
Instead
sir.
Of,
yes.
Yeah.
Yes, sir.
Okay. Nirmal, rapidly, I request you for your presentation of both the paper, directly come to the objective and research result. Okay, because the methods followed by all the scientists are quacher method. If is there any change in the methods, then you narrate it. Okay, please
Okay,
share
sir.
your screen and proceed.
Oh my God. So, have you seen?
No, not visible.
Okay, 1 second, sir.
Share your screen.
Okay. So, is it visible?
Yes.
Yes, sir.
Okay. Good afternoon, sir. Respected Chairman Dr. Getia sir and Dr. Kiri Parmar. I am going to present my field experiment on pesticide residue, which is sponsored by Biocrop Science Limited. I am direct. Earlier, so many scientists has narrated the method and so on. So I'm directly going to resolve in this my PPT. As you know, this is my recovery. It is all in Sante guideline between it should be 17 to 120, but we have got the nearer to 100%. So my accuracy is more than more. I got the recovery. My accuracy is more, sir. So another same. So looking to the result, Mancozeb. I got the result. Yeah, in standard dose it BDL after the 10 days, and double dose I got the BDL after 10 days. So and there is I also noted out the half life. In phase 1, the residue is immediately the decimation phenomena is follow there. 0 to 7 days is a first phase, and second the phase 2 for phase 2 was 7 to 10 days. That is 1.63 CS2 per gram, sir. So I am going to conclude the analytical method culture along with the LCMS was accurate and precise as per validation by EU scientific guidelines, and is no MRL fixation by this. The LOQ is a default. We have say that 0.05 milligram per kg for mancozeb on this crop, and on the basis of this value, PHI of 15 days can be suggested for mancozeb. Thank you for your kind, sir.
Okay, Mr. Chauhan, have you another presentation related to the acid persistence of acid in tomato? Is it with you?
Yes, sir. Yes, sir.
So please share it and present it.
Yes, sir. Yes, sir.
Sir, yes, definition of combination of fungicides, not available.
Please proceed rapidly.
Sir, is it visible, sir?
Mr. Chauhan.
Is it visible, sir?
Visible and audible continue,
Yes,
sir.
yes,
Okay,
visible.
sir.
Please.
So the another trial was taken by me, residual persistence of the acid 75% SP on tomato under open field condition. There is the same repetition. So I'm now going to rapidly. I'm going to rapidly on tomato. This is our set procedure for the taking the experiment field at our field condition. This is field photo, sir, and or chickpea, and this is method. It is, we call it Kretcher method. It is a multi-residue method, sir, and we have all the sophisticated instrument and equipment we have. So we don't want to take any risks regarding the result. We have take the all instrument very all calibrator and all is a standard instrument we are using, sir, for pesticide residue analysis. This is recovery data. We got the same. We got the recovery in the between 70 to 120, and we got nearer to 100%. So our accuracy also is good in the both aciped and methanophos, sir. Looking to directly, I'm going to result the in this. If you are going to acipate the BDL, we got the means below 0.5 milligram per kg microgram per gram towards the 10 days after spray. In methanophos, it is a metabolite acephate. In 0 days we got the BDL, but is the higher dose we got the BDL 1 days, and later on this goes another 7 days. So in combination, BDL got the after 10 days, sir. Looking to the result, we say that the 0.05 milligram per kg is taken a default MRL for acephate, and on the basis of this value, 15 days can be suggested for acephate as a pre-harvest interval. So farmer can follow this pre-harvest interval 15 days after they are going to spray for acephate in tomato. Thank you, sir.
Okay, thank you. Now topic is open for discussion rapidly. Okay, now next session is related with the poster presentation, poster session.
Hiren Patel, sir. Hiren.
Yeah, I think is he with us?
I think.
Okay. HV Patel. Okay, please. But this is the poster. Okay. Unmute Hiren Patel. Dr. Rajesh, please unmute to
Yes,
Hiren.
sir. Yes.
Okay. Hiren, you simply just narrate your work, okay, with your objective, and directly come to the result.
Okay, sir.
Your objective here, please, rapidly. Mr. Hiren.
Yes, sir. Okay, good afternoon all. Respected chairman, co-chairman, and dear participant. My abstract title is impact of the different household processing on the reduction of the insecticides residue in or on brinjal fruits. Now, objective is to study the impact of the different household processing on the insecticide residue in or on brinjal fruit. Generally, variety we can use Surti Ravaya, and the research was carried out at the Navsari Agriculture University, Navsari, and the residue analysis was carried out at the food testing laboratory at NAU Navsari. The standard of methodology was followed for the.
Come to the result, please. Dr. Hiren, please directly come to the results.
Result among the different treatment washing the fruit with the tap water as well as the cooking for 10 minutes was found to be the best process in which regard the level of the insecticide residue was dislodged from the brinjal fruit. Means the washing the brinjal fruit with 10 minutes and then after the cooking for 10 minutes, all the 6 insecticide residue are found below the determination level. And the conclusion is washing of the fruit with the tap water as well as the cooking for 10 minutes were found to be the best process in which the greater level of the insecticide residue was dislodged or removed from the brinjal fruit. Thus, the process resulted as a below determination level of all the insecticides residue in brinjal fruits. Okay, and my second topic is
okay, proceed for second two. Yes.
Okay.
You are allowed to proceed for the second poster, okay. Please.
Now, second abstract topic is dissipation of the chlorpyrifos and imidacloprid residue in or on brinjal fruits. The objective to find out the chlorpyrifos and the imidacloprid residue status in brinjal fruit at 1, 4, 7, and 10 day after the application of the insecticides. The standard methodology are adopted for the residue analysis, and the result is delivered at the chlorpyrifos at the rate 0.04% residue at 0 days, means the 24 hours after the application of the insecticide, 0.89 microgram per gram residue was found, and they are dissipated 0.65, 0.40, 0.10, and 0.04 microgram per gram after first, 4th, 7th, and 10th day after the application of the insecticides. Where in case of the imidacloprid, 0.72 microgram per gram residue was found at initial level, and 38.89, 76.839% residue dissipation was found after the first and 4th day of the application, and their residue was found below determination level at 7 days after the application of the imidacloprid. And the conclusion is that the initial deposit of the chlorpyrifos in or on brinjal fruit was reduced to 0.65 microgram per gram, 0.40, 0.10, and 0.04 microgram per gram at first, 4th, 7th, and 10th day after the application, respectively. Imidacloprid residue was progressively decreased from first day onward, and the maximum 79.66% reduction was observed on the 4th day of the application of insecticide. The residue of imidacloprid reached to below determination level at the 7th day after the application. Okay, thank you, sir.
Okay, Hiren, I want to ask the one question related with the first poster presentation.
Yes,
you had utilized the tap water for washing.
Yes, sir.
And this quality of the tap water is vary from region to region. Okay.
Okay, sir. Yeah.
So can you able to tell me that if the tap water with least salt content is there, what will be the impact of washing of fruits with the tap water with least salt content and with high salt content?
If the
can you able to answer it?
If the water with the high salt water, the outer skin residue is easily removed due to because of the residue are dissolved in the NaCl solution, salt solution easily, and the least dissolved salt.
So from your result, it is from Navsari, okay, and Navsari has the higher salt content.
Sorry, sir.
So can we recommend this in all the region other than the Navsari region, that washing of the fruits will give the same results in the middle Gujarat or north Gujarat?
No, sir. Due to the quality and presence of the salt containing water in region by region.
Thank you very much.
Okay, sir.
Now next one, I request Israt Jahan. Israt. Israt, please. Dr. Rajesh.
Yes, sir. She is, I think, sir, not present. She has written one message. Just
She is nice PPT, Dr. Ravi. She
yeah,
is replying.
she has written, sir, but right now she is not present.
Oh, unable to present it.
Okay, next.
Then Milan Joshi.
So he is with you? Yeah.
Yeah, yes. Yes, Joshi, please. Directly with objectives and results. Please present your poster rapidly. Hello, Milan, can you listen me? Milan?
Sir. You are audible. Please continue.
Yes, directly.
Sir, my screen is pushed.
Hello.
Hello. So are you able to present?
Hello, we are listening you.
Sir.
You are audible.
Okay, sir, myself.
You are audible.
I am presenting.
No problem, sir. Please continue.
Okay, sir. Okay, done. Sir, myself, Ravi Kansaria. Today I present the poster presentation of dissipation of ethion, 50% EC in green chili. Our objective is to establish the maximum residue limit on green chili, a safe waiting period and pre-harvest interval to establish and find out the acceptable daily intake capacity and the risk assessment. So, by methodology, the investigation was conducted at the ANP on pesticide residue ICR Unit 9, Orange Agriculture University. The experiment was laid out the 3 replication, 2 foliar replication of ethion, starting from the fruit initiation stage at 10-day interval at the rate 500 and 1000 gram AI per hectare respectively. The sample of green chili fruit were drawn the 0, 1, 3, 5, 7, 10 and 20 day after second spray. We are using the Quacher method and all the reagent and solvent used in the analysis were either PR grade. So my result, you can see the persistence of ethion study in the chili fruit after 2 foliar application at 10-day interval. So result in the 6.47 and 9.44 microgram per gram deposit in standard and double dose respectively 1 hour after the application. In standard dose, the results were erratic. However, by obtaining the 7-day data, the dissipation followed the biphasic mode with half-life of 1.8 day for the phase 1, 0 to 3 days, and 9.1 day for the phase 2, 3 to 15 days. In double dose, the dissipation followed the first order kinetic with half-life of 9.7 day for omitting the 5th day. The residue were below determination level on 20 day for either dose of application. So, there is no MRL available for ethion. The LOQ of 0.05 milligram per kg is taken as the default MRL for ethion. On the basis of this value of PHI can be suggested by 6 days for the ethion. Thank you, sir.
Okay, Ravi, you have another poster too.
Sir, you have another poster.
Sir, Dr. Sushil want to ask on this. Dr. Sushil, please unmute Dr. Sushil. Rajesh bhai.
Yes, sir.
And Ravi, be ready for your second poster presentation.
Ravi, can you hear me? In your data, you find that up to 15 days, 0.84 milligram per kg is available. Please go through the first slide, the first presentation.
Yes, 6 per.
For your earlier presentation, this is your, okay, I will ask the question. In that into 20 days, the 0.84 at standard dose and 2.32 was at the double dose. But in the next 5 days, almost the the residues dissipated very fast and it becomes less than VQL, that is 0.05 ppm. What might be the reason? Have you got the question?
Yes, sir.
Have you got the
Can
question?
you get it? समझ में आ गई बात?
Sir, आवाज नहीं आ रहा.
Okay, I will repeat.
Ravi.
In your earlier study, the dissipation up to 15 days of ethionine in chili was, the residues were 0.84 and 2.32 ppm at on up to 15 days. But next 5 days, it is beyond detection limit, that is 0.05 ppm. So what happened in that 5 days? Can you elaborate that? How these are dissipated very fast in that phase only?
Yes, sir.
So Dr. Singh, he has no any idea about the reason.
Okay, no problem.
So if you have any reason, you can please rapidly.
Okay, might be the temperature, because might be the temperature and other environmental factors. So it would be better if you have also include this type of information also.
Okay, thank you. Ravi, your next presentation, please.
Okay, sir.
Dr. Ravi.
Yes, sir. Okay.
Please share.
My voice is audible.
Yeah, audible.
Okay. Second presentation, my poster presentation is the persistence of cypermethrin, the 25 EC in capsicum under the open field. The methodology is same. 1 hour after the last foliar application, the residue were 0.55 and 1.25 microgram per gram in standard and double dose, and residue dissipated gradually and BDL on the 20 day with the half-life of 5.79 day for the standard dose. In case the double dose, the initial degradation was relatively faster during the 3 day, where residue dissipated more than 60% and 0.48 microgram per gram on the 3rd day. Then after, the rate of dissipation observed was relatively slow and residue were below determination level on 20 day. The half-life observed of the phase 1 0 to 3 days was 2.28 day, and for phase 2 3 to 15 day was 6.84 day for the double dose. So no MRI available of the cypermethrin. The LOQ of 0.05 milligram per kg is taken as default MRL for the cypermethrin. On the basis of this value, the PHI of the 6 day can be suggested for the cypermethrin. Thank you, sir.
Okay, open is open for discussion. Rapidly, please. If anyone has any query, please.
No query, sir. Please proceed.
Okay. Okay. Thank you. Now I come to Sapteswari. Sapteswari.
Shivam.
Hello,
Shivam,
sir.
Yeah, continue.
Shivam, please proceed.
Sir, is my screen is visible?
Yeah, exactly. You share your screen in PPT mode, please.
Sir, I have already shared.
Slideshow. Okay, good. Now you proceed rapidly.
Sir, I cannot see the screen. Hello, sir.
Okay, just a minute. Dr. Rajesh.
Yes.
Just pray upper arrow.
Just a minute, sir.
Yeah, no problem.
Sir, either I can do here, I can share my screen from.
Okay, sir. Do from your side, Shivam.
Okay, sir. Okay, sir. Screen is visible to you, sir?
Yes, visible and audible. Please
it is.
continue.
Okay, sir. Good afternoon, everyone. My name is Shivam Sapteshwarya. I recently have passed out my MSc in entomology from Anand Agricultural University. And my experiment topic, which I present today, is residue analysis of effective insecticides in or custard apple. Coming to our introduction, the custard apple, that is Anonas commus, is a native of South America. It's a very hardy tropical food crop. The main
Directly come to the objective, please. Objective and result, please.
Okay.
And slideshow. You are not screening.
Okay, sir.
Okay. Yeah. Continue.
Okay. The objective of my experiment was to study the residue analysis of effective insecticides, that was buprofosine 15% plus acifate 35% and thiamethoxam 12.6% plus lambda cyhalothrin 9.5% ZC, along with the control. Coming to the result, a linear study was performed to determine the performance of LC-MSMS and GC-MSMS, and a graph of detectors response versus concentration was plotted. As per the data obtained in linear study, both the insecticides that are buprofosine plus acifate and thiamethoxam plus lambda cyhalothrin were found linear. The residue obtained were for buprofosine 0.114 ppm, for acifate 0.217 ppm, for thiamethoxam 0.210 ppm, and for lambda cyhalothrin 0.110 ppm. Here, the MRLs of the tested insecticides on custard apple are not available in FSI and a codex. Thus, the limit of quantitation, that is LOQ, of all 4 pesticides were taken as their MRL, that was 0.01 ppm. Coming to conclusion, out of 9 insecticides evaluated for bioefficacy, 2 insecticides that were buprofosine plus acifate and thiamethoxam plus lambda cyhalothrin were analyzed after 15 days of last spray. That was post-harvest period was 15 days. The MR of the tested insecticides are not available at FSI and Codex. Thus, it can be suggested that the present study need to be validated at 4 different locations so as to undertake risk assessment for fixation of MRLs. Okay, sir. Thank you, sir.
Okay, topic is open for discussion. Then next one, I request Nirmal Chauhan. Nirmal Chauhan.
Nirmal Chauhan, please. He is able, sir. He is present. I think.
Please unmute, sir. Me. Hello.
Yeah, you are already unmuted.
Sir, if you are talking means you are unmute.
Okay. Thank you, sir. Good afternoon, sir. Again, respected Dr. Getia, sir, and Dr.
Okay. No need of formality, please. Time is constraint for all of us. So, please rapidly come on your presentation.
Okay.
Please share your screen.
Yes, sir. Are you visible my screen, sir?
No. Oh. Your poster is invisible.
It is almost 1 hour late, sir.
Visible, sir?
No. Yes, visible. Now you proceed.
Okay. My study was a decimation of combination fungicide fluvirum plus typhloxobin in onion. As we know, onion is a great exporter among the vegetable fruit crop, vegetables in India and in Gujarat, it is also is a good exporter vegetable commodity, particularly our Mahua and Bhavnagar farmers are doing so many.
Did not to focus on introductory part. Directly come to the objectives and result, please.
My objective is to study the decimation pattern of both combination fungicide in one onion crop followed by good agriculture practices. Second objective was to establish the maximum acid limit on onion, and third was to establish safe waiting period and pre-harvest interval. Looking to the result, before initiate the trial, we have to conduct the recovery data. So my recovery was in between the 7760 to 106. It is acceptable according to scientific guidelines. My limit of contribution was 0.05 milligram per kg for all fungicides. So in result, after the spraying of the fluparam, the residue was 0.28 and 1.10 milligram per kg is stable, standard and double dose, and corresponding level of trifloxystrobin was 0.27 and 1.10 milligram per kg, and the reaching the time of the due to the desiccation pattern, and it is in the desiccation there is a low of kinetics follow. So initial deposition is corresponding to the how much the deposited concentration is follow the low of kinetics. So here we also find the low of kinetics. So decimation we say that we do not get the pesticide after the 14 days. That is we get, but it was below quantified limit, that is 0.50 per milligram per kg in 40 days in fluparam, and same result found at trifloxystrobin. And we have also taken the sample at the harvest stage. So we taken the spraying of fungicide, we do not get any residue of that fungicide. So we said, and there is also we got the one interesting fact that the metabolite of this both fluparam trifloxystrobin, that is fluparam benzamide and CGA 1,2,3,1,3 were not detected in all the substrate also with the soil. And so looking to the there is no available MRL, so we say that 0.05 milligram per kg can be proposed. And for farmer of the Gujarat, we can say that if you are going to spray this combination of fungicide, you should follow the PRS interval of 14 days suggested. I am going to direct now conclusion. I noted the conclusion very generic form. Looking for the benefit to the farmers, the consumption of the dry onion bulb is safe after the spraying of the combination fungicide containing fluparam and trifloxystrobin. However, consumption of the immature onion bulb and leaves should be avoided up to the 14 days after the spray of the fungicide. Looking at both the compound, so it is very beneficial to delay the resistance in the onion against the disease, because the fluparam has a mode of action on the direct the fungicide respiration system, and the fluparam is inhibit the mitochondrial in the cell system, so nematode cannot be survive. So both has a different mode of action. It is more useful in our IDM that is insect disease management program. Thank you, sir. Thank you.
Okay, thank you. Anybody have any observation? Okay, now I request Mr. Kaushik Parmar, Dr. Kaushik Parmar. This one is the last poster.
Nirmal, leave your screen. Nirmal Chauhan, leave your screen.
Yes, you said it.
Yes, Kaushik sir, you can proceed now. Sir, I have changed the setting. Now you can share.
Okay. So all windows.
It is visible?
Yes, sir.
Okay, so this is regarding the residue and persistence of pyraclostrobin 20 Wg in tomato. The experiment was carried out with the objective to generate the information on persistent and residues of pyraclostrobin in tomato fruits and fixing up the MRL and PHI. So as the pyraclostrobin is widely used in tomato crop for the control of these diseases, so all the experiments just carried out with the same procedure, same method validation procedure and same extraction procedure with previous one. And so directly I come to the this table, residual persistence of pyraclostrobins. As the data shows that initial deposit was 0.39 ppm in recommended dose. While in double of the recommended, it was shows the 0.54. And the residues degraded gradually and it was found below the determination level on 20 days.
Okay, what happened? Is it visible?
No, sir. Sir, maybe there is some technical issue. So
Okay. He can present his work without this showing the poster.
Okay, no problem.
Mr. Kaushik, you can.
I think now it is visible.
Yeah, visible.
Yeah, so the data shows that the half-life of this pyraclostrobin in the recommended dose, it was in a two phase, that is 0 to 5 days and 5 to 20 days. So in the first phase, the half-life values was 4.36 days, while in the phase 2 it was the 15 days. So by using this the data, the conclusion is the 3 foliar spray pyraclostrobin 20 Wg in tomato at the rate 100 gram active ingredient per hectare starting from fruiting stage resulted in residue below the MRL. The MRL value of pyraclostrobin in tomato fruit is 0.3 ppm. So by using this MRL in tomato fruits, if harvested 3 days after the third spray, so therefore PHI of 3 days could be suggested for the pyraclostrobin in tomato. Thank you.
Okay, anybody have any queries? Okay, so altogether, I have the 11 oral presentation we had already presented and 8 poster presentation, altogether 19 research paper on pesticide residue science. We had the good residue status session, and I am highly thankful to my colleague Dr. Parmar and both reporter. So I am highly thankful to all the participants, all the presenter. They had nicely presented their work. So friends, I congratulate all of you. Thank you.
Thank you very much,
Dr. Rajesh, please.
for a marathon presentation in the residue status theme, and this was the certificate which we are going to present you, sir. And as we are almost 1 hour late, and the next speaker almost waiting from the an hour, so sorry for the this inconvenience, madam. Now I request Dr. Yogeshwari Jhala madam to deliver a lecture. Dr. Yogeshwari Jhala madam is an assistant professor in the Department of Agriculture Microbiology, AAU Anand. She has experience of 14 years on the agricultural beneficial microbes, and considering his her experience, she is a pioneer work in isolation and characterization of methylotropic bacteria for mitigation of the methane emission from rice ecosystem, with reporting of 5 unique strains of methanotropic bacteria. And she has been awarded by All India Best Research Award, Young Faculty Award, and established an Indian record for discovery of methylotropic bacteria. She reported one standard strain of methane oxidizing bacteria and deposit at ATCC. She has more than 30 international and national paper published. Almost 9 Springer books she has published. More than 20 book chapters in a reputed publications. Popular article 14. And she is a editorial board member in 8 international and 10 national journal. And she is also she has a grant from the DBT, and these other financing agency, and the source for the running the project. Now I request Dr. Yogeshwari Jhala madam to deliver a present, and kind request to take the care of the time, please, ma'am.
Sir, am I audible?
Yeah, audible and visible both, ma'am. Please continue.
Okay, sir. So quickly, I will start with my presentation. I am very thankful to you all for giving me opportunity to share my views on this platform. So quickly, starting with my topic, role of microbiome in
Ma'am, your voice. There is some technical issue. She is sharing her screen again.
Am I audible, sir?
Yes, ma'am. Please share your screen.
Screen is also shared.
Screen is not visible, ma'am.
Okay, okay, sir. Wait a minute. Is it visible?
Sir? Hello?
Yes, visible, madam.
Okay. So sorry for there is some connectivity issue. So microorganisms are colonizing each and every part of plant, and they are the main drivers of plant health as far as plant health is concerned. So if we want to emphasize on plant health management, then we do not only concentrate on plant disease management, we just have to concentrate on plant nutrient management as well as environment management, which is associated with the plant. So I am going to touch all the three areas, and I am not going to discuss about traditionally known thing, but what we are doing at our research institute, that I will emphasize. So at AAU Anand Agricultural University, Gujarat, we are working as a Department of Agricultural Microbiology with the thrust area of research on agriculturally beneficial microorganism. We are concentrating on biofertilizers, biopesticide, biodegraders, and bioenergy. So firstly, I will discuss about plant nutrient management through biofertilizers. We can say them as plant probiotic, and general terminology is plant growth promoting rhizobacteria. So when this biofertilizer formulations comprising of agriculturally beneficial microorganisms are incorporated into the soil through seed, seedling, or soil application, they will affect the plant in two ways. By direct mechanism, they will enhance nutrient uptake, they will stimulate plant root growth, and they can produce phytohormones or plant growth hormones, which can stimulate plant growth. And in indirect way, they can increase resilience to biotic and abiotic stresses. They can improve soil structure and texture, and they can also change in the carbon allocation pattern. Altogether, this will lead to the improvement in plant growth and yield. So they increase above ground biomass, they improve the yield of crops, as well as increase the resilience or resilience to the constraints like biotic and abiotic stresses. And all the microorganisms which are colonizing plant, all the beneficial microorganisms, they are doing these things. So why we want to pursue research in biofertilizer area? Because if we look at the global CAGR of biofertilizer industry, then it is increasing from 2014. And in 2022, it is predicted to increase by 14.08% due to increased demand of fertilizers, development of biofertilizer manufacturing technologies, and research and marketing is also strengthened in this area. So this is the scope of biofertilizer industry. So at AU, we have developed 7 products of liquid biofertilizer comprising of nitrogen fixers, azotobacter, azospirillum, and rhizobium, phosphate solubilizers, potash solubilizers, and we have also prepared a combi product which are known as BioNP consortium and BioNPK consortium. So BioNP consortium is comprising of nitrogen fixer and phosphate solubilizer, whereas BioNPK consortium is comprising of all the three NP and K availability increasing bacteria. So this task was supported by government of Gujarat since 2000. Since 3 decades, but in 2012, when Narendra Modi ji is our CM, he has promoted this liquid biofertilizer in the state during Krishi Mahotsava, and in Krishi Mahotsava we have sold almost 1 lakh liter of biofertilizer in the Krishi kits to the farmers of Gujarat. And recently in 2020, Modi ji has also said that we have to nurture our soil. We have to prevent the chemical fertilizer input from going into the soil, and we have to secure our mother earth from this harmful chemicals, and this can be done by using the biofertilizers. So we are doing research in this area. We have one patent of BioNPK consortium. This is the joint patent of Anand Agricultural University and Department of Biotechnology, Government of India. And this product is having very good feedback. And in 2014, this patent is been published. These are some unique features of this liquid biofertilizer technology. It ensures shelf life of more than 1 year. having 10 to 50 crores of bacteria in ml concentration. It is very easy to use and transport better efficiency. It can save nitrogen, phosphorus and potassium fertilizer with 8 to 10% yield increase. And it is an eco-friendly and it is highly useful in organic farming as well as drip irrigation system. So we believe that we are technology generators, but to some extent we are selling this biofertilizer for the benefit of farming community. But besides this, we have commercialized this biofertilizer technology to various entrepreneurs. We have commercialized the technology to 7 agri entrepreneurs in Gujarat state, and from this technology commercialization we have generated 11 million rupees from this. And here are the products of that beneficiary companies, and you can see that as compared to our cells, their cells are going upwards because they are dedicatedly working on only biofertilizers. And our one of the beneficiary, which is also associated with Gujarat government, that is GAIC, it has sold 2.6 million liters of liquid biofertilizer after getting this technology from us. And we can say that altogether these beneficiary companies and we have sold around 3 million liters of biofertilizer in last decade. And we can say that if we consider the application dose of 1 liter per hectare, then we have benefited 3 million hectares of land of Gujarat state with this beneficial product. So this is glimpses of our production unit. We have 1 lakh liter production capacity unit at AAU, and we have a automated bottling unit also. It is having capacity of 300 bottles per hour. So if we look at the cost saving or economics of this biofertilizer use, then nitrogen fixing biofertilizers, rhizobium, azotobacter and combi products can save 20 to 35 kg of nitrogen. Of course, these are the data which I have got from FAI website, but this amount may vary depending on the strain's efficiency as well as soil conditions. So 20 to 35 kg nitrogen per hectare is the range. It may vary. And similarly, phosphorus can be mobilized by 12 to 15%. So if we look at the economics, then 1 bag of DAP will cost around 1200 rupees, whereas that work can be done by azotobacter and PSB 1 liter culture. It cost about 120 rupees. We are selling it at 120 rupees. Some of the companies are selling because they are getting some government subsidy, so they are selling it lower at a lower rate. So maybe 100. So 100 or 120 if we consider, then it is 240, and net saving will be 960 rupees per hectare. And this is of course the bigger amount for farmers. We have demonstrated this technology in tribal regions of Gujarat state since last 14 years. We have done thousands of demonstrations in maize and wheat, and farmers are very satisfied with the performance of this liquid biofertilizer. They have reported 25% of saving in NNP fertilizers as well as 10 to 15% yield increase they have got out of this technology. Similarly, right now, the area under drip irrigation is increasing in our state. So farmers are also eagerly using this liquid biofertilizer in drip irrigation, because previously solid biofertilizers are available, powder-based formulations. So there are limitations that they can chalk up the pipes of drips. So this liquid will go with water, and they have no constraint of using this in field. So looking to the success of this biofertilizer technology, Ahmedabad Municipal Corporation has tied their hands with us in their very big project. I can say that they are having the project of hygienizing the dry sewage sludge, which they are collecting from Ahmedabad city. And they are hygienizing that sewage sludge with BARC, that is Bhabha Atomic Research Institute technology, and then they want to fortify that product with biofertilizer to get good quality manure. So recently they have released that product named Bio Gold. So what is the process that I will briefly explain you. So here you can see that this type of sewage treatment plants are available throughout the Ahmedabad and in any municipal corporation. So they are basically collecting wastewater, domestic as well as industrial wastewater, and treating it with number of processes. And the after water treatment, whatever the solid material is there, it is known as sludge. That sludge is transferred to sludge drying bed, and they are totally dried. Then our role comes. This dried sludge is crushed by crusher, and it is filled into the aluminum container, and that aluminum container are passed through radiation unit. It is containing cobalt 60 source, which is emitting gamma irradiation, and that gamma irradiation will hygienize this sludge. It will remove all the pathogens and all the microorganism, I can say, from this sludge, and that sludge is now hygienized. This hygienized sludge is then transferred to the containers like this, and it is moved to the bio-NPK spraying station, and this bio-NPK consortium is sprayed over that hygienized sludge, and it is now nutrient rich organic manure. And we have got 3 years of project of AMC, BRC and AAU, and we have got some very good result in crop production also. We have tested this product in wheat, tomato and potato, 3 crops. Here you can see the upper one is 100% recommended dose of fertilizer, whereas lower one is 80% recommended dose of fertilizer. And you can see that the growth of plants are very good, and yield also we have got very good yield. One major constraint while using this sludge as manure is heavy metal concentration, heavy metal load which is going with sludge in the soil. So we have tested that heavy metal content in the soil as well as edible part of the crops, and we have got that within permissible limit. This is why, because Ahmedabad Municipal Corporation has avoided some sewage treatment plants because they are having high heavy metal content. They have avoided that sludge material, and we have got that within permissible limit. So now that project is running in Ahmedabad, and I can say that that one is the Asia's first largest biogold production plant. And right now we are tying our hands with other Indore Municipal Corporation and other municipal corporation for the same technology. So these are the new things which we have done. Then second, this is all about plant nutrient management, which we have done and what we are doing. Now I want to switch over to the plant disease management. You all know about the microbial pesticides and all that, so I don't want to go in depth of that. This is the basic mechanism of a biocontrol agent. One is antibiotic secretion that will kill the that will kill the pathogen. Another one is lytic enzyme secretion that will kill the pathogenic organism, mycoparasitism or hyperparasitism. Then competition for nutrient and space. Nutrient in the sense this organism produces folate ion, and it reduce availability of the pathogen. Similarly, this microorganism, when they colonize the plant, they will yeast, and somebody is saying something, I think.
Yes.
Sir, can you mute them, please?
Okay, just a minute. Yes, ma'am.
Audible, sir?
Yeah, continue.
Okay. So they will colonize the plant roots, and that plant roots are also not having any space for pathogens to attack, and that's how plant will get protected. Another mechanism which is comparatively less explored is induced systemic resistance or plant immunization with PGPR. Comparatively, it is less explored. Right now, many people are working on this, but still we have to do some research in this area. So basically this inoculation of PGPR to induce ISR is known as priming with PGPR. So in upper figure you can see that this is the normal plant without any priming, and when it will get attacked by pathogen, then there is normal defense response. But when we prime them with PGPR, then they will be sensitized and accumulate the signal molecules, and after challenge inoculation of the pathogen, that accumulation will lead to the enhanced defense response. This is the simplest thing what we are doing. So basically this ISR is enhanced by 4 group of organisms, Pseudomonas fluorescens. Many more are also involved, but these 4 are in hotspot. Pseudomonas fluorescens, Pseudomonas aeruginosa, Bacillus amyloliquefaciens, and Bacillus subtilis. These are the 4 major group of organisms which majority of researchers are working on that. So SAR and ISR, these are the 2 phenomena. SAR, you all are very much aware about, being pathologist, that is systemic acquired resistance. But what I want to emphasize over here is ISR, that is plant rhizobacteria mediated response. It is dependent on jasmonic acid and ethylene hormone, that are the signaling components, and NPR1 gene products are the key component in this pathway. This will lead to the enhanced defensive capacity of the plant upon pathogen inoculation. So what is the basic difference is induced systemic resistance is mainly induced by PGPRs. There is no accumulation of salicylic acid and PR gene product. Of course, some exceptions are there, like chitinase and glucanases, which are the PR gene products, they will get accumulated. And they use regulated pathway of jasmonic acid and ethylene as compared to SAR, which is using salicylic acid mediated pathway. So how this ISR is working? There are 2 to 3 mechanisms. First one is structural and ultrastructural cell wall modifications in the host plant. So this PGPR inoculation induce lignification of cell wall. They will thicken the cell wall. They will reinforce the cell wall structure. This altogether delays the infection process and allows sufficient time for the host to build up other defense reactions. And it will also restrict pathogen growth to the outermost layer of the plant tissue, and they will not allow the pathogens to get easily entered within the plant tissue. Some biochemical and physiological changes induced by this ISR inducing microorganism includes higher level of phenol accumulation as well as defense enzyme accumulation. Defense enzyme includes peroxidase, which is important enzyme in lignin biosynthesis. So basically it will help in thickening of the cell wall. They construct intramolecular linkages and catalyze the oxidation of many phenolic compounds to highly toxic quinones, and thereby reduce pathogen infection. Polyphenol oxidase, it is also having role in oxidation of phenolics to quinone. And phenylalanine ammonium lyase, this is the key enzyme for synthesis of salicylic acid, phenolics and lignin precursors, and phytoalexins. So this all together, this leads to the restriction in pathogen infection. Moreover, induction of hydrolysis and hydrolytic enzyme also occurs, like chitinase and beta-1,3 glucanase, which can restrict the growth of pathogen. So another thing which we have tried is biocontrol bacterial consortium, which is fortified with botanicals. We all know that botanicals are very helpful for control of pest and diseases as well as microbial agents. So what we have done, we have tested 3 biocontrol agents, Providentia vermicola, Pseudomonas putida, and Pseudomonas fluorescens. These all are our native isolates, and we have fortified that formulation of consortium formulation with plant extracts of Azar directa indica, then Ipomia carnea, and Brassica juncea. These all 3 are having a very good capacity, and we have targeted nematodes over here. So we have prepared a fortified formulation which is having shelf life of 10 months. And this, when this fortified formulation is applied into the field, then you can see that there is increase in yield and plant growth parameters, as well as there is a reduction in root knot index and soil nematode population observed. So we can say that as compared to chemical control, you can see that there is a significant reduction in root knot index was observed. So we can say that this formulation is working in control of nematodes, especially root knot nematodes in cucumber. These are the some pictures of that experiment. After developing this technology and testing in field, we have also given this product to various farmer. Around 50 farmers we have covered, which are practicing this cucumber cultivation in net house and greenhouse. And they here also you can see that in figure A, it is farmers field improved practices in which we have given formulation, and lower one is the local practice, which is normal farmers practices. And here you can also see improvement in crop health. So this is all which we are doing newer in this area. Now third one, which is environment management through microorganism. So environment is the basic thing in our inaugural session. Also it is mentioned that environment health is climate change is leading to very unpredictable weather, and pest and disease incidences are also increasing due to that climate change. So how being an agricultural risk, how we can reduce this climate change impact, that also we are working in. So environment management, we have targeted 4 areas: biodegradation of agro waste, mitigation of greenhouse gas emission from agricultural system, degradation of plastic, and bioplastic production. So first of all, I will like to say this is also a nutrient aspect, biodegradation of agro waste by microorganisms. So you can right now in this season, due to rice stubble burning in the northern part of India, you will experience that smog problem, and shortly we will see many news channels will cover that item. So that is due to the short span between the rice harvesting and wheat sowing. So what we can do over here is we have developed some cellulolytic and lignolytic microbial consortium. So these are the cellulolytic organisms which can digest cellulose, which is the major component of any crop residue, with the help of enzyme endoglucanase, exoglucanase, glucosidase, and cellobiose phosphorylase. These are the enzymes which are found within this microorganism responsible for cellulose degradation or cellulolytic activity. Some of the microorganisms are having lignolytic activity. We have supplied lignin in the plates and seen that this microorganism can luxuriously grow on this lignin containing media, utilizing this lignin as their carbon source. Here in figures which are having blue plates, you can see that surrounding the colonies of microorganism, you can see the zone of clearance. This is due to the lignin degradation or solubilization, whatever is there, with the help of enzyme lignin peroxidase, manganese peroxidase, and lacase. These are the 3 enzymes which we have found in our organisms. So basically how these organisms are working, first of all, we have isolated these strains and tested it in plate, and then we have tried it on filter paper, which is also having cellulose as the basic material. So here you can see that in T8, T9 and T11, which is comprising of different bacteria and fungi, here you can see that it is completely degraded. Filter paper is completely degraded after 9 days of inoculation. And this can be seen in close-up with lower diagram. Here you can see that slowly this microorganism are forming biofilm surrounding the edges of this filter paper and start degrading that cellulose containing filter paper. So similar things will happen with agro waste. So this was then transferred into the flask assay. Here we have tested cotton stalk waste. So at 0 days and at 95 days, you can see that inoculated flask will show you very good degradation of cotton stalk. It is also a very huge problem. Cotton stock waste is similarly here in lower figure you can see that in as compared to control, T8, T9 and T10, which are having different formulations, showed very much faster degradation as compared to control. So then this is also tested in crates or plastic containers, and here also you can see that at 90 days it is very converted in very good degraded compost as compared to control. You can see that this is the control at 90 days and this is inoculated when it 90 days. So this is the difference in degradation ratio. So here are the results. Here are the degradation rates and improved CN. Here are the CN ratio of various cotton waste degradation experiment, and CN ratio was also improved in the treatment which is inoculated with this consortium product. This consortium was also tested for banana pseudostem waste degradation, which is the major problem in our region, where banana pseudostems are just dumped on the roadside and creating pollution. So that banana pseudostem waste is inoculated at farmers field with this bacterial biodegraded compose consortium, and within 90 days we have got very good quality degradation at farmers field. Similarly, we have tried this in vermicompost experiments also with banana pseudostem waste, and here also we can see that as compared to untreated, the treated one is showing very good quality compost formation. So accordingly, we have modified the NADEP method of composting, in which we have inoculated this lignocellular-lytic microorganisms, and here we can see that incubation time for composting will decrease up to 64 days. Of course, this is another experiment. So here in that case, we have got this result in 64 days, and after very good degradation of this material, we have enriched that material with azotobacter and PSM, that are the biofertilizers. So we have got very good quality compost with total nitrogen and phosphorus ratio increased. Second aspect which we are targeting and which is my area of research or interest too is bioremediation strategies for mitigation of methane from rice agriculture. So first of all, I will like to introduce something to you, that these are the very recent reports. In 2019 only NASA has published these reports on website to alert us. So what they observed is the largest glacier in Antarctica is rapidly decaying, and it is showing hole in there. It is having a huge cavity in its structure. It is about 14 billion tons of ice is melted within last 3 years due to this global warming, and these are the authentic reports from NASA website. Similarly, in East Antarctica, the largest glacier, which is known as Totten Glacier, has also started melting, and number of small glaciers has also been activated and started melting due to this increasing global temperature. Here in red areas you can see that this is the major glacier, and surrounding that number of small glaciers are there that has started melting. So this is somewhat alarming sign for us also. And one major thing which is also not considered by scientists is methane clathrate. These are nothing but ice which is containing methane or having entrapped within methane within its biomass or its mass. So when this ice will melt, then it will emit methane in atmosphere and increase concentration of methane within the atmosphere. So the previous thing, melting of glaciers, is also correlated with this melting of methane clathrate, and methane clathrate will definitely emit large amount of methane in the atmosphere. And if you burn that ice, then it will if you show fire to that ice, then it will start burning. That type of methane clathrates are already available in Antarctica, and if this glaciers and all that thing will melt, then this will definitely release a huge quantity of methane in the atmosphere. So if we see the greenhouse gas emission by gases, then carbon dioxide is the major one, 65%. It is coming from fossil fuel burning and industrial processes. 11% is coming from forestry and other land use. Methane 16% is there, nitrous oxide 6% is there, and fluorinated gases are 2% there in atmosphere. So being an agricultural scientist, if we look at agriculture, forestry and other land use emission, then it is 24% emission. After energy sector, we are emitting largest amount of GHGs. And if we look at the continent-wise scenario, then Asia is the largest producer, contributing about 44% of the total GHG emission. And from Asia, if you look at the scenario, then our Indian region is having a major role, having 17.7% emission from our region. And among this 77%, enteric fermentation is emitting 34%, whereas paddy crop production is emitting 22% of GHG in this Southern Asia region. So because we are growing paddy in high intensity, right? So why we have to concentrate on methane and nitrous oxide? Methane is emitted from rice cultivation, that you all know, and nitrous oxide is also emitted from fertilizer usage. And we have to look at global warming potential of this greenhouse gases. So methane is having, as per recent IPCC report of fifth assessment report, it is having a global warming potential of 28. Means one molecule of methane is equivalent to 28 molecules of carbon dioxide when it comes to global warming effect or capturing of infrared radiation. Similarly, nitrous oxide is having global warming potential of 298. So you can see the seriousness of these gases, because in smaller amount these gases can do a large thing in as far as the global warming is concerned. And their atmospheric lifespan, if you look, then it is methane's atmospheric lifespan is 12.4 years, and nitrous oxide is having 120 years lifespan in atmosphere. So this is somewhat which we have to concentrate. We have to reduce this emission from our ecosystem. And remember the one thing, that for methane there is no remedy except biological. There is no remedy for methane in globe. So if we look at the rice growing area in world, it is 158.8 million hectares, and in India it is 43.86 million hectares. So we are approximately emitting 3.27 million tons of methane from rice cultivation in a year. So this is how methane and nitrous oxides are produced. These are very basic mechanisms. This is the methane production scenario. Methanogenic bacteria carry out anaerobic degradation or decomposition of organic matter, and it will produce methane. Then methane is being emitted from ecosystem by a number of ways, but remedy is provided within the ecosystem itself in the form of methanotropic bacteria. These organisms can consume methane. How that organism consume methane is, number of enzymes are involved over here. Methane monoxygenase is the key enzyme which carry out methane oxidation to methanol. Then methanol is converted into formaldehyde. Formaldehyde has three ways to go. Either it will be converted into carbon dioxide and incorporated within the plant biomass, or it will be incorporated into the microbial biomass of methanotrop type 1 or in type 2 methanotropes through different pathways. So there are a number of strategies given by scientists world over. That is water management. If you manage the water, then there are chances that nitrous oxide emission will get increased. And SRI method is also given to farmers, but somehow less adoption of these techniques are there, because it requires change in the traditional mindset of farmers, and that is a very hard thing to do, as we all are agriculturists, so we all know that thing, that constraint. Then organic matter management, crop management, development of new varieties. You can develop new varieties which requires low water. And fourth one is management of microbial community. This one is the easiest method. It do not require any change in the mindset of farming community. So we can easily do this thing. So what we have done, we have isolated methylotropic bacteria from rice ecosystem and tested its enzyme activity, production of methane monoxygenase activity. Here in these plates, we have inoculated these tubes which are evacuated one with methylotropic bacteria and methane gas as a sole carbon source. And after 10 days of inoculation, we have checked their population. In upper two plates, methane is not provided. So there is low microbial growth, whereas in lower two plates, methane is provided and you can see the luxurious growth of microorganisms. So you can say that from previous pathway, what I have shown you, that microorganism can utilize this methane and incorporate within their biomass. So it will go inside the microbial biomass as a carbon. So we don't have to worry about that. So these are unique thing about this methylotropic bacteria. So we have also tested presence of genes for soluble methane monoxygenase. We have found particulate methane monoxygenase, which is another form of methane monoxygenase enzyme. We have also found that gene. Then methanol dehydrogenase enzyme we have found. Of course, there are much more things to do at molecular level in this area, and we are planning that studies right now. So after preparation of and testing in laboratory, we have tested in farm at farm level, and we have collected that methane gas from rice ecosystem. What we have done over here, we have reduced fertilizer N and P by 20% because this organism, methylotropic organism, are having plant growth promoting activity also. One strain is also having capacity to fix nitrogen, and up to as per FCO norms, they are qualifying as a biofertilizer. So we want it to promote as biofertilizer, because if you go to the farmer and tell them that use this product and it will reduce methane emission, then they have nothing to do with that methane. They do not get easily convinced for that. So what we have to do, we have to promote it in another way. In Gujarat region, due to this government efforts, number of farmers are well aware about biofertilizer. So we just have to give them this bottle and say them that apply this will reduce your chemical fertilizer and all that. But hidden benefit or Trojan horse mechanism for this is it will reduce methane emission from agro ecosystem. So we have devised this gas collection assembly, which is somewhat designed by us over here, and we have collected gas samples over the day and over the growing season. And here also we can see that these are the treatments, treatment T4, T5 and T6, in which we have inoculated methylotropic bacterial consortium. We can see reduction in methane emission. These are conducted at our farm level, and untreated samples have shown somewhat higher methane concentration. Similarly, at another location, we have seen the same thing. And here are the pictures of field trial. Here you can see that T1, which is having 100% recommended dose of fertilizer, and T4 is having 80% recommended dose of fertilizer. With this methylotropic bacterial consortium, we have got good yield out of that, and there is no loss in yield we have observed. So this has generated confidence in us, in our technology. And then we have got a DBT funded project, and we are now through with the technology. We have demonstrated it to the farmers field level, and we have got some concrete result. And right now we have filed a patent for this technology, and that gas collection assembly, we have filed 2 patents out of that. So these are the unique thing which we have done. And another thing we have done out of this research is we have deposited one standard strain of Bacillus aureus from AAU in 80 cc as a standard strain. So whoever want to work on methylotropic bacteria, now they can use this strain as the standard strain by collecting it from 80 cc. So another area which we are touching right now is plastic. Plastic is also having.
Madam, may I request you to please expedite it, because already we are running short.
Yes, sir. Last 2 slides are there.
Thank you very much. I'm sorry.
So this plastic is colonized by this microorganism, and it can fasten up the process of degradation. Of course, this is not a magic bullet. like 500 years process can be completed into 15 days. This is not like that, but yes, of course, this has potential to reduce the time for decomposition. And in plastic also, we are targeting bioplastic production from azotobacter, which is known biofertilizer. It is having capacity to produce PHA and PHB, which are there in their microbial cells, and that can be used for bioplastic production. So this is the team microbiology of AAU. This team is led by Dr. R.V. Vyas sir, which is right now acting Vice Chancellor of Anand Agricultural University, and this is our staff. And lastly, I want to end up with saying that let's save the earth by microorganisms, because they are very great, superior and powerful creatures on earth. Thank you very much.
Thank you very much for the very nice presentation. Is there any question? If there is no question, then sir, may we start the another session?
Thank you very much, madam, for a nice presentation. So because of this time limitation of the time, so I forget to introduce the chairman of our session, Dr. Angad Suryavanshi sir. He is the head of Department of Plant Pathology and Associate Dean, College of Agriculture, Latur. And he is working as a principal right now. He is a co-host. Sir, I request you to unmute, sir. Dr. Angad Suryavanshi sir, please unmute you. And this session is co-chaired by the Dr. Lalit Mahatma sir, the man of biofertilizer in Navsari Agriculture University and the pioneer worker in the biofertilizer under South Gujarat condition. And the reporters are Dr. Meena Koche from the PDKV and Dr. Sanjay Patel from the College of Agriculture, Bharuch. I request Dr. Angad Suryavanshi sir and the Lalit Mahatma sir, please chair the session, sir, and the reporter to the Meena Koche and Sanjay Patel sir. Please, sir. Now, Mahatma sir, the platform and the session is yours, sir. Please.
Thank you very much. Thank you very much. With the permission of Honorable Chairman, Dr. Suryavanshi sir, I invite all the speakers, and I request all the speakers to please look at the time, because already we are running very short of this, and after that we have to complete one more session. So I request all those who are having oral presentation to come one by one and present their presentation. First, the presentation will be by the Ajit CR, and after that Bhanupriya should be ready. So dear friends, 5-minute time is allotted to you, and I request you to more focus on the work what you have done or you are doing, instead of discussing more about the methods, because which are uniformly for almost all the activities, and more of the introduction part. So I request here with Ajit to present his talk on the influence of native isolates of the Trichoderma species on nutrient uptake in tomato plants. After that, Bhanupriya should be ready. Further, I request all the speakers to please have some point mentioned in the chat. Save some point mentioned in the chat, in the sense that whether you are present for the presentation or not. If there will not be any response in the chat box, then I will consider that the speaker is not present here for the presentation. The chance will be given to the next speaker. So please have a point in the chat box so that I can see and start it. Is Ajit is there? Ajit CR. The first speaker of this, Ajit CR, is present here.
If you are present, write in a chat box. If any,
please write in the chat box.
If you join from another name.
Otherwise, Bhanupriya should be ready. With
Bhanupriya sir, I think she is not present, sir.
Bhanupriya is not present, then we will go for the next presentation by the Desai NB.
Sir, Bhanupriya is present. Bhanupriya is present, sir.
Okay, so what I request, because we have to manage the time also, Doctor. Okay. I request all the speakers to give some point in the chat box. Bhanupriya, can you start?
Sir, she left.
She left. Then shall I request Naresh Desai, because he has put a point in the chat box that he is present here.
Yes, sir.
Mr. Desai, are you ready for the presentation?
Desai, please unmute.
Yes, sir. Yes, sir.
Please start.
Will you share your screen or from our side?
Yeah, sir. Document sharing now.
Yeah. Okay, just a minute. Don't share. Okay, just a minute. We will share.
Okay, sir.
We will have first oral presentation, and after that we will be having the poster presentation.
Desai, please start.
Let's start.
Okay, sir. Good afternoon to the chairman, sir, and all the participants, and my dear friend and the student. Today, myself, Desai Naresh, I am pursuing PhD at Anand Agriculture University. Today, I am going to present my research work on the integrated nutrient management in W Indian bean in crop variety GNIB 22. Sir, next. This is content, sir. Next. Sir, in an introduction, okay, integrated nutrient management is a combined application of the different resources like the organic matter, inorganic, and biofertilizer for the sustainable crop production on the long-term basis without the degrading our natural resources. So next. Why we need? Because the availability of the why we need the integrated nutrient management, because the lack availability of the fertilizer and also the soil pollution is there, and the continuous depletion of the soil nutrient is there, and the unavailability of the chemical fertilizer as per requirement. Sir, next. Sir, the mainly component of the INM are the chemical fertilizer, organic manure, and biofertilizer. For today's, the biofertilizer is main concern. So we are discussing about the biofertilizer. What is the biofertilizer? Sir, next slide. Biofertilizer
Can you go to the
are the preparation which are okay, sir. Methodology slide, please. Directly.
Yeah, please.
Sir, okay. So, the experiment is conducted at the college farm of the NM College of Agriculture at the Navsari Agriculture University, Navsari, in the Rabi season of 2017-19. And the experimental design used for the experiment is the factorial RBD with the 3 replication, and the total 12 treatment combination are used in the experiment. And the crop use is the Indian bean and the particularly variety the GNIB 22, which is recently released by the Navsari Agriculture University Pulse Research Station. And the total spacing used is the 60 by 30 centimeter in the crop. Sir, next. Total 12 treatment combination are used, in which the 3 RDF doses are the 50, 75 and 100% RDF, and the 2 organic matter sources like the FIM and biocompost 2.5 ton per hectare. And the biofertilizers, 2 application means the 2 sources of the biofertilizer, with biofertilizer and without fertilizer. In the biofertilizer, the rhizobium in PSB is used as a seed treatment for the particular pulse crop Indian bean. So next. The first table is showing that the integrated nutrient management on the plant population, which is not all the treatment of the biofertilizer and also the chemical fertilizer are not significantly affected on the plant population. Sir, next slide. This table shows that the effect of the integrated nutrient management on the plant height of the Indian bean, in which at the 30 days and at 45 days, the biofertilizer application of the rhizobium plus PSB biofertilizer are increasing both the 30 days and 45 days plant height over the no biofertilizer application. This might be due to the positive effect of the nutrient on the growth character, and which is augment the cell division and the cell expansion in the plant time. This table shows that the number of branches, how the number of branches per plant is increases at the 30th day after sowing and the 60 days after the sowing. This biofertilizer application are increasing the height over the non-biofertilizer application at 30 and 60 days. This might be due to the inoculation is benefited by the providing the atmospheric nitrogen fixation and the phosphorus in available form, which is rendering near the root periphery, and this is enhancing the availability of the phosphorus, which favor the nitrogen fixation and the photosynthetic activity in the plant, and which is more accumulated the diameter and the number of branches per plant. So next. This table shows the diameter accumulation in which the rhizobium plus PSB biofertilizer application is shows the 24.44 gram of the diameter accumulation per plant over the non-biofertilizer application. This shows that the number of branches is increases, so the diameter accumulation is increasing. So next. That is to 50% of the flowering is not affected by the any application because it is a genetic factor which is governed by the particular variety. So next. This table shows that the number of the pod per plant which is affected by the integrated nutrient management, in which the application of the rhizobium plus PSB is increasing the number of pod per plant over the no biofertilizer application. So next. This the number of seed per pod is not significantly affected by the any application. So next. Also, the pod length is not significantly affected by the any application of the biofertilizer, inorganic fertilizer, and organic manure. So next. This table shows that the test weight is increased by the application of the rhizobium plus PSB application over the non-biofertilizer application. This increasing into the this test weight is due to the increasing into the photosynthetic activity. So the more source to sink to the source accumulation is there, which result into the increase into the test weight. So ultimately, the all the yield attribute and yield is increasing. So next. So the test weight is ultimately increasing. So the pod yield per plant is also increasing. So the application of the rhizobium plus PSB biofertilizer is increasing the number pod yield per plant, which is 50.51 over the no biofertilizer application, means the 9 gram per plant per increasing with the application of the biofertilizer. So next. This table shows that the pod yield is increased with the application of the biofertilizer over the non-biofertilizer. With the application of the rhizobium plus PSB, the 2706 kg pod yield is over the 2361 kg over the no biofertilizer application. In this table shows that the fertilizer into the organic manure interaction is significant, but in the today's topic, so biofertilizer, so it is not important, and this might be due to the significant and the progressive effect of the dual ejaculation of the rhizobium and PSB on the yield attribute, which result into the high seed yield and the store yield into the plant. So next. This table shows that the stover yield how is affected by the integrated nutrient management. In the application of the rhizobium plus PSB is increasing the stover yield over the no biofertilizer application. So next. Similarly, the harvest index is increasing over the no biofertilizer application because the stover yield and grain yield is increasing with the application of rhizobium. So ultimately, the harvest index is higher in the application of the rhizobium plus PSB. So next. This table shows that the how the nutrient content like the NPK content is affected by the application of the rhizobium plus PSB biofertilizer over the no biofertilizer. So the in the application of the biofertilizer, the nitrogen content is 2.315, which is increasing over the no biofertilizer application. Similar way is seen into the P2O5 content in the phosphorus, which is increasing because the phosphorus solubilizing bacteria is there. So the it might be due to the phosphorus solubilizing bacteria is enhancing the availability of the phosphorus to the plant, which is utilized by the crop, and the greater root development and the nitrogen fixation is there, which is into the soil nodulation. So the increase into availability of the nitrogen and phosphorus, which results into the greater uptake and nutrient content into the plant. So next. Similarly, the application of biofertilizer is increasing the nutrient uptake because the uptake is increased, so the content is increased into the plant. So the biofertilizer are nitrogen fixing, and another one is solubilizing this rendering phosphorus. So the uptake is increased. So next, this is the interaction effect between the organic matter and biofertilizer, which is significant for the uptake of the phosphorus. With the highest uptake of the phosphorus is seen into the O2B2, which is the biocompost 2.5 ton per hectare, and with the biofertilizer application, which is shows that the 40.96 kg per hectare phosphorus uptake is there. So next. This table shows that the organic carbon available, nitrogen, phosphorus and potassium in soil after the harvest, which is influenced by the various treatment, in which the application of the biofertilizer is not significantly affected the organic carbon and available potassium content, but the available nitrogen and phosphorus is significantly affected, in which the highest data of the available nitrogen and phosphorus is recorded with the application of the biofertilizer like the rhizobium and PSB, because the it might be due to the higher mobilization of the nitrogen, phosphorus and potassium into the soil, and also it might be due to the available of the nutrient which is not utilized by the particular crop, so it is remain present into the soil. So next, this table shows that the economics which gained by economics we gain in this, the application of the biofertilizer over the no biofertilizer is gain. In biofertilizer application, we gain the net return of the 41,903 rupees over the net return of the no biofertilizer application is the 33,121. Means the approximately 7,000 rupees profit is having the farmer with the application of the biofertilizer. So it is ultimately the benefited to the biofertilizer application, and which is boon for the farmer. And we shows the benefit cost ratio is there, which is 2.30 over the 2.4 with the no biofertilizer application, means the also the benefit cost ratio is increasing into this application of the biofertilizer. So next, this is the overall the 12 treatment combination. 12 treatment combination economics is there, in which the F3 O2B2 treatment is shows the highest net return and gross realization and the benefit cost ratio. But in point of view, for the economic point of view of the farmer, we are applied the F2 O2B2, means the 75% RDF organic biocompost as
Mr.
a organic
Desai,
manure, and the yes sir.
summarize it fast.
Okay
Time
sir,
is running
it is okay sir. The BCR is 2.91, so the this is economically feasible to the farmer. So next, from the foregoing discussion, it can be concluded that the application of the biofertilizer like the rhizobium and PSB, which improving the growth attribute, yield attribute, and yield of the Indian mint. The biofertilizer cannot be replaced the chemical fertilizer, but it can supplement the plant nutrient requirement. So it is boon for our farmer and for the economic purpose of farmer. Thank you, sir.
Thank you very much. Is there any question? In between, I request if somebody is having any question while presenting, he can write in the chat box, so we can look at the chat box and can ask the question to the speaker, isn't it? So was it your MSc research work, Mr. Desai?
Yes, sir.
Who
Yes,
was
sir.
your
My
major
MSc.
advisor? Who was your major
Sir, R.L.
advisor?
Lewis, sir, from Navsari, Surat Research Station, Biotech.
from
Dr.
Surat.
R. N. Leva, sir,
Why-
Biotechnology College, Surat.
Why
sir.
his name is
Yes,
not
sir.
there in the author?
Sir, I am putting my abstract only, sir. So I am in hurry, so sir, I am
See,
forgot
this
to
is
put in
you are
there.
a student and you are learning right now. It is a learning phase, isn't it? Please
Yes,
avoid
sir.
doing
Yes, sir.
such mistakes in the future, isn't it? Okay,
Okay,
the
okay,
next
sir.
the
Okay.
next speaker is
Okay, thank you very much.
Satyakala. Kadali Satyakala is there.
Yes, sir.
So you have to present now. And please remember, you don't waste time in introduction. Go to the result what you have obtained, and you have been given 5 minutes.
Okay, sir. Okay, sir. Within time, I will complete. Sir.
Yeah, please. Thank you. Thank you very much. Dr. Rajesh, can you please share a screen of?
Sir, my topic is on the role of sulfur oxidizing fungi.
Excuse me, what is the name?
Satyakala Kadali or Kadali Satyakala.
Yeah, role of sulfur oxidizing fungi.
Yes, sir. Yes, sir.
Yes, go ahead.
Okay, good afternoon to respected dignitaries and all the participants. Myself, Satyakala. I am going to present my master thesis presentation. Okay, that is influence of sulfur oxidizing fungi on growth and yield of soybean. As we know, sulfur is an important macronutrient and it is the integral component of plants, microorganisms, humans and animals. Okay, generally plants utilize the oxidation state of sulfate, so it must be oxidized before the plant uptake. As we know that approximately 98% of sulfur is present is in organic forms. So sulfur oxidation is must. Thereby, heterotrophic microorganisms are mainly responsible to make available sulfate from elemental sulfur through oxidation process with the intermediate formation of thiosulfate and tetrathionoate in the soils. Okay, this is
Sorry
the
for the interruption, madam. It will be better if you go for the directly main your content methodology.
Okay, next slide, please. So, microorganisms are responsible to oxidize the sulfur. As this view, these two objectives were studied. Isolation and characterization of sulfur oxidizing fungi from rhizosphere of oilseed crops. Then evaluate the effect of sulfur oxidizing fungi on growth and yield of soybean. Next slide, please. Okay, this is the methodology. For this study, we collected rhizosphere soil samples from different oilseed crops. Then prepared PDA media for isolation of the fungi, then purification and maintenance of these fungi. Next slide, please. Then screen for effective sulfur oxidation of isolates. This was done with the help of Zepak Dogs broth with the addition of elemental sulfur 1%. For this study, there is 2 steps: analysis of reduction in pH of media by pH meter, and also estimation of sulfate sulfur formation by spectrophotometer. For these two organisms, there is a drastic reduction in pH of the broth media, and also sulfate sulfur formation is very high in case of Aspergillus niger when compared to Trichoderma harzianum in vitro conditions. Then next slide, please. After that, identification and confirmation of isolates by their physiological and morphological characteristics. Then confirmed that isolate 1 is Aspergillus niger and isolate 2 is Trichoderma hertzianum. Then next. With the combination of these two isolates along with recommended dose of fertilizers and different doses of sulfur, we prepared treatment details like this. After that, observations are noted on number of pods per plant, grain yield, 1000 seed weight and sulfur uptake in a field trial of soybean with the variety Jawahar soybean 335. Next slide, please. Next one, sir. Okay. These are the results from second objective. Here, with the inoculation of sulfur oxidizing fungi, the increase of yield parameters of soybean, that is number of pods per plant, grain yield and 1000 seed weight was significantly increased with inoculation of sulfur oxidizing fungi without sulfur, elemental sulfur, okay, when compared to the control. Here, the treatment 6 shows highest, that means recorded maximum number of pods per plant and grain yield 1000 seed weight when compared to the remaining treatments here. And also sulfur uptake by the plant, that is based on dry weight matter. It is also recorded highest in T6 treatment. Next slide, please. Okay, before conclusion, I want to mention one thing. Before going to the field trial, we also conducted analysis of initial status of sulfur in the soil. And after completion of the crop, we also estimated the available sulfur content in the soil. This was also increased after completion of the crop because it is inoculated with sulfur oxidizing fungi. Then coming to the conclusions, the yield parameters of soybean, that is grain yield, number of pods per plant and 1000 grain weight was significantly increased due to inoculation of sulfur oxidizing fungi. Then sulfur oxidizing fungi appear particularly useful for oxidation purpose since their spores or mycelium can be produced economically in large quantities and have good survival characteristics both in the inoculants and in the soil. Because of sulfur oxidizing microorganisms, we can reduce the doses and cost of chemical fertilizers to increase the crop production. An additional advantage of these fungi is that technology can be used for large-scale production, and also they behave as biocontrol agents such as Trichoderma hertzianum already exist in the soil and readily to be adopted to inoculate sulfur oxidizing fungi. So sulfur oxidizing process can be efficiently done by the sulfur oxidizing fungi and also it has a potential biocontrol agent. So we can use, thereby we can reduce the doses and cost of the chemical fertilizers.
Thank you very much for the nice presentation and completion of your presentation within the time. There is one question in the chat box I would like to repeat. The question is like, what is the method and dose of fungi during application?
Seed inoculation, sir.
It was seed inoculation. And what are the doses?
5 grams per kg of seed, sir.
5 grams per kg per seed. It was carrier based or liquid?
No.
It was carrier based.
Yes, sir. Powder based fertilizer. Sorry.
It was powder based formulation, isn't it? Fine. What is the recommended dose of sulfur for the soybean?
20 kg per hectare, sir.
20 kg per hectare. Yes. And in your one of the treatment, you took 20 kg per hectare sulfur, and then you used the recommended dose of fertilizer. So what it was having, by the way?
Sir, it is the actual recommendation of chemical fertilizer, that is recommended dose of NPK and recommended dose of sulfur, sir.
What was the source of sulfur in case of your research?
Elemental sulfur, sir.
You used elemental sulfur 20 kg, isn't it?
Yes, yes, sir.
Okay, thank you very much. Is there any question? Now I request the next speaker, Dr. M.D. Kuhn. He is there as per the chat.
Okay, sir. Thank you.
Thank you very much, madam.
Thank you, sir.
Dr. Kuhn, you have been given 5 minutes to complete your presentation. So you need not to discuss all those introduction and other part. You should directly focus on the result, what you have obtained, and material and methods, if some unique materials and methods you have used in the research. Please carry on.
Sure, sir. Respected chairman, co-chairman, respected professors and dear friends, myself, Dr. M.D. Kunt from Navsari Agriculture University, and I am presenting my topic on effect of PGPR in the mitigation of soil stress. So initially, we all know that salinity confers the deleterious effect on the crop growth as well as plant productivity. And for the mitigation of soil stress, we do have different chemical methods, but they are not only costly, but it is causing the environmental impact too. And we all know that plant growth promoting rhizobacteria have the mechanism which is called IST, or induced systemic tolerance. And due to this mechanism, they are having the power to mitigate the abiotic stress, including salt stress. Next, please, sir. Next, please. So methodology encompassing following steps: collection of soil sample from the rhizosphere soil of mung bean, which is salinity affected. From that sample, isolation of PGPR, and isolated PGPR were screened in vitro for different plant growth promoting characters. And the most potent isolates were tested by paperboard method. And finally, two most potent isolates were evaluated under field condition on mung bean at two different locations. Next, please, sir. Now results. Next, please. Now table 1 narrated the collection of soil sample and its characterization. So out of total 14 collected sample from Navsari district, we have characterized them on the basis of physicochemical and microbial parameters. So in general, as we noted that as we increase the salinity or sodicity of soil, SCCDMNS producing PGPR increased and total microbial population decreased. So we can say that with the increment of the abiotic stress, soil have the capacity to increase the beneficial microbes like SCDMNS producing microorganisms. Next, please. Now, from the collected soil sample, from the sample number 1, we have isolated SSRS3 number isolate, which was showing most potent because it was having SCCDMNase activity, phosphate solubilization capacity, and indolacetic acid production capacity under in vitro conditions. Next, please. And from 9th number sample, isolate 2, VAD2, was also having phosphate solubilization capacity and indolacetic acid production capacity. So out of total 54 isolates, we choose these 2 isolates on the basis of their capacity. Next, please. And further, both the isolates were judged on the basis of a few more PGP characters like zinc solubilization, potash mobilization, percentage growth inhibition against Fusarium oxysporum, siderophore production, and nitrogen fixation. And we also found that both the isolates were sharing 3 to 4 characters in common. And finally, both the isolates were identified on the basis of biolog, that is 96-well biochemical method, as well as 16S rRNA sequencing method. And we found that SSRS3 was belonging to gram-positive Bacillus subtilis, and VAD2 was belonging to gram-negative serratia liquefaciens. Next, please. And these are the in vitro PGP characterizations. Next, please. Now, in terms of paperboard evaluation method under in vitro conditions, it was common that when we apply Bacillus subtilis plus serratia liquefaciens plus rhizobium in combination, it was showing highest percent seed germination, highest seedling length, as well as seedling weight as compared to individual inoculation as well as control treatment. Next, please. Now, in this particular slide, we can see the yellow box. and the box represented the height of the seedling. So we can say that last treatment, which was having the consortium application of all the 3 bioinoculant, produced the highest seedling length as compared to other treatment. So the next, please. So the treatment was carried forwarded under field condition, and we have checked the total 9 application, including absolute control as well as individual inoculation of each bioinoculant with combination of each other and combination of all the 3 bioinoculant. And we have given the seed treatment at the rate of 10 ml per kg seed, as well as we have also given soil application. And we have tested the experiment at 2 different locations, including NAU Navsari, AES3, and NAU Dati Ubrat, NAU AES4. So 2 field experiments were carried out. Next, please, sir. Now this is the field view of different treatment where T7 and T9 has been compared. T7 is the best treatment and T9 is the control treatment. So we can see the crop strand under different conditions. Next, please. Now this is the effect of PGPR on percent seed germination. So we observed highest seed germination in the consortium application of all the 3 bioinoculant, and lowest seed germination was recorded in control treatment. Next, please, sir. Now this is the effect of PGPR on root and shoot length. Next, please. This is the effect of root and shoot fresh weight. Next, please. This is the effect of PGPR on root and shoot dry weight. So in the 3 different slides, we have seen the effect of PGPR on root and shoot length, root and shoot fresh weight, and root and shoot dry weight at 15 days after sowing. And from the data, it is clear that all the 3 characters were maximum in the T7 treatment, or it was at par with T6 treatment, and lowest value was recorded in RDCF treatment. Next, please. Now, when we checked the effect of PGPR on number of branches as 30 days after sowing and 50% flowering, it remained non-significant. Next, please. Now, this is the effect of PGPR on leaf area index at 30, 60, and 90 days after sowing and at harvest. So again, same trend was recorded, where the maximum value was recorded in consortium application of all the 3 bioinoculant, and lowest value was recorded in RDCF treatment. Next, please, sir. This is the effect of PGPR on chlorophyll content, means PED value at 30, 60, 90 days after sowing and at harvest. So again, T7 treatment was the best treatment, and the lowest value was recorded in control treatment. Next, please, sir. Now this is the effect of PGPR on number of nodules per plant at 30 and 40 days after sowing. Next, please. This is the effect of next, please, sir. This is the effect of PGPR on nodule fresh weight per plant at 30 and 40 days after sowing. Next, please, sir. This is the effect of PGPR on nodule dry weight per plant at 30 and 40 days after sowing. So 3 previous tables were recorded number of nodule, nodule fresh weight, and nodule dry weight at 30 and 40 days after sowing. So highest value was recorded in the T7 treatment, where we have applied combined application of all the 3 bioinoculant, and it was at par with T6 treatment, where sericea liquefaciens and rhizobium was applied in the combination, and the lowest value was recorded in the control treatment. Next, please, sir. Now, when we recorded the effect of PGPR on number of pods per plant, highest value was recorded in T7 treatment, and it was at par with T6 treatment, and lowest value was recorded in control treatment. Next, please. Now, in terms of percent increase in pods per plant over control, it was recorded that nearly 30 to 36% rise in the number of pods were recorded. Next, please. Effect of PGPR on number of seeds per pod remain non-significant. Next, please. This is the table which narrated the effect of PGPR on seed weight per plant. Highest value was recorded in consortium application of all the 3 bioinoculant, and it was at par with sericea liquefaciens plus rhizobium application, and the lowest value was recorded in RDCF treatment. Next, please. On the basis of percentage, nearly 25 to 32% rise in the seed weight per plant was recorded due to combined application of all the 3 bioinoculant over control. Next, please. The table narrated effect of PGPR on the test weight, and it remained non-significant. Next, please, sir. Now, finally, in terms of yield attributing parameters, this is the effect of PGPR on the seed and straw yield. Now, highest seed and straw yield was recorded in the application of all the 3 bioinoculant, and lowest was recorded in RDCF treatment under saline conditions. Next, please. Now, on the basis of percentage, nearly 39 to 40% increase in the seed yield was recorded over control treatment due to PGPR application. Next, please. The straw yield also nearly 32, 43 or 44% rise in the straw yield was recorded due to PGPR application. Next, please. Now, this is the application of PGPR and its effect on the EC at 30, 60, 90 days after sowing and harvest. Generally, lowest EC value was recorded in BS plus SL plus R treatment, means TEC1 treatment, and highest value of EC was recorded in control treatment. Next, please. Now, in the
make you fast.
Yes, sir. Yes, sir. Yes, sir. In the pH, the treatment remained non-significant. Next, please. Now, in the total microbial population, highest microbes were recorded in BS plus SL plus R treatment, and lowest was recorded in control treatment. Next, please. And finally, if we calculate the economics, the treatment RDCA plus FYM gives the yield means a profit of 43,000 rupees. And if we add a PGPR extra, then it gives the income of 52,612 rupees per hectare. So there would be net gain of 9,609 rupees. So in terms of percentage, we can say that application of PGPR would increase 122% more profit as compared to control treatment. Next, please. So finally, conclusion, isolated PGPR can effectively mitigate salt stress in mung bean, and PGPR can improve the yield and yield attributing characters in mung bean. And this is the heart of the finding, that single inoculation of PGPR is effective over uninoculated control. However, combined inoculation of 2 or more PGPR acted synergistically and gives superior performance. So this is the finding. Next, please. Next, please. Next, please. Thank you, sir.
Thank you very much. Is there any question, please? In the chat book, there is no question regarding this presentation. Dr. Khon.
Yes, sir.
Is those PGPR which are generally being used for the other ecosystem, like the ecosystem which is not saline,
yes,
or
sir. Yes,
general crop plants,
yes,
can
sir.
be used for the mitigation of saline or salinity in the saline ecosystem?
Sir, actually what happens when we are considering the normal organisms, they are not having perfect mechanism to tolerate the salinity, that is EC level. And when we are isolating the microorganism from saline affected habitat, and that microorganisms have the power to adapt the salinity conditions. That's why the organisms isolated from salinity affected soil are more superior as compared to normal organisms.
So those organisms which is being used for the mitigation of salt stress should be isolated from the area where the salt stress is there, isn't it? You mean
yes,
here?
sir. Yes, sir.
Fine. Fine. Thank you very much. Is there any question?
Thank you, sir.
I request the next speaker, M. Srinivasan Rao, to please present his talk.
Yes, sir. Yes, sir. I'm ready to get.
Yes, sir. You have to use 5 minutes max.
Yes, sure, sir.
Because the time is running out.
Yes,
we have another session after that. Thank you, sir.
Yes, sir. I think it's visible, sir. My screen.
Yeah, it's visible, sir. Carry on.
Very good evening to respected chairperson and co-chairperson for this session, and organizing members as well as guest speakers and my dear participants. I am Dr. M. Srinivas Rao, Assistant Professor, Agronomy, Agriculture College, Naira, Angru. So today I would like to present my PhD work on a part of the PhD work, that is a study of organic management practices on plant growth in relation to the soil, chemical and microbial population in rice crop. So first of all, I will skip the introduction, as we all know that importance of rice. So why I have taken this work experiment, that present experiment was carried out at BCKV, Kalyani, Nadia, West Bengal, with the objective to study the effect of organic nutrient and as well as plant protection management practices, especially on soil chemical properties and nutrient release pattern in relation to the nitrogen as well as microbial population at different growth stages of rice. So we know that biological control, botanicals and plant extracts and chemical inducers all are the eco-friendly measures that controls pest, pathogen as well as that improves the soil fertility. So that's why I have taken this experiment. So the materials and methods. The field experiment was conducted in model organic form at C block farm, Bedan Chandra Krishi Viswavidyalaya, Kalyani, Nadia, during the year 2013-14 Kharif season. The soil is a typical alluvial gangetic alluvium soils with the sandy clay loams. The experiment was laid out in a strip plot design with 24 combinations, number of treatment combinations. One is horizontal strips as well as vertical strip. So main is nutrient treatment includes, main plot treatments includes, first is N1, that is a vermicompost, that is 60 kg nitrogen equivalent per hectare as a basal and as a top dressing. Then second one is mustard cake, that is also at the rate of 60 kg nitrogen equivalent per hectare as a basal as well as top dressing. Then third is combination of vermicompost 30 kg per hectare nitrogen equivalent per hectare as basal, and combination of mustard cake, that is at the rate of 30 kg nitrogen equivalent per hectare as a top dressing. Then fourth one is a combination of three nutrients, that is the Sesbania green manure, incorporation of Sesbania green manure at the rate of 20 kg nitrogen equivalent per hectare at the time of puddling, and followed by vermicompost, that is at 40 kg nitrogen equivalent as a top dressing. Then N5 equal Sesbania green manure at the rate of 20 kg nitrogen per hectare plus mustard cake at the rate of 40 kg nitrogen per hectare as top dressing. Then N6, that is Sesbania green manure at the rate of 20 kg nitrogen equivalent per hectare plus vermicompost 20 kg nitrogen equivalent per hectare plus mustard cake. That means combination of three nutrients. Then next subplot, that is this is the field view. Then subplot includes plant protection measures, that is seed treatment with Brahmastra, that is botanical. Then foliar spray with Brahmastra at different growth stages, 15 days, 30 days, 45 days, 60 days, and 75 days. That is every 15 days interval. Then P2 is seed treatment with Brahmastra plus Trichoderma harzianum, the soil application, that is at 1:40 ratio at the rate of 130 kg per hectare. Then P3 is combination of P1 plus P2. Then P4 is no treatment, that is a control plot. Then the variety taken for this is the IET 4786 Shatabdi. The recommended dose of fertilizers for that is 60, 30, 30. So next is preparation of Brahmastra. The Brahmastra is the plant extracts, plant-based extracts, that is a mixture of ingredients like water, cow dung, cow urine, and neem leaves, castor leaves, calotropis leaves, custard apple leaves, pongamia leaves, and bitter gourd leaves, then parthenium leaves. All these mentioned plant and animal products collected in a container, mix it in and boiled for 30 minutes, then cool it and kept for 30 days for fermentation. Then this material use it for foliar application as well as seed treatment. Then details of soil analysis I have taken for total nitrogen by using different methods, microgelral methods, we all know this one. Then the balance sheet of nutrients. So balance sheet of nutrients in the soil was determined by using the formula, that is B equal to 5 Y minus X minus A minus N, that is uptake of nutrient by crop, initial nutrient status of the soil and final nutrient status of the soil, and nutrient added through fertilizers, that is through organic manures or through extracts. Then final carbon stock assessment was calculated by using the formula area into SOC, soil organic carbon, into soil bulk density, into soil sampling depth, that is 15 centimeters divided by 100. Then brief research findings are, so if you see that vegetative growth is greatly medicated by the nitrogen source, especially in rice crop cereals, especially that is in rice crops. If you see that the graph, the pattern, nutrient release pattern is explained in this graph. So up to 45 days of the transplanting, N1, that is a vermicompost soil application, recorded the superior values for the total number of tillers as compared to the other treatments, which was followed by N3, that is vermicompost and combination with mustard cake. But this may be due to the faster release of the major nutrients in their available forms by vermicompost as compared to the other nutrient sources, that is mustard cake and Sesbania green manure, because these two they took time for decomposition as well as release of nutrients. Whereas after 40 days after transplanting to up to harvest, the crop treatment N6 recorded superior values, followed by sole mustard, that is N2. So N6 is the combination of three, that is green manure as 20 kg equivalent nitrogen plus vermicompost 20 kg equivalent nitrogen plus mustard cake. So that recorded major superior values. Then it followed by sole mustard application, that is N2. The change in the nutrient release pattern during the first few weeks after applying organic amendments are important as it implications crop growth. and it directly significant contribution to the tillering number as well as dry matter production. So ultimately the grain yield also improved. The N2 recorded the highest grain yield, N6 recorded the highest grain yield followed by N2, but those two are on par with the each treatment. So next is soil characters, that is main organic carbon, available nitrogen, available phosphorus, available potassium. After the end of the experiment, all these N2 treatment recorded the highest values. Whereas in total microbial population, if you compare total bacteria, actomycetes, fungi, the treatment N6, that is the combination of three nutrient organic sources, that recorded the highest values for total bacteria, actomycetes as well as fungi. So if you see the balance sheet of the nitrogen, at the end of the final initial stage for all treatments, the nitrogen status is the equal irrespective of treatments. Then final stage after experiment, N2 recorded, that is the sole application of vermicompost mustard cake, recorded the highest values. The balance sheet of nitrogen after the completion of 2 years experimentation under different nutrient management practices has been worked out. The sole mustard cake recorded the exhibited maximum positive balance as compared to the other treatments. It is mainly due to the less loss of nitrogen from the soil and efficient use by the rice crop. We can see the graph in soil nutrient treatment as well as plant protection treatments and combinations. Then next is the percentage increase and decrease in the initial to the final organic status. In this also N2 recorded the highest values, a percentage of increase in nutrient status in organic carbon content, total nitrogen, phosphorus, potassium, as well as final carbon stock in the soil. So in this graph, we can see that bacterial population, microorganism population in respect to the plant growth, that is nutrient uptake, nitrogen uptake, in relation between the increase in microbial population to uptake major nutrients. Incorporation of green manure in lowland rice might have scavenged the soil mineralization, prevented the nitrogen losses through denitrification, and also helped effective recycling of nitrogen. The dynamics of various nutrients in the soil and the soil microbial activity are variably affected by the composition of the different organic nutrient sources. So in this N6, we have given three combinations. So the microbial population, that food habits are varied in microbial population depends on bacterial, actomycetes, and fungi. So the N6 recorded the highest plant uptake as well as the microbial population.
Sir, last minute.
Sir, 10 minutes is over.
Yes, sir, completed, sir. So after this results, it's concluded that organic package that equivalent containing Hispania, green manuring, vermicompost, that is N6, recorded the maximum values as compared to the remaining treatments. So future scope means that the great need of studying the nature of decomposition and different sources of organic nutrients, either sole or in combination with nutrient releasing pattern, that is most important, at different growth stages. And it is relevant to study the relationship among the quantum of availability of nutrients in soil in their economic yield in system manner. So thank you, sir.
Thank you. Thank you very much, sir. There is a question in chat box. What quantity of the trichoderma you use for the seed treatment?
So that is a 1:40 ratio, sir. That one part of trichoderma powder form, then 40 parts of the soil mixed with the soil along with some part of cow dung, sir.
But in seed treatment, you used soil also as one of the components.
No, not for soil, sir. Only trichoderma, sir.
So how much trichoderma you used?
250 grams, sir. Actually, 5 grams per kg seed, like that.
5 grams per kg seed.
Yes, sir.
This is 60 kilogram nitrogen equivalent you used in vermicompost and all other things, na?
Yes,
so
sir. Yes, sir.
How much it is coming? 60 kilogram means it's a huge quantity.
Too costly, sir. That's why, sir, in farmers' condition, it is difficult to practice sole practice, sir. Depends on sole application of any organic sources. That's why combination of any green manure as well as mustard cake or vermicompost or any other organic sources. That is the final, that gives the less cost of cultivation to the final farmers, sir.
Okay, thank you. Thank you very much.
Yes, sir. Thank you, sir.
After that, Dr. Sanghani is there?
Yes, sir. Sanghani is here.
Can you please?
Before him, Muthu Kumaran.
Yeah. I'm sorry, I missed it.
No, sir. He is present, sir. Muthu Kumar.
Yeah. I wrote Muthu Kumar. Yes. Dr. Muthu Kumar, please continue.
Sanjay bhai, share karo.
Sir, are you hearing me?
Yeah, you are audible and visible, sir. Please continue.
Yes, sir.
And come to the directly, sir. Your objective, methodology, and result.
Yes, you know well, sir. Bhendi is an important and the most popular vegetable crop. The fruit borer is the major insect pest causing damages up to the 90%. You know very well the pesticide cause the different damages to the environment as well as the non-target organism. So that's we are going with the post-plant resistant is the one of the important basic components in the integrated pest management. Next one. So here you have see the different symptom of damage caused by the areas vitilla. The different bio stages. It is start with the larva, pupa, as well as the adult of the areas vitilla. The next one. Yes, next one. Yes. Regarding the trichomes, trigome is nothing but the plant hairs. It is the common anatomical features are present in the plants. It's offer the resistance to the insect pest. So here it's affect the insects locomotion, attachment, sheltering, feeding, digestion, as well as the oviposition. So here I identified the two different type of the trichome, glandular as well as the non-glandular. Other types we will see it later. Next one. So here you have seen the different type of the trichomes which are present on the plant. It's offering the resistance to the different insect pests. So here we are going to discuss some major glandular as well as non-glandular trichome which are present on the bendy and how it's arrest the insects movement by application of different microbial inoculants. Next one. Yes, objectives of the study is to evaluate the influence of different biofertilizer on the trichomes of bendy and their hybrid derivatives against the shoot and the fruit borer areas vitilla. Next one. Yes, regarding the materials method, here the 38 bendy accessions evaluated. Among the 38, 3 promising accessions identified. Salem local, Madurai local, and Anu. These are the promising accessions, intercrossing them and obtain the different hybrids. And the Arca anamica is also evaluated as the susceptible check. Next one. So here, by using the biofertilizer, it's an induction of resistance to the areas vitilla. Azospirillum, phosphobacteria, pseudomonas, and K solubilizer, potash solubilizer, Preturia aurantia. So these are the different bioinoculants are used to induction of resistance in the bendy against the shoot and the fruit borer. So here I have listed out the different dosage per part, then the day of application, then method of application. Next one. Then regarding the estimation of trichomes on the fruit surface of bendy, just we have to cut the 1 centimeter square and under the microscope observe the how many numbers of trichomes are there. Then the length and the breadth of the trichomes hairs is also measured by using the compound microscope. Next one. Yes, regarding the entrapment test, it's matured fruits collected from the plants. It's a treated plants and it's releasing the 10 neonates larvae. Just we have to observe up to the 12 hours. Then, if no reaction was evident, the neonate was designated trapped and dead. The next one. Impedance test. Just we have to release the third instar larva on the fruit surface by using the instrument which is developed by me. Just it's releasing the larva in the one end. Next one. So the next one. Yes, this is the instrument. Actually here we have to kept the skin of the bendy in between the two foam sheet and releasing the larva in the one end, and how much time taken by the larva reach to the second point. So if the trichomes are higher, means larva took the maximum time to reach the second point. If the less number of trichomes are present, mean it can easily travel and it's reach to the second point. Next one. Yes, regarding the result, among the different treatment on the bioinoculant, K solubilizer treated hybrids have the maximum numbers of trichome when compared to the parents. Then among the hybrids, hybrid 2 had the maximum number of trichome length as well as the breadth is also high on the hybrid 2. Among the 4 type of trichomes, here 2 types were the glandular and another 2 types are the non-glandular. Next one. So here you have see the two different type of glandular trichomes are identified. So here the type 1 is the one head are present on the trichome. In case of the type 7, we have see the two heads present on the top of the trichome. The next one. So here you have see the non-glandular trichomes. Yes, next one. Sir, next one, please. Yeah. Yes, this is a non-glandular trichome. So it's a whip-like trichome is the type 1 trichomes. In type 2 trichomes, it's have the some of the head-like projections in present in the end of the trichomes. The next one. Then regarding the entrapment and impedance test, to analyze the entrapment of Aedes vitella larva, the maximum larval mortality was recorded on the hybrid 2 treated with the potash solubilizer. Estimating the movement of larvae, it was observed that the larva took the maximum time on the hybrid 2 because of it have the high density of the trichome, both the glandular as well as the non-glandular. Next one. Next one, please. Yes, here you have see the larva trapped on the non-glandular trichome, which is treated with the potash solubilizer. So here you have see the larva, so pseudo-legs have the crochets are there, that the crochets they can easily trapped by the trichomes. So here the photograph. The next one. Yes, this is the glandular trichome. When the larva it's a move on the fruit surfaces, the glandular trichomes are the burst and releasing the sticky material, so that the larva took the maximum time to reach to the second point. Next one. Yes, regarding the conclusion, the increase in the population of K-solubilizing microbe in the rhizosphere would enhance the ready availabilities of the potassium. So once we have enhancing the potassium level in the fruit tender, it's directly enhancing the trichome density also. The trichome density have significant positive correlation with the K content. If we are going to enhancing the availability of potassium content in the soil, it's also enhancing the content of the K in the fruit. So if the content of the fruit is enhanced, it's also enhancing the numbers of trichome, both the glandular as well as the non-glandular. So by using these methods or different breeding programs are using these bioinoculants, we have to enhancing these characters in the bendy to management of shoot and fruit borer Aedes vitella. Next one. Thank you.
Thank you very much, Dr. Muthu Kumar. I have one question. That you used 200 mg per pot different microbes, when in case of potash, Frechuria aurantia, you use 300 ml per kilogram of the seeds. Why there is variation between these two?
Yes, sir. Actually, it's not the 300 ml, sir. 3 ml per kg of
yeah, it is 3 ml per kg.
Yeah.
But instead of this, if you have used 200 mg prepared product of the Frechuria aurantia, then it would be more uniform and it would be more authenticated.
Yes, sir. For Azoospermillum phosphobacteria is 200 gram because of directly mixed with the pot soil, sir. Here it's a seed treatment, sir. 3 ml per kg of seed, sir.
But my concern is this, that also if you would have done it now, it would be uniform, because generally when we are doing research, we are taking care that everything should be uniform. Then only it can be tested.
Okay, sir. Sir, it's a method of application is very, so that we have accordingly we have to fix the.
Yeah,
yes.
See, that's very good. I appreciate that the way of work and the different type of the work you are showing here, so that I appreciate. But the question I was having in my mind, so I thought let us clear it. The second thing, sir, is that how much days it takes for fruiting in case of okra generally?
Sir, but sir, please.
How much time it takes for fruiting in okra?
Yes, sir. Almost from here, these are the some accession collected from the Madurai, Salem, it's a part of Tamil Nadu. So it's reach up to the 35 to 45 days, sir.
35 to 45 days. So,
yes,
so you took observation at the just fruiting stage 35 days, isn't it?
Yes, sir. Yes, sir.
Fine. Thank you. Thank you very much.
Thank you, sir. Thank you very much, sir.
Dr. Sangani.
Good afternoon to all, respected chairman, co-chairman, and all participants. Effect of foliar fertilization of old bare orchard. Material and method. We have spread different fertilization. First spread flowering initial stage, second spread peanut stage, and third spread marble stage of the bare fruit setting. Now we have taken the three different treatment at like urea at 2%, DAP at 2%, KNO3 at 2%, and novel organic liquid nutrient at 2, 4, and 6%, and NA at 20 ppm. Novel organic fertilizer is patented product of Navsari Agricultural University. Now come to the result. This experiment was conducted at two different locations at Bharuch and first table. It is a maturity days. Maturity days gives the non-significant result pulled over the year and location. Effect of the foliar fertilization on the TSS in the fruits. TSS gives the non-significant result, both Bharuch and Tansha, and pulled over the old. Effect of foliar fertilization on sugar content. Sugar content gives the significant result at 4% of the novel organic liquid nutrient, both location and pulled over the year. Now effect of the nitrogen content in the fruit. So T2 gives the higher, significantly higher, which was at par with T4 and T7. Now effect of the P in the fruits, it gives the non-significant result pulled over the year and location. Effect of the K gives the T4 and which was at par with 5, 6, and 7 treatment. Pulled over the year. Now come to the fruit yield. Fruit yield at the 4% of the novel organic liquid give us the significantly higher yield pulled over the year and the location. Now come to the conclusion. 3 years of the pooled result revealed that the quality of the bare fruit can be improved by foliar application of 2% and 4% novel organic liquid nutrient, whereas for giving the higher yield of the bare foliar application 4% gives the may be done. Thank you.
Thank you very much. Is there any question? Dr. Sangani, have you used any microbes in your research?
No, sir, we didn't include the microbes in the research.
So it was like some organic as well as chemical, isn't it? Novel you use, so I considered it as organic, isn't it? Novel you use, no?
We have only used.
Banana pseudostem shape you use.
Banana pseudostem.
Yeah, novel organic.
Yes. It is
a
patented product of our university. We have over 30 international patent we have done up to this.
Yeah, thank you. Thank you. Thank you very much.
Thank you, sir.
After that, H.A. Patel is not available. Then Sanjay Kumar Ji Patel is available?
Yes,
sir. Yes, sir.
Yes, sir. Present, sir.
Just 1 minute, sir.
Yeah,
just
please.
Just a minute, sir. Sanjay.
Sir, I'm audible? Mahatma sir?
Yes, please carry on.
Yes, sir. Good evening, one and all. Okay, my topic is a free living nitrogen fixing bacterial diversity with respect to seasonal variations. The objective was the isolation of different free living nitrogen fixing bacteria with respect to different seasons, their biochemical characterization of isolates, evaluation of the PGP and antimicrobial activities. For the study of isolations of microorganisms, before there is a 4 site was selected, and it was the regional cotton research station Bharuch, NARP Bharuch, agriculture research center Tansha and Achalia. Before the rain seasons and after the rain seasons, the CFU count, colony forming unit was counted from the different research center, and this was sampling was carried from the different depth of the soils. Beside this, the isolation of free living nitrogen fixing bacterial organism was carried out by using the Ashwesh mannitol agar medium before raining season and after raining seasons. So before raining seasons, the number of count was lesser, and after raining seasons, the total viable count as well as the number of free living nitrogen fixing bacteria was also higher. Now, for the different study, various properties, the total 31 isolate was carried. Isolation of free living nitrogen fixing bacterial was carried out on the Ashwesh mannitol agar. Free living nitrogen fixing bacteria was carried out, and this same organism was checked for the different PGPR properties, that is the phosphate solubilization, HCN productions, Sidarov productions, and IA productions was carried out, and from this total isolates, some of the isolates have shows the all kind of activities like a phosphate solubilizations, HCN activity, Sidarophore activity, and IA productions activity. Among these 30 isolates, 10 isolates show the phosphate solubilizing properties and 10 show the Sidarophore productions. 6 have the IA activity and no one organism produce the HCN activity. Only A19, A24, and A28 isolates have multiple characteristics to produce Sidarophore, phosphate solubilizations activity, and nitrogen fixing ability. A12 isolate have the nitrogen fixing ability as well as phosphate solubilizer and IA productions potential. A21 have nitrogen fixing, Sidarophore, and IA production potential. Now, zone of phosphate solubilization of all isolates are measured, and results showed in the that the A12 and A16 have highest zone of utilization to the colony ratio. Here the table shows that the different isolates and their phosphate solubilizing activity. Among these, isolate A12 and A16 have given highest phosphate solubilizing activity. IA productions is given by the 6 different isolates, and among these isolates 26 and A15 gave highest productions of the IA productions among the all isolates. Out of the 33 nitrogen fixing, 13 organism isolated before rainy season and 20 organism isolated after rainy seasons. And among these, those isolated before monsoon seasons have much more number of gram-positive bacteria, while isolated after the monsoon, they are the more number of gram-negative bacteria. These are the results of 16S RDNA of the different isolates, which is carried out the pre-monsoon and post-monsoon seasons. These are all are the free living nitrogen fixing bacteria organisms. The conclusion was the microbial populations along with the bacteria is increased in post-monsoon season. Before rainy season, the number of gram-positive bacterial population is higher than the gram-negative bacteria. Isolate A19, that was the Streptomyces polycolor, A24 Enterobacter species, and A28 Bacillus altidinus have potential characteristic to produce the Sidarophore as well as nitrogen fixing and phosphate solubilizing properties. Isolate A12 has characteristic to produce EAA along with the nitrogen fixing and phosphate solubilization properties. This is the some of the activities given by the Sidarophore isolates. Okay.
Thank you very much, Dr. Sanjay Kumar.
Yes, sir.
Can you give me the reason why the gram-positive bacteria were more before the rainy season and gram-negative bacteria were more after the rainy season?
Sir, generally in after summer, this gram-positive bacteria, they are generally some of they are spore forming, so they can survive in the even dry environment. So the number of higher was in the pre-monsoon season of gram-positive bacteria, and post-monsoon seasons, the number of gram-negative was higher. So there is no another other reason, but in a dry environment, the number of populations of the gram-positive bacteria is higher compared to the gram-negative.
You have taken the soil from the rhizosphere?
Pardon, sir?
You have taken the samples of soil from the rhizosphere or some barren land?
Sir, it is taken from the different depth, generally from the 0 to 15 centimeter.
0 to 15 centimeter you have taken.
Sir.
Was any crops growing in that place?
Pardon, sir?
Was any crop was growing there, or it was just the fallow land?
No, sir. It was there was no crop, sir.
Okay, fallow land. Thank you very much for timely completing your presentation. Now I request Aditi Chauhan to present her presentation, and after that there will be the last oral presentation will be presented by Chintan Kapadia, Dr. Chintan Kapadia. So I request Dr. Chintan Kapadia also to be ready after that, isn't it? Now Aditi Chauhan, please carry on.
Hey, Chintan Kapadia. Just 1 minute, sir. We are uploading this.
Yeah, please.
Sir, is my screen visible?
It is. This is Aditi's seminar. After that, Chintan Kapadia's presentation will be there.
Okay. Aditi.
Sir, am I audible, sir?
Yeah, I'm getting your voice, but let your presentation come first on the screen.
Okay, sir.
Just 1 minute, sir.
Sir, I'm sharing my screen.
Please share. Please share.
Now it is here. We have shared the screen, sir.
Sir, is it visible, sir?
Yes, sir.
Sir, can I start, sir?
Yeah, please carry on.
Okay, sir.
Skip introduction.
Skip introduction. You have been given 5-minute time to complete.
Okay, sir. Good afternoon to all. Myself, Aditi Chauhan. I am pursuing a PhD in Soil Science and Agriculture Chemistry from Navsari, and today I am here to present my MSc research work on response of fertilizer level, biofertilizer and biocompost on yield and quality of Rabi fodder sorghum at a South Gujarat condition. And the objective of this research work is to study the fertilizer level, biofertilizer, and biocompost on growth, yield, and quality, content, and uptake of nutrients. And these are the experimental materials, factorial RBD, and these are the treatment combination in which 100%, 75%, 50% RBD with biocompost and biofertilizer, and with without biofertilizer and biocompost. And the biocompost at 5 ton per hectare, and this is the biofertilizer application like azospirillum and PSB, both the seed application as well as soil application at the time of sowing. And these are the different methods which I have used for plant sample and quality parameter. And the result of this research work, so first of all, the yield of fodder sorghum, which influenced by the different treatments of fertilizer level, biofertilizer and biocompost, in which 100% RDF with biocompost and biofertilizer have significantly higher yield of both green and dry fodder yield. And the interaction of these fertilizer level and biofertilizer on both the green and dry food aid, in which 100% IDF with biocompost biofertilizer have significantly higher yield of green fodder and dry fodder, and the application of 100% IDF without biofertilizer and 75% IDF with biofertilizer, both of which are at par with first treatment. And this is the qualitative parameter in which chlorophyll content and crude protein content, in which saying the 100% IDF with biocompost and biofertilizer have significantly higher chlorophyll content and crude protein content. And this graph we can see that the application of 50% IDF without biofertilizer and biocompost have significantly lowest HCN content, and HCN content is lowest is good for the fodder purpose, and crude fiber content also low content is good for fodder purpose. So 100% IDF with biofertilizer and biocompost have significantly lowest crude fiber content. And this interaction of fertilizer level, biocompost and biofertilizer on chlorophyll content, in which application of 100% IDF with biofertilizer and biocompost have significantly higher chlorophyll content, and crude fiber content is the lowest with the application of biocompost and biofertilizer. And the macronutrient content like NPK, which is highest with 100% IDF with biocompost and biofertilizer. And this is the interaction of biocompost and biofertilizer on nitrogen content and phosphorus content. The both nitrogen and phosphorus content are significantly higher with application of biofertilizer with biocompost and biofertilizer. And this is a micronutrient content of fodder sorghum, which is influenced by different treatment, in which application of 100% IDF with biocompost and biofertilizer give a significantly higher iron, manganese, zinc, and copper micronutrient content in fodder sorghum. And this is a micronutrient uptake like NPK in fodder sorghum, which is influenced by different treatments.
Go with your objective, methodology, and the result, please.
Okay, sir. So, saying this treatment has significantly higher micronutrient uptake, and this is the interaction of fertilizer level and biofertilizer. So application of 100% IDF with biofertilizer has significantly higher nitrogen and phosphorus uptake, and this is a micronutrient uptake in which the same treatment has significantly higher iron, manganese, zinc, and copper uptake. And this is the interaction in which 100% IDF with biocompost have significant biofertilizer and biocompost have significantly higher iron and manganese uptake, as well as zinc and copper uptake. So from this experiment, we concluded that the higher yield and profit and quality like a chlorophyll content and crude fiber and protein content for the Rabi fodder sorghum obtained by the application of 100% IDF and biofertilizer and biocompost. Thank you. So,
thank you very much. Is there any question? No question, then thank you, Aditi. Now Dr. Chintan will present, and that will be the last oral presentation for this quarter.
Okay, it is audible and one can see this slide properly.
Now slide is not seen. Yeah, now we can see slide as well as can carry on.
Pardon. Okay, so myself Chintan Kapadia. My topic is Heliotrolan Microbial Consortia as an alternative strategy to enhance growth in saline environment, and belongs to Department of Plant Molecular Biotechnology, Hospital College of Horticulture and Forestry, NAU Navsari. These are my objectives: identification and characterization of salt-tolerant bacteria from saline rhizosphere, and to study this effect on the presence and absence of salt stress on a tomato plant. These are some brief findings of my particular research. Like what I did in the entire experiment, I took water from the seashore and I mixed them with the soil and make at a different EC level, like 0.2258 EC level. I use a different microbial consortia, belongs to Delphia, Enterobacter, Achromobacter, and Bacillus. And I use them as an inoculant in with the tomato plants, so that I can easily identify, I can easily evaluate the growth kinetics and growth pattern of the tomato plants. These are the location from where I have isolated the halotolerant microbia, and it is very near to Dandy seashore. These are the methods, as you all are aware of the microbiological techniques, so there is no need to discuss all these methods. These are the methods, and then these are the isolates which I have isolated from the saline environment. Okay, I have got lot of organisms, among them whatever the organism repeatedly grown on the culture media that I have listed over here. So there are some 8 to 10 organisms and their culture morphological characteristics. As we study the culture and the morphological characteristics, we learn and we identify their genus and species by performing 16S rDNA sequencing by using 1473 reverse primer and 27F forward primer. We already submitted our organism into the NCBI database, and they assigned the accession number to the organisms, and these are the accession number to the organisms. To confirm that organism which belong, they all are belongs to Bacillus only, on that we perform the phylogenetic analysis, and we got a very good phylogenetic tree. You can see all the Bacillus lying on the same clades, so that indicates that our sequencing also was very good, as well as our morphological characterization was also very good. Then we carry out the PGPR characterization of the microbes. You can see the A, B, C, there are different names given to the organisms, and there are different code name given to the organisms. And all of them were very good in terms of the production of different PGPR characteristics. We also performed at 15% NaCl concentration, as well as we have performed at 20% NaCl concentration. And the organism which are in colored red in this particular slide is that we used in a consortia. Okay, you can see all the parameters are very good at 15% NaCl concentration, but as you go on increasing 20% NaCl concentration, there is a less production of different PGPR trait by the organism. Then we perform the HPLC analysis of all those microorganisms in the presence of Picowaski growth. These are retention time, and these are the standard graph. These are the slides, and these are different results of organic acid produced by different microbes. And these are the organic acid produced by the microbes. Yeah, we haven't used gluconic acid and alpha gluconic acid, but whatever organic acid we have used, we found that pyruvic acid was produced by most of the microorganisms. These are the summary of the PGP activities shown by the isolated microorganism. And these are the microbes that we used in our consortia. One belongs to Acrobacter, Bacillus, then Delcia and Enterobacter. Now, surprisingly, you can see here the Delcia, MH122388. These are the sulfur oxidizing bacteria, and there are very limited reports of Delcia as a sulfur oxidizing bacteria reported in the literature. Only 2 to 3 reports you will find, and that we used in our consortia. These are the synergistic effects. As I told you, that when you perform a consortia, when you prepare a consortia, the organisms should be synergistic with each other. So we perform synergistic experiments, and we found that all organisms can be easily grown with in the presence of each other. You can see the consortia preparation. We add the organism in a respective number into the particular flask, and when we reach the number of organisms 10⁸ per ml, we inoculate them into the particular pot. These are the activities shown by the consortia. You can see the high activity by different consortia, with the different like ammonia production, phosphosolubilization, IA production. These are the particular antagonistic effects against the Ralstonia solanacearum. These are my enter experiments in a tub. I performed these thrice in a tubs only. As I told you that we mix the different like 40 EC seawater with the tub, so that we can get different EC of the soil, and then we fill that soil into the tubs in order to maintain that 0.2, 2, 5 and 8 EC. Now these are very important slides that gives you the idea about the entire experiments. A1, that is my control, that only contain distilled water. A2, which has a microbes in it. B1, that is a control soil, 0.2 EC. B2, 2 EC, 5 EC and 8 EC tab. C1, I took the observation at 0 day, 15 day and 35 day. I performed the Duncan multiple range test at 0.01 level of significance, as with the help of statistical software R. These are the soil parameter. You can find the first column that EC doesn't change even if you increasing the this pH doesn't change even if you increase the EC level. And these are different sodium, potassium and other parameters of the soil. Now, surprisingly, you will find that when you add the microbial consortia into the particular tub, like A1, B1 is your control, okay, without any kind of microorganism. While A2B1, fifth row, which is inoculated with the microbes at 0.2 EC. There is a surprisingly 57% increase. Well, if you see the dry biomass of root, dry biomass of shoot, and dry biomass of leaf, there is altogether 57% to 63 to 68% reduction without addition of microbes. But there is reduction of only 33%, 22%, 28%. So even though there is an effect of salt on the plants, but the effect is very less when you add PGPR. And when you add PGPR, the plant perform better at 5 EC. You can compare A1B3 with A2B3. You can see the different value. The values are very high compared to the 5 EC without inoculation. There's another table, another slide which give you more idea about. Just a minute. That something happens. Okay. Now see, there's a root length, shoot length, number of leaves, number of secondary leaves, and chlorophyll content. Same pattern observed over here also. When you compare, compare the control uninoculated at 15 days with control with uninoculated at 8 EC, you will find there is a drastic reduce, 72% reduction. While if you add microbes, the reduction is only 44%. But if you compare again with the 5 EC of uninoculated with the inoculated, it will give you almost 33 to 45% higher. So this was the effect of the PGPR on the growth of the tomato plant at 5 EC and 8 EC. As we know that most of the vegetable having the salinity tolerance level is 2 EC. 2 EC, mine is 2 EC. I am putting my tomato as 5 EC. So if my tomato with inoculated microbes give me better response at 5 EC and it gives optimum response at 8 EC, that this PGPR consortia could be helpful for the farmers, could be helpful for the further research, so that it will help in a reduction of the stress on the plants. These are the sodium potassium ratio of the leaf, stem, and root. What is this particular slide indicates that sodium contents are found more in the stem in inoculated plant compared to the uninoculated plant. So that indicates the stem accumulate more amount of sodium and doesn't allow the sodium to move to the leaf, so that transpiration and chlorophyll content doesn't affect. Last slides that indicates the results. As I already told you, inoculated halophilic and halotolerant PGPR consortia mentioned in the result enhance the tolerance limit of tomato plants from 5 decisiemens per meter to 8 decisiemens per meter. So this consortia enables plant to survive properly at higher EC level, which is a tolerance level of vegetable is 2. While this helps the plant to survive, give better response at 5 EC and optimum response at 8 EC. So this acknowledgement, I'm thankful to Department of Plant Molecular Biotechnology, Hospital of Horticulture and Forestry, Nursery Agriculture University for providing financial support to carry out such work. At the end, thank you. I'm happy to answer your queries and the questions.
Thank you very much, Dr. Chintan. Any question to Dr. Chintan? Nice work, Dr. Chintan. Any question?
Yes, sir.
Yes, sir. Can we start for the poster?
Yeah, thank you very much, Dr. Chintan. With this, we finish our oral presentation of this slot. Now we will go for the poster presentation. As per the discussion we had at the time of oral presentation in the chat, there are 3 posters going to be, like 3 participants are going to present their poster. First, I invite Ms. Banderi to present her poster. You can go for the poster presentation, sir.
HM Bandari, please.
Yes, sir.
You have been given 2 minutes time to present your poster, isn't it? Because we have given 5-minute time for the oral presentation and 2-minute time for this. Okay. You have to make it very fast, Hiral. Okay, what are the outcomes?
Okay, sir.
The research, you just have to discuss.
Good evening, sir. I, Hiral Bhandari, completed my MSc from Navsari Agriculture University in Agriculture Microbiology under major guidance of Dr. M.D. Cook. And today I am present my poster on the topic of exploring rhizospheric bacteria for biohardening of micropropagated sugarcane. Objective of my research is first one is isolation and characterization of PGPR from sugarcane rhizosphere. Second one is in vitro screening and identification of most potent PGPR. And third one is the effect of PGPR isolation on hardening of sugarcane tissue culture plant. Here methodology, first one is isolation of
Go to the result directly. Go to the result, Dr. Hiral.
Okay, sir. Okay, sir. Total 38 isolates were obtained in pure culture from and were characterized for morphological and biochemical parameters. That data suggests that isolates were diverse in terms of both the parameters. Table number 1 indicates in vitro screening result of the all 38 isolates. It is the evidence that all the isolates were variable and diverse in terms of the different reactions. Here the out of 38 isolates, 2 isolates are positive for the old reaction. This one is OF4 and SWM4. Here the figure 1 represents the percent of the total isolates showing positive reaction of specific parameter. It is the clear that highest number of isolates shows antagonistic potential, and half of the isolations are positive for protease, amylase, and lipase activity. Wherever less percent of isolates were found in positive for the SEC deaminase activity. Here the figure number 2 illustrated the application of the single or dual PGPR 2 times at the time of the primary and secondary hardening, significantly improved various plant growth parameter over the control treatment. Conclusion, among all the isolates, 2 multipotent PGPR isolates, OF1 means Myriadis profoundly, and SWM4 it means protease mirabilis were screened and identified on the basis of the lab test. Further, both these isolates have been evaluated on its efficiency of biohardening of sugarcane individually and combination with each other. PGPR application twice at the time of the primary and secondary hardening, so higher plant growth promotion over one time of application of PGPR isolate. Thus, the application of microbial consortium 2 times at the primary and secondary hardening, so most promising result over the single inoculation of PGPR, and it can be used in the biohardening of micropropagated sugarcane plant. Thank you, sir.
Thank you very much. Any question?
No question, sir. Please second.
Okay, go for the next presentation. I think this is Purvesh. Out of the 3 speakers, the second speaker is Purvesh, isn't it? Can you,
yes, sir. Purvesh.
Can you display the poster of Purvesh, please?
Yes, sir.
Am I audible, sir?
Yeah, you are audible, but you are given only 1 minute, isn't it? Because we already have discussed what is the biohardening and all.
Okay,
it is the same things what the earlier speaker has discussed, isn't it? So what is the outcome and what is the difference between your outcome and the outcome of the previous speaker? You just have to discuss that. Please go ahead.
Okay, sir. Sir, Hiral Bhandari's research work is based on sugarcane, and my research work is based on banana. Sir, I isolate 54 different bacteria from the soil samples, and from the screening, I found 2 most potent PGPR, OF2, which is known as the Bacillus subtilis, and BPF12, which is known as Enterobacter cloacoi. Further, both these isolates have been evaluated on this efficacy of biohardening of banana, individual and combination application with each other PGPR application. At the time of primary and secondary hardening, so higher plant growth promotion over the time application of PGP isolates. Thus, application of microbial consortia 2 times and primary and secondary hardening showed more promising result over single inoculation of PGPR, and it can be used the biohardening of microbial banana plant.
Okay. Thank you. Thank you very much. Any question? Can I request the third speaker, Anti B. Patil?
Yes, sir. Yes, I'm here, sir.
Dr. Rajesh, can you please display the poster of Dr. Kranti?
Yes, sir. I am sharing the screen, sir.
Please, sir. Please, sir.
Hello, may I audible, sir?
Yeah, you are audible.
Okay, good evening, very good evening, sir. Myself, Dr. Kranti B. Patil. Just I completed my PhD from Navsari Agricultural University, Navsari, under the guidance of Dr. Sonal Tripathi, ma'am. And today I am going to present my poster on effect of application of arbuscular mycorrhizal in sugarcane and the South Gujarat condition. Sugarcane crop, phosphorus is a major nutrient,
You
and
go for result, madam, directly. Everybody knows about the significance of crop and worm. Sorry,
from the 2 years experiment with the use of the arbuscular mycorrhiza with other phosphorus levels and doses, I found that with the use of arbuscular mycorrhiza 2 times, that is after planting and at the time of earthing up, 250 grams and 200 grams followingly, sugarcane growth attributes like content in cane and trash was found also significantly highest with the use of arbuscular mycorrhiza biofertilizer, and that will result into the sugarcane uptake of the phosphorus, that is 17 and 5 kg in cane and trash at the time of harvest, and significant interaction also was found significant in the terms of a number of millable canes, cane yield, phosphorus content and uptake with the use of phosphorus with split. That means 50% at the time of planting and 50% at the time of earthing up, along with arbuscular mycorrhiza, gives a significantly result in terms of NMC and cane yield, and which is found at par with the 100% application of phosphorus with the mycorrhiza as a basin, and it's found better interaction significant in terms of phosphorus content and uptake by using of phosphorus in split. The split application was not followed in the Gujarat and that south condition. The results was found better because the phosphorus as a level has a less in the soil. It gives very good results by having the interaction with roots of sugarcane crop and mycorrhiza. And from that study, I conclude that from the result of 2 years, it can be concluded that for South Gujarat condition, sugarcane crop production potential and the nutrient acquisition can be improved with split application of phosphorus along with arbuscular mycorrhiza, the fungal biofertilizer. Thank you, sir.
Thank you. Thank you very much. With this, we end this session in which there were 9 oral presentations, sir, and 3 poster presentations. At the outset, I would like to express thanks to the organizer for giving us an opportunity to co-chair this session along with the respected chairman, Dr. Suryavanshi sir. He requested me to look after this session because he was little busy in other assignment because of the so many multiple assignments Dr. Suryavanshi sir is having. So I thank you, Dr. Suryavanshi sir, for allowing me to handle this entire session. Thank you very much indeed. I enjoyed handling this session along with Dr. Suryavanshi sir, with whom I have earlier associated for one of the session as a reporter. So thank you very much everybody for the very nice presentation. Now, Dr. Rajesh, you can please start the next session. Thank you very much, Dr. Rajesh, personally.
Thank you very much, sir, for this nice and wonderful handling the sessions. As you are the expert in this theme, biofertilizer, so it will be great pleasure to share your knowledge and the experiments among the students also. I would like to thank Dr. Suryavanshi sir, Dr. Lalit Mahatma sir, Dr. Meena Koche, Dr. Sanjay Patel for handling this session nicely. Now coming and moving towards the next session, I request Dr. KB Rakolia sir to act as a chairmanship, Dr. Arya Chaudhary sir to act as a co-chairmanship, and Dr. C.M. Balia and Dr. Nilesh B. Pawar to act as a rapporteur into the session.
I
Just
also thanks my rapporteur in this session. Thank you very much.
okay. Okay. Now come Vijay Kumar Yadav. Be ready for the oral presentation. Rakolia sir, please unmute yourself.
Okay, hello. Good.
Yeah, you
Hello.
are.
Hello.
Sir,
Okay.
you have a list. First oral presentation and then poster presentation, sir. Vijay Kumar sir, please unmute yourself.
Okay, Rajesh sir.
Yeah, you are audible and visible, sir, now.
Okay, may I start?
Rakole sir. Rakole sir, please unmute. You have to give the instruction, sir.
Yes. Hello, good afternoon, everybody. Sorry, good evening. Now I request to the VK Yadav, please start the presentation.
Sir, good evening to one and all present here.
I request all the participants, instead of the formality of the instruction, directly go for your objective, methodology and the result, please.
Okay, within 5 minutes you complete your presentation.
Okay, sir. Okay, thank you, sir. Myself, Vijay Kumar Yadav. I am presenting my paper on host pathogen relationship and progress of infection of Sericlodia oryzae, the incident of sheath rot of rice. Okay, next, sir. Okay, India is, we know that 7th largest sovereign country in the world by area, with about 329 million hectares, and the net area shown, that is 140 million hectare and 39% under the irrigated condition. And most of the contribution under the rice, that is 90 metric ton. And we know that Chhattisgarh is known as a rice bowl, and rice is as a staple food of in this state. And the major contribution of rice, that is 3.63 metric ton. Next, sir. Next, please. Okay, the sheath rot of rice is one among the most important diseases inflicting the economic damage to the rice crop. And in recent times, it has been recognized as one of the major disease of the major rice growing area of the country. The characteristic symptoms that is produced by Sericlodia oryzae, that is oval to irregular lesions that develops on the flag leaf sheath that encircling the young panicle. And when we will open this flag leaf sheath, a clear powdery mass of the spores that also visible inside the sheath, and the infected panicles, depending on the severity and time of onset of the disease, they may be partially filled or sometimes unfilled. Okay, next, sir. So the present study is conducted with this sheath rot pathogen by isolating the pathogen, confirming its pathogenicity, assessment of extent of seed-borne infection in commonly grown varieties, that is Chapti and Bamleshwari, and the infection progress in germinating infected seed into seedling and up to the flag leaf sheath. Okay, next, sir. So the collection is starting from the collection of these samples from the different places of Chhattisgarh, and the uppermost leaf sheath that encircles the young panicle and showing the symptoms were collected, along with the paddy grains which is infected with the disease. They were also collected from these different regions, and the isolations were made from the 21 varieties of rice that is commonly grown in Chhattisgarh. Sir, next, sir. These are the name of these rice varieties which is commonly grown in Chhattisgarh, and the pathogens were isolated from these varieties, that is Bamleshwari, Pantfor, and so on. Okay, sir, next, sir, please. These are the names of 21 varieties from which the pathogen were isolated and the pathogenicity were. And next one, sir. These are the isolated pathogen of Serocladium oryzae, which shows the morphological variations, and the clear morphological variation was there from the isolated pathogen, and its pathogenicity were confirmed by taking the susceptible varieties, that is Swarna. Okay, sir, next, sir. After isolation, the purification were made by repeated isolation of the pathogen, and the isolates were identified on the basis of the standard reports and on the basis of the vegetative hyphae, fillets, and conidia. The vegetative hyphae, that is irregularly knurled outline, is the characteristic feature of the Serocladium oryzae, and the fillets, which is directly arising from the undifferentiated vegetative hyphae, or sometimes from the corydiophore, and they tips tapering towards the end of the corner of the fillets, and there the conidia were arranged in a ball-like manner, and these conidia were cylindrical to sometime oval shaped, and they were blunt ends. Okay, sir, next please. This is the identification feature of the Serocladium oryzae. And for inoculating the varieties, the half-cooked rice grain bits method were used. And these half-cooked rice grain bits, they were gently rubbed over the 7 days old culture, and they were inserted into the boot of the susceptible variety. And all these isolates, they were confirmed by testing the pathogenicity on Swarna variety. And to know the seed-borne infection, the agar plate method was also used. And for that purpose, the surface sterilized entire seed was used for plating on the agar plate and outer circle that contains the 8 seed and 2 at the center. And they were identified on the basis of the colony characteristics under the stereobinocular and by taking the photograph under the microscope. And the Chapti varieties, which is taken for the isolation of the, for knowing the external infection, that recovered the 72.5% infection on the outer side of the husk, and the Bamleshwari that shows the recoveries, that is 65.25%. It means the Chapti varieties that was more susceptible for the infection by the sheath rot pathogen. Okay, next, sir. And for knowing the component plating was also done to know the presence of the internally seed-borne infection also. For knowing the internally seed-borne infection, the component of the seeds were also placed, that is dehusked grain and the husk, they were separately plated on agar plate, and the recovery of the pathogen were also confirmed that the pathogen is externally.
Yeah,
and
please come to the conclusion.
Yes, this, okay, sir. And husk, that is recovered 67%, and dehusked grain, that is 26% in case of Chapti variety, and in case of Bamleshwari, that was 63.5 and 12.5%. Okay, sir, next please. Okay, to know the progress of infection in the seedling and at the boot stage, the 90 seedlings were grown in 30 pots, and they were randomly selected, and they were cut into 2 halves and placed in decolorizing solution. After their decolorization, they were stained with the lactophenol blue and cotton blue, and after their staining, they were plated on the slides. And for the infection, the seedlings, they were not showing the symptoms, but when they were stained with the lactophenol blue, out of the 20 seedlings, the 5 seedlings showed the infection. This confirmed that the pathogen was present in the seedling. Next, sir. And similarly, in case of tillering and boot stage, that was also taken the samples, and same procedure was adopted. And at the tillering stage, 40% of the seedling plants shows the infection, whereas at boot stage, the 50% plant shows the infection, along with the spore mass of the pathogen was also confirmed. And at boot stages, out of 20 plants, the 3 plants were also showed the symptoms. So these all about my study, and the conclusion of this study, that most of the common rice varieties were found infected with the Serocladium oryzae infection, and pathogen showed the variability among the isolates. pathogen recovery was more from seed surface, that was confirmed with component plating. And the last, that is infection progress from seed to seedling, tillering at boot stage, but visible symptoms only observed at boot stage, and maximum infection found at boot stage, that is 50%. Thank you, sir. Thanks to the organizer for giving me such invaluable opportunity. Okay, thank you. Thanks to
Okay,
Chairman.
thank you very much. Now next to the PV Patil. Okay. Please. Hello. Now I request to the next speaker, PV Patil.
Just a minute, sir.
Yes, sir. Okay.
Please unmute Dr. Pankaj Kumar Patil.
Yes, sir.
Yes, please proceed, sir.
Sir, I am audible. I am audible.
Yes, you are audible. Please start.
Okay. Good evening, sir. I am Dr. Pankaj Kumar Patil, and welcome to my presentation. Next slide.
Please come to the direct on the materials method and result.
Yes, sir. So today I am going to present the topic, that is appraisal of different tomato genotypes against the tomato fruit borer Helicobacter parmigiana infestation. Next. Okay. So, sir, tomato is very important vegetable crops which attacks by a number of insect pest species, thrips, aphids, whiteflies, mites, and Helicobacter parmigiana.
Yes, yes.
Hello.
Haji.
Hello.
Haji, start it.
Hello. Okay.
Hello, continue it.
So the Helicobacter parmigiana is able to cause more than 50% damage in tomato crop. So the experiment is carried out at Department of Horticulture, Mahatma Phule Krishi Vidyapith, Rauri during 2016 and 17. This is 2-year data. And in the Kharif season, so recording, this is the showing of tomato genotypes and their seedlings. Next, these are the transplanting of tomato carried out. These are the plots of the screening of the genotypes during Kharif 2016, and this is during the Kharif 2017. So coming to this, so the table shows that this is the mean larval population I have recorded during 2016. So here, the 20 genotypes which I have taken for the study of the screening against the Helicobacter parmigiana. Amongst that, results show that is T1CV raised to oval. This genotype reported the minimum larval incidence, that is near about 0.72%, 0.72 as compared to rest of the genotypes, and the T1CV raised 3 round is shows the blue color, that is the shows the maximum number of larvas as compared to the other 19 genotypes, and the remaining T1CV race 4 oval, race 6 oval, race 9 oval, H86 and 17-1 are at par with each other. Next. Next, please. Next. Next. Okay. So similar type of result found during the 2017, that is the T1CV raised to oval, that genotype is confined the minimum larval population of Helicobacter parmigiana as compared to the remaining genotypes. Next, please. Okay. So this is the full data of the both the years. Next. Next, please. Next. Okay, so this is the graphical presentation of this mean larval population of different germplasm of tomatoes. Next. Okay, so here these are the healthy tomato fruits and these are the damaged fruits due to the Helicoverpa armigera. Next. Okay. So next one, I have taken some observations. Likewise, susceptibility of tomato genotypes against fruit borer on the basis of this number basis. So in that, similar results are found. So T12CV raised to oval, this genotype is confirmed that reported 9.75% of the incidence as compared to rest of genotypes, and the genotype T12CV raised 3 round is found the maximum number of percentage on number basis, that is near about 33%. Next. 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 Okay. So I have taken the data collect on the weight basis also, and results show that the T12CV raised 3 round is show the maximum number of incidence of Helicoverpa armigera as compared to other genotypes. The best genotype among all that is T12CV raised to oval, and the genotypes T12CV raised 4 oval, raised 9 oval. Next, H86 and 17-1 are the at par with each other. Next, please. So this is the graphical representation which shows the incidence of this Helicoverpa on weight basis. Next. Okay. So on the basis of that, the grading is the system of grading given by the Kashyap. So on the basis of that, I have graded these 20 genotypes in different categories that are the high resistant, then moderate resistant, then susceptible, moderate susceptible, and highly susceptible. So among that, the T12CV raised to oval, the genotype is confirmed found to resistant one as compared to rest of the genotypes. And the T12CV raised 3 round, the genotype is found the susceptible one. And T12CV raised 4 oval. Okay, so this is the categorization. Next slide. Okay. So this is the categorization on the basis of percent fruit damage from rating from 0 to 40%. So here, none of genotype is free from the attack of the Helicoverpa armigera, but only the one genotype is T12CV raised to oval. This is found during my studies. This is the resistance on the basis of which confirm with the minimum percent incidence as well as larval incidence as compared to rest of genotypes, and also none of genotypes reported the high susceptible. Most of genotypes is in between the moderate susceptible to the susceptible. Yes, sir. Yes, sir. Next slide. Next, please. Okay. So among the genotype, the T12CV raised to oval genotype notice resistance and raised 4, raised 9, 17-1, and H86. These are the after that better one, or we can say the moderate resistance on the basis of larva number of weight, number basis, weight basis, and larval basis. So the study, the aim and object of the study there to provide the breeder point of view, identify the source of resistance, and that will help in the breeding program to develop a new resistance variety. Thank you.
Thank you, sir.
Thank you so much for giving the opportunity and listening.
Okay, thank you. Now the next, Poonam Rani. Hello.
Poonam Rani, if you are present, write in the chat box. Rajendra RB Patel, sir, please ready.
Okay.
Please, RB Patel, sir, please
unmute.
Unmute Poonam Burman. Isn't sir?
हाँ पूनम रानी है
क्या
Hello.
हाँ जी hello.
Hello. Am I audible, sir?
Yes, yes.
Yes. So I can present my. Cannot share this.
Hello.
Hello.
Poonam Rani is presented.
Okay, okay.
Hello.
Hello, okay, okay. Then next I.
Hello.
Yes, sir. Poonam. Yes, sir. We are sharing her presentation.
Okay.
Sir, अलग नाम थी join.
Hello, sir. Hello, sir.
Yes, ma'am. You start your presentation.
Okay, sir. Good afternoon, sir. Sir, here is Dr. Poonam. Yes, sir. Here is Dr. Poonam Burman, and I am representing my oral presentation on pathogen associated with black point and canal bunt symptoms in the wheat. So, in India, we know that among various post-harvest diseases, the canal bunt and black point is the most important disease. So, I here isolate 13 fungal species representing 8 fungal genera were isolated and identify all these. Sir, next, please. So all these are identified by using keys and monographs. So, sir, please, next. So text provided by several authors as well as from the plant pathology department of PAU Ludhiana. So during the cropping year 2018, 17-18 and 18-19, total 15 varieties were collected, but most susceptible varieties for both the diseases are PBW 509, HD2733, HD3086, HD2967, HD2733, PBW 550, and HD3086. So I done mostly my work on these all, and germinate will be affected in case of only canal bunt, but in the case of black point, there is no much effect of the germination up to the severity point of the 5th. And next, so I here calculate the 13 fungal species representing 8 fungal genera were isolated. Next. So here we see that in case of canal bunt, the seed germination affected up to the last 5th point at the severity. There is very less, up to the only 27% germinations of the seed, and there is no more effect on the germination of the black point. Next. So in case of in this case, I will take the two method. One is the blotter method, and next is the agar plate method for the good frequencies of the fungus. The result also revealed that mean percent infection of the Alternaria alternata is 39.3, is found in maximum frequency on wheat seed, high in blotter paper, and Alternaria 37.6 was more frequently isolated fungus on agar plate method. Here I use two method because I found that blotter found to be the most efficient, economical, and international reliable method. And I found that there are the 13 fungal species with their different characteristics. Next, like the Penicillium notatum, culture has greenish orange, produce ascospores in either a closed fruiting body or spore balls, and Penicillium carcinogenum reproduce by forming dry chains of spores from brush spade conidiophores. Curvularia lunata appears as shiny, velvety black, fluffy growth on the colony surface. Fusarium oxysporum white cream culture forms sporodochium, mycelia, and conidia, which are rod-shaped, visible on the slides. And also there are Helminthosporium sativum. The conidia are characterized being very darkly melanized and having multiple pseudosepta. Rhizopus stoloniferia, zygosporangium structure of rhizopus also observed. So there are sexual reproduction also reappear. Study of carnal bunt pathogens. The present study result revealed that as we compare of healthy seeds with infected seeds, all parameters like seed germinations according to the disease severity, they are decrease. And I found that on the PDA media and potato dextrose yeast agar were used to multiply the pathogen isolated from the infected grains of the wheat as described in material and method. Two media were used, but potato dextrose yeast agar observed good for growing culture as compared to the media PDA. So the growth of fungal colonies in case of PDYA media were grow fast and clearly seen after the 7 days. Fungal colonies of Teichia indica on both slant observed at 19 degrees centigrade, and from both media the fungal colonies were critically examined both morphological and microscopically. So here we see that primary source of the infection is the only primary spreadia, and the main infection of the kernel bunt is the secondary spreadia, which are of two types, filiform and allantoid, which are responsible for the secondary infection. So this information is very good for the plant breeders as well as for the plant pathologists to further the study of the kernel bunt and black point. Thank you, sir.
Thank you very much, madam.
Thank you, sir. Welcome.
RV Patel, sir. RV Patel, sir, please unmute.
Yes, yes.
And sir, directly go for methodology and result.
Yes, yes.
Chalu ji, Raju bhai, continue.
May I audible?
Audible, visible, continue.
Yes. A field experiment entitled Screening of Pigeonpea Genotypes Against Port Borers and Port Fly in Natural Field Condition was conducted at National Agriculture Research Project Bharuch during Kharif 2016-17 to 18-19. A set of 30 genotypes was shown during all these 3 years, and the larval count of Helicoropta and Maruca vitrata was recorded from randomly selected 5 plants. The number of maggot of Melanogaster obtusa port fly was counted from randomly selected 10 ports. Further, the total number of damaged ports were counted from 5 plants, and they were converted into the percentage. Now the percentage of pest susceptibility rating was worked out by the formula given as per the Abbott 1925. The pest susceptibility rating 1 to 9 is given, and from on the basis of this, next, the 3 years pooled result revealed that the highest Helicoverpa larval count was found in the Upas 120, and it was at par with most of the genotypes except BP Sol 16-256, BP 15-21, BP 16-256, NPAK 15-14, and ICPL 87, NPEK 15-25. The lowest number of Helicoverpa larva count was found in the genotypes BP 15-11. Next, please. The Maruca count, highest Maruca count was also found in the entry Upas 120, and it was found at par with most of the treatment except AGT2 and BP 15-11, BP 15-21, NPAK 15-09, NPAK 15-11, and NPEK 15-4. The lowest number of larvae was found in the SSN's GJP 1303. Next, please. Based on the pest susceptibility percent and PSR rating, the genotypes or SSNs which has counted less than 5 ratings are considered as moderately resistant, and in this against the port borer infestations, and in this category the AGT2, BP 15-11, SKNP 1413, GJP 1303, NPEK 15-25, and NPEK 15-03 were considered as moderately resistant against port borer and port fly, whereas UPAS 120 is considered as highly susceptible, and BP 16-257 was considered as a susceptible ones against port border infestations. Next, please. Similarly, from the 2 years results revealed that the port fly count was found highest in the SSN's BP 16-256, and it was at par with most of the treatment except AGT2, NPK 15-09, and BDN 711 and NPK 15-03. The lowest number of maggot count was found in SKNP 1413. Next, please. On the basis of pest susceptibility percentage and PSR ratings, the entries, namely AGT2, BP 15-21, BP 15-09, and KNP 135, BDN 71111 were considered moderately resistant against the port fly. Among 30 genotypes screens against the port border, the SSNs, namely AGT2, BP 15-11, SKNP 1413, GJP 1303, BP 15-23, BP 12-06, NPEK 15-09, ICPL 87119, and NPEK 15-25, NPEK 1503 were found moderately resistant against the port border complex, where against the port fly, the SSNs, namely GJP1, AGT2, BP 15-11, BDN2, BP 16-256, GT103, BP 16-261.
Okay, Raju bhai.
Okay.
So very good.
Okay, thank you all of you.
Now I request to Shivangi Kancharia. Hello.
Sir, Dr. Kalpit Shah from Junagadh.
Okay.
Dr. KD Samuel.
Okay, sir. Am I audible to you, sir? My screen is
Audible and visible. Please continue, sir.
Okay. Good evening to all. So the research paper on susceptibility of green gram varieties or genotypes against thrips, that is Cetothrips dorsalis, infesting the particular green gram crop. The content has been divided into introduction, need and aim, materials and methods, result, discussion, summary, as well as the future thrust. As we all are knowing that pulses are expected to touch 29.30 million ton by 2020. So it is a continuously need to look after these pulses, and it has this much quantity of this digestible protein, fat, calcium, and phosphorus. Looking to the insect pest that they are damaging, more than 65 different species they are feeding on the green gram crop, and they are considering the losses of about 73.86%. Looking to the insect pest, among the different sucking insect pest of green gram, thrips, that is in the initial growth of the period of the crop, that is the major concern of the Saurashtra region. And the damaging symptoms, it is visible in the figure also, that drying up of the smaller and tender leaves, and which hinders the vegetative growth and development of the green gram crop. So that is basically the research has been carried out for the Saurashtra region, and considering the population has been built up in the early stages. And looking to the identification of the pest is based on the observations and morphological characters, it was found that is Cetothrips dorsalis, it is damaging on the green gram crop. Need of the research, that is this resistance and susceptible varieties or genotypes, that is useful for minimizing the damage of thrips and so the yield losses. So the aim to find out the susceptibility of green gram varieties and genotypes against the thrips, it was commended. Looking to the materials and methods to study this susceptibility trial, different 10 varieties and genotypes has been taken, and with the spacing of 2 lines of each variety, and recording the observations from the 5 plants per line, and the observations was recorded from 3 leaves from upper, middle, and lower portion of the plant during early morning at the weekly interval, and the obtained data were analyzed for least susceptible genotype against the thrips, as well as the seed yield was also recorded. This is the experimental site and the experimental plot. Looking to the materials method is continue that scale we have used based on the reference of Patel. He has taken the reference from the coconut plantation. The category has been divided from highly resistant to highly susceptible. Result and discussion point of view, the infestation of thrips in green gram varieties, and looking to the data, this EC 501566 that recorded very low number of thrips populations, followed by IPM 2057, and our variety GM4 that recorded the highest thrips populations per leaf during the experimental period. And looking to the data of the yield, the highest yield was recorded from genotype EC482907, followed by genotype EC501566. And our GM for the developed variety, it has also recorded the second highest yield, that is 600 approximately kilogram per hectare. This is the graphical presentation of this infestation of thrips. So, and the looking to the categorization of the green gram varieties and genotypes, not a single variety has been comes in the highly resistance and resistance perspective in the category, but the IPM 2057 and GM4, these both are considering into the moderately susceptible, and this IPM 2057 is comes in the moderately resistance variety. So in all the summary, the lowest thrips incidence was recorded in EC 501566, that is 0.8 thrips, and IPM 2057, that is 1.10 thrips per leaf of green gram, and was categorized as moderately resistance, while the highest thrips population was recorded in GM4 and variety, and it was categorized into moderately susceptible against the pest. Looking to the future thrust, that needs to study the morphological characters as well as biochemical characters which imparts the resistance to this insect, and need to understand the genetic mechanism which will helpful to the plant breeders and all. Thank you. So it is now a game of chess with the insect, whether it is comes only with the varieties or we have to go for the insecticides also. So it is a continuous process. Thank you all.
Okay, very good presentation. Now any query?
Any questions?
Hello.
Yes, sir.
Jyoti.
Hello. Now Shivangi Kanchara, please presentation for you.
She has done already, doctor.
No, sir, she has not submitted and even not present.
Okay, sorry, yeah, list में कोई बात.
Shall I present myself, Dr. Jyoti Jain?
Yeah, you continue, ma'am.
Okay, thank you.
Okay.
Wait a moment, I am sharing.
Okay, no problem.
Share just wait a moment, I'm finished.
No.
Am I audible?
Yes.
Okay, good evening, everybody. Myself, Dr. Jyoti Jain. I'm working as plant pathologist with Department of Plant Breeding and Genetics at PIU Ludhiana. And I'm here to give oral presentation of my research work on identification of slow blasting donors among basmati rice genotypes against neck blast caused by Pyracularia oryzae cavara. So as we all know that the rice blast, it attacks different organs of the rice plant, and the neck blast is the most devastating, which is a global threat. And according to the Commonwealth Mycological Institute, now it has been reported from more than 85 countries of the world on more than 80 gramineous host plants, and losses up to 20 to 100% have been reported from
Sorry for the interruption, but go for your objective methodology and result.
Yes, sir, I'm coming. Just give me a single minute.
Okay.
Yeah. So besides these all factors, the major factor regarding the rice blast fungus is that it is highly variable, leading to the formation or the evolution of the races at a very short period of time. And if we look into the literature, lot of literature is there regarding the resistant varieties which are present to the leaf blast. And most of the breeding programs, they are they are basically concentrated towards breeding for leaf blast resistance by assuming that that resistance will further convert to the neck blast resistance. But actually that is not happening in the field. So these are the symptoms. So actually why I decided to choose this problem, because when we look in the scenario, the orange colored part shows that the earlier the disease was present only in the submontaneous tracts, but with the introduction of the susceptible varieties, particularly the PUSA 1121 and the PUSA 1509, and the increased acreage under these varieties, the southwestern districts shown with the red colored spot became the hotspot area. And moreover, you must have read in the newspapers regarding the rejection of the basmati consignments from the European countries. That is again due to the fact that since the neck blast, it appears near the maturity, and the farmers in the panic, they are spraying the unrecommended fungicides at a very late stage. So that is why the fungicide residues, they remain within the seed. So the consignments were cancelled, which caused a huge loss to the farmers as well as to the economy of the country. So this problem was decided basically to identify the donors for which have the slow blasting character or which are partially resistant to the neck blast disease. So this is how the pathogen was isolated. So in this study, the most virulent isolate identified in our previous studies was used. So the for the mass multiplication of the pathogen and for artificial inoculation of the pathogens, we need the mass culture. So this is the study which was lacking internationally, and we have developed a technique at PAU known as the bit wrap technique, where we are using the grass bits of this paspalum grass to artificially inoculate the necks of the basmati rice plants. So this is how we inoculate the necks of the rice plants at maximum tillering stage. Not at maximum tillering stage, after ear emergence stage. So the bits are wrapped around each neck, and the data is taken starting from the 4 days to the 15 days after inoculation.
Ma'am, sorry, 2 minutes remaining.
Yeah, I will complete within the stipulated time. So a set of 45 aromatic and basmati germplasm lines, they were artificially inoculated with the pathogen culture using the bit wrap technique, and the lesion length was observed. So that from that lesion length, the area under disease progress curve was calculated, and the genotypes, they were designated into different categories as given in the table. So from the AUDPC, further the susceptibility index was calculated, and according to the susceptible index values, again the genotypes were categorized into different categories. So this is the table which is showing various parameters for the different varieties in terms of AUDPC, RAUDPC, apparent rate of infection, susceptibility index, and so on. So based on these factors, we could find under artificial inoculation conditions that only 4 genotypes, they were showing moderately resistant reaction, and none was found to be resistant, and only 3 were moderately susceptible. Similarly, these genotypes, they were evaluated at CSHK Wheat and Rice Research Station Malan, which is a hotspot for neck blast. So similarly here also we got these introgression lines INGR 15001 and 15002 as the resistant. So from this study, we found that the donors which are promising among the moderately resistant category are TTEP, Injure lines, and the PUSA Basmati 1637, which has been already released for cultivation in Punjab state. And the moderately susceptible were some aromatic lines like Nagra, and the basmati lines like Sion Basmati. So these donors, they have already been enrolled into the basmati breeding program at PAU, and may further can be used as a donor in basmati resistance breeding program. So I would like to thank my co-workers, Dr. Sandeep Jain, Dr. Lore, Dr. Navjot, and Dr. Sachin Upmanyu. And thank you for giving me an opportunity to present my work in this national symposium. Thank you so much.
Interesting, ma'am. Just for the curiosity, is there any planning regarding blast of wheat screening?
Blast of wheat?
Yeah, it is upcoming. This is nowadays.
Yeah, that is upcoming disease, but as the decision has been taken at by the Directorate of Wheat Research that we do not want to introduce the pathogen in India, so we are not conducting any studies on wheat blast yet.
Okay. Thank you very much, ma'am.
Yeah. Any other question?
No question. Next.
Thank you so much.
Hello.
Rathod AJ. Sure. Rathod AJ, if you are present, just send me the one message in chat box. Rathod. Maybe not present. Sanjay bhai, Bhumi. Unmute yourself.
Yeah, sir.
Bhumika.
हाँ, sir.
Okay, now start.
Come to the methodology and objective and result only, Bhumi. You have only 2 to 3 minutes. Okay.
Okay, sir. Okay, sir. Good evening to Chairman, sir, Co-Chairman, sir, and other participants. Today I am going to present my poster presentation topic on screening of potato varieties genotypes for resistant against root knot nematode Meledogani incognita. My poster presenting objective is identification of resistant sources against root knot nematode Meledogani incognita in potato crop. First of all, methodology. In methodology, one pot experiment was conducted on screen 40 different potato variety or genotype made available by the Potato Research Station DISA against root knot nematode Meledogani incognita to locate the source of resistance during the Rabi season in 2017-18 at the Mist House, Sardar Krishnanagar, Dantewada Agriculture University, Department of Nematology, CP College of Agriculture. The plastic pot having 24 centimeter diameter were sterilized with 4% formaldehyde or formalin 40 EC and filled with 10 kg Meledogani incognita infested soil having initial nematode population of 120 juvenile per 100 gram soil. The nematode-free potato tubers weight ranging from 91 to 95 gram of each variety or genotypes were shown in different pots. The experiment was conducted using completely randomized design with 3 replication. Pot were put in net house.
Come to the result. Come to the result. Do not read out each and every sentence. Come to the result.
Okay, sir. Result 40 potato variety or genotype skin in pot condition 10 kg soil with initial nematode population, and I find that root knot index on root and root as well as tuber, the variety Kufri Anand was highly resistant in both, but in tuber cases, other 2 varieties Kufri Sinduri and MS 1906 were found highly resistant, and we can see in the table Kufri Anand is highly resistant on root, but Kufri Sinduri and MS12 1906 and Kufri Anand also resistant on root as well as tuber. Based on the root knot index on root as well as tuber, total 14 variety or genotype were screened against root knot nematode Meledogani incognita. Among them, variety Kufri Anand was highly resistant. Thank you, sir.
Okay, thank you very much.
Okay, thank you very much.
Now, Rajesh Bhai.
Yes, sir.
Now, any presentation for the poster? Next.
Now this is the last call for the Priyanka Kumari Meena.
Yes, sir.
Is he Rathod? A.R. Rathod. If A.R. Rathod, Priyanka Kumari Meena is present, please. NB Pawar.
Yes, myself is present.
Okay. Sir, Kalpana ji Rathod is joined.
Okay, I am audible. Excuse me.
Yes, sir, you are audible. Please continue.
Okay. Myself, Dr. N.B. Pawar, working as Assistant Professor at College of Agriculture, AU Vaso. I am going to present a poster on influence of inoculum density of Rhizoctonia bataticola isolates, especially weakly pathogenic isolate and highly pathogenic isolate on chickpea root rot incidence. The objective of this study is only to ascertain the different inoculum levels on different varieties like resistant variety, moderately susceptible variety, and susceptible variety, highly susceptible variety. As we all know that this for the chickpea production, these soil-borne pathogens, especially the Fusarium and Rhizoctonia, they are being the most menace for the cultivation, and the losses due to this Rhizoctonia bataticola may goes up to 70% at full podding stage. And being considering the soil-borne nature of this pathogen, Rhizoctonia bataticola, the inoculum level should be ascertained to check the to check the population of the pathogen. The methodology included for this experiment, for that the 15-day-old inoculum of virulent and weak pathogen isolates of Rhizoctonia bataticola were multiplied on 9:1 same sorghum medium in a conical flask, and it was multiplied at the proportion 1 gram, 5 gram, 10 gram, 25 gram, 50 gram, and 100 gram inoculum per kg of the soil, and the inoculum was in the soil, and the pots were kept in a randomized design.
Sir,
and this experiment.
Sorry for the interrupt. Please come to your results, sir.
Okay. Now the results from the experiment, it is being found that the irrespective of increase in the inoculum levels of especially the weakly pathogenic isolates of Rhizoctonia bataticola, the seedling mortality was not increased in resistance cultivar and moderately cultivar, except the susceptible cultivar, that is JG62, which is a susceptible one, recorded 10% mortality when the inoculum level of 100 gram per kg soil was applied, and this mortality was noticed at 60 days after sowing. And at that time, the final CFU count that was recorded is 303 propagules per 50 milligram of the soil. But in case of the highly pathogenic isolates, the increased level of inoculum will result subsequently in increase in the percent mortality of JG62 susceptible variety, and 100% mortality was observed when 100 gram per kilogram soil inoculum is being applied. And in case of the moderately susceptible and high resistance variety, that is AKJ 96, it is a moderately susceptible, and Saki 9516, it is a resistance variety. They recorded the minimum mortality, especially that is the 13.3 and 7% respectively at 100 gram inoculum per kg of the soil. And the final propagules that are being observed when 50 milligram of soil at 60 days after sowing increase with the inoculum level and attains maximum, that is 321.3, 314 and 308 milligram, 8 milligram per 50 gram, 8 propagules per 50 milligram of the soil, and in the pots JG62, AKG 46 and Saki 9516 respectively at 60 days after sowing. From this, and in the one experiment, in the above treatment, 6 treatment, 1 treatment was included, that was the absolute control, in which none of the inoculum is being added, and in that inoculum there was no mortality of the plants, chickpea plants was being observed. From this experiment, it has been concluded that the severity of chickpea root rot increased with increased inoculum level of Rhizoctonia bataticola. Also, the rate of multiplication of propagule count decrease with increased inoculum of both isolates, that is weakly pathogenic isolates and highly pathogenic isolates of Rhizoctonia verticolor. Thank you all of you. That is everything from my side. Is there any question? Welcome, please.
Okay, thank you.
Thank you very much. Now, Rakhulia sir, over to you. Sir, Rakhulia sir. Rakhulia sir. ये मन म्यूट कर दीजिए राकुल साहब ने क्या राकुल सर
Haji, now any more for the poster presentation?
No sir, second number, fifth number and ninth number, they are the absent. Okay.
So only okay.
Only two poster presentation.
Only 2 and 5 for the oral presentation. Okay.
Yes sir.
Okay, now thank you for my co-chairman Dr. R.F. Chaudhary sir and Dr. Rajesh for help me for this session. Thank you very much.
Thank you very much, Chairman Dr. K.B. Rakhulia sir, Co-Chairman R.F. Chaudhary sir, Reputed CM Bhalia sir, Dr. N.B. Pawar sir. Thanks for the smooth and the comfortably handling this session. Thank you very much.
Thank you. Thank you.
Now today's all sessions are completed. We will meet tomorrow at 8:30 sharp. Sharp 8:30.
Okay.